So all this is Dr. Mubin Sayed from drbeen.com. We have the FDA's live committee meeting going on for Novavax. So I thought we would listen in together, and every once in a while I would definitely take a break as well. This is the first time I'm doing it, so my apologies if it sounds clunky. I think it will be fun. I'm going to share it with others as well. So give me a minute. Tell me if you can hear it. Let me see if I can increase the volume. Currently, there are 3 COVID-19 vaccines available in the US for use in individuals 18 years of age and older. Pfizer-BioNTech COVID-19 vaccine, a mRNA vaccine licensed as Comirnaty. Moderna's COVID-19 vaccine, another mRNA vaccine licensed as Spikevax. Janssen's COVID-19 vaccine, not licensed but available under EUA. Use of Janssen COVID-19 vaccine is limited to individuals for whom other FDA-approved or authorized COVID-19 vaccines are not accessible or clinically appropriate, and individuals who elect to receive the Janssen COVID-19 vaccine because they would otherwise not receive the COVID-19 vaccine. So here is today's agenda. After my introduction, Dr. Vedar Kovi from CDC will give you an overview of current epidemiology of COVID-19 and COVID-19 vaccine-related threats in the United States, followed by Dr. Tom Timabukolo from CDC will give you an overview of COVID-19 vaccine-associated myocarditis, followed by sponsor's presentation, emergency use authorization request by Novavax for a vaccine to prevent COVID-19 in individuals 18 years of age and older. There will be a short break, followed by Dr. Lucia Lee, the lead medical officer from Office of Vaccine at CBER FDA, who will present FDA's review of effectiveness and safety of Novavax COVID-19 vaccine adjuvanted in individuals 18 years of age and older. There will be a 45-minute lunch break, followed by open public hearing. There will be a short break, and then additional question and answering session regarding the sponsor and FDA's presentation, followed by committee's discussion and voting, and then meeting will be adjourned. So here is the voting question for the committee. Based on the totality of scientific evidence available, do the benefits of Novavax COVID-19 vaccine adjuvanted when administered as a 2-dose primary series outweigh its risk for use in individuals 18 years of age and older under EUA? So that is the question that they need to answer for or vote on.
And thank you, Dr. Sen. You've given us a good overview of the entire day's proceedings. We have a few minutes now, and if the committee has any
Doug, I see your comment
about the guidance, about EUAs and the rationale for emergency use authorization, you can raise your hands now. Okay, Dr. Rubin, is that your hand raised? I'm not seeing it in green here, but
yeah, that's me, Doctor.
So if I reduce the volume, can you still hear it? What I do not know is if my system can internally route the audio. So can you hear it?
Dr. Chen, there are 2 approved and 1 authorized vaccine out there. So I'm curious as to how this meets the criteria for a product that for which there is a necessity given the existing product.
Okay, so I'm going to increase the...
Dr. Mark, would you like to respond?
Yeah, I'm happy to, or Dr. Fink can. Trying to get my camera on here. There we go. Thanks very much for that question, Dr. Rubin. The statute says it allows us some leeway because it gives us the ability to look to have products that are either they would fulfill some unmet need, and in this particular case, although we have mRNA vaccines out there, we have the Janssen vaccine out there. The Janssen vaccine is currently not being used as a frontline vaccine the same way as the mRNA vaccines, which leaves the issue of vaccines for those who might not want to take an mRNA vaccine because of concerns they might have with an mRNA vaccine as needing potentially an alternative, having a protein-based alternative may be more comfortable for some in terms of their acceptance of vaccine. And I will use this as a moment on the bully pulpit to say that we do have a problem with vaccine uptake that is very serious in the United States. And anything we can do to get people more comfortable to be able to accept these potentially life-saving medical products is something that we feel we are compelled to do. Does that answer your question?
That does. That's great. Thank you very much.
Thank you, Dr. Mark.
Thank you. Dr. Levy.
Hello. Thank you for that helpful introduction. If I understood correctly, there have been some concerns with the manufacture of the protein that is the basis of this Novavax vaccine. And for that reason, some of the data from some of the other international studies will not be considered with regards to vaccine efficacy and immunogenicity today. My question is this: could FDA say a few words about what the nature of that manufacturing problem was? And also, are we as a committee to assume that these issues are completely solved now and that the latest version of the way the protein is manufactured will not lead to any manufacturing problem?
So my mic is such that it can only direct voice from one side. So when I put it this way, I cannot speak very well. So hopefully now you can hear the
No, Dr. Manto, thank you. I think we will discuss that during question and answer session. We can discuss a little more about that. So I'll pass it on now.
Okay, thank you. Dr. Gallen.
So team, my mic is unidirectional. So either me or the speakers. So I'm going to put it on the speakers at this time.
We can't hear you.
Muted.
Yep, you're muted, sir, on your phone.
Okay. Got it. Thanks. Sorry. So about dosing, we're asked to review the safety and efficacy of a 2-dose schedule. If you wind back the clock, that's how this all began, and we learned subsequently that 2 doses was not really the full need. And then with this confusion about what's a booster versus a 3rd dose. Maybe we're likely to get into this later, but what are we going to be doing about more than a 2nd dose? And then a related piece is that this is entering a marketplace with other vaccines. And while there may be some who've been waiting for this as their only vaccination, there are others who might want to think about how they optimize their own immunity with mixing, matching with other things. So hopefully we can hear something and learn something about that. Thank you.
Again, it's up to you whether you want to answer these questions now or later.
Dr. Manto, Novavax has submitted the booster dose data. And once we complete this primary series, FDA is going to review those data and we'll discuss that in future.
Finally, Dr. Marasco, a very short question. We're run out of time.
Yes, it's a real question to Dr. Marks and Siva, really. It's a follow-up to Dr. Rubin's question. So this vaccine that we're going to hear about today is adjuvanted, and I'm curious in terms of fever. Have you guys really, considered, what the public is hearing and seeing, which is waning immunity, and is there any emphasis in this particular vaccine on the fact that it's adjuvanted and
It sounded so
maintains
dark.
the durability of the response?
Dr. Marks?
Yeah, no, thanks for that question. I think that'll be something for the committee to discuss today, and I think the sponsor may be presenting some information on that as well. There's the issue of durability of response, as well as the breadth of protection, which I think are both things that will be open for discussion.
Thank you all. We've heard some questions which we need to park, and we can bring these up later on as we get into discussions of the substance that we're going to be handling today. Now let's get back to the background, which I jumped to before, and I'd like to reintroduce Dr. Scobie. Dr. Scobie, tell us about COVID vaccination rates in the United States.
Good morning. Can you hear me?
We can.
Great. The Omicron variant has been shown to have increased transmissibility, but decreased severity relative to previous lineages.
How is this?
Omicron has many mutations in the spike gene, including 15 mutations in the receptor binding domain, as shown in the picture on the right. These mutations are associated with reduction in the efficacy of some monoclonal antibody treatments and a reduction in neutralization by sera from vaccinated or convalescent
Laura, thank
individuals.
you.
This is a graph of the number of SARS-CoV-2 sequences submitted globally to the GISAID public genomic data repository since Omicron was first detected at the end of November 2021. The blue color shows the Delta variant being displaced as the Omicron BA1 sublineages in salmon color quickly rose to predominance, followed by the rise of the Omicron BA2 sublineages in peach. The other Omicron sublineages like BA4 and BA5 are not readily apparent in the figure because they are still a relatively low proportion of submitted sequences. The total number of submitted sequences globally has shown a declining trend since January of 2022. This stacked bar shows recent U.S. trends in the national weighted estimates of variant proportions and nowcast projections of circulating SARS-CoV-2 lineages by week of specimen collection date from CDC's COVID data tracker. Omicron sublineages depicted in different purple and pink shades have been over 99% predominant since late January. The BA1.1 sublineage in dark purple was gradually displaced by the BA2 sublineage shown in lavender, and more recently the BA2.12.1 sublineage in pink, which was 59% of circulating lineages as of the week ending May 28th. BA4 and BA5 are not shown in this graph because they were less than 1% for this period, but these sublineages will be shown in the variant proportion estimates released later today. This map shows the relative proportions of BA2.12.1 in pink, BA2 in lavender, and other Omicron sublineages in the darker purple shade across the 10 Health and Human Services subregions. You can see that BA2.12.1 is at least 50% predominant in all regions, except region 10 in the Northwest. This graph shows the trends in daily numbers of COVID-19 cases reported in the United States since the beginning of the pandemic. The number of cases associated with the Alpha variant were relatively small compared with the Delta variant and then the Omicron variant. Nationally reported cases show increasing trends in April and
You're welcome, Ryan.
May, though this trend may be starting to turn in the last week or so. Reported cases still remain relatively high. I note that the number of reported cases is likely underestimated due to the increased use of at-home tests, whose reports are mostly unreported to public health departments. As of June 5th, there have been over 84 million cases of COVID-19 reported in the U.S.
It's 6:17 a.m.
This is a
here.
graph from a recent MMWR and CDC's National Commercial Laboratory Seroprevalence Study
Coffee time.
and shows trends of infection-induced SARS-CoV-2 antibodies by age group. These results do not include anti-spike antibodies from vaccination, nor do they reflect the percentage of the population that might have sufficient antibodies to be protected from reinfection. The percentages of people with previous infection noticeably increased following the rest of the Omicron variant. Greater seroprevalence was noted in younger age groups, likely related to these groups being eligible for vaccination in later months and different exposure risks compared to older age groups. National seroprevalence during February 2022 was 58%. This graph shows the trends in the daily number of reported COVID-19 deaths in the United States since the beginning of the pandemic, including during the waves associated with the Alpha, Delta, and Omicron variants. Even though the Omicron infection is less severe overall relative to Delta, the number of deaths related to Omicron was relatively high because Omicron case numbers were very high. As of June 4th, there have been over 1 million deaths due to COVID-19 reported cumulatively in the U.S. These are the weekly trends in COVID-19 associated mortality rates by age group. The data show that higher mortality rates are consistently observed in older age groups, most notably on this graph among ages 75 plus, 65 to 74, and 50 to 64, as shown in the purple and pink colors. These are the weekly trends in the rates of new COVID-19 inpatient admissions by age group. Similar to the previous graph, you can see higher hospitalization rates in the older age groups, with patients 70 plus in purple, 65 to 74, and 50 to 64 years in the pink colors having the highest admissions rates, followed by other adult age groups in shades of blue. As of June 2nd, more than 221 million people in the US have been vaccinated with a primary vaccine series, which is 71% of the eligible population aged 5 years and older. There are also over 103 million people, or 49% of the population aged 12 years or older, who have also received the first booster dose, and about 15 million people, or 23% of the population aged 50 years and older, who have also received a second booster dose. This graph shows trends over time and by age group and the percentage of people who have received at least a primary series on the left and a booster dose on the right. In both figures, vaccination coverage is higher in older age groups, indicated in the purple and pink colors. We can also see that coverage with the primary series for ages 5 to 11 years, shown with yellow and the dotted line, a yellow dotted line on the left, is still relatively low at 29%. Booster dose coverage on the right remains under 50% for age groups less than 50 years, shown in blues and yellows. From data reported to COVID Data Tracker, over 230 million, or 89% of US adults ages 18 years and older, have received at least one COVID-19 vaccine dose. Using these data and census data, we can estimate that there are about 27 million adults who have not yet received a vaccine at this time. I'll also note that most adults aged 65 years and older have already received at least one COVID vaccine dose. This is data from the National Immunization Survey on adults who have not yet received a COVID-19 vaccine by age group, race, and ethnicity. Across the age groups, we can see that people of non-Hispanic, other or multiple races, and non-Hispanic white people have the highest percentages remaining unvaccinated, while Hispanic and non-Hispanic Black people have the lowest percentages remaining unvaccinated. Next, we're going to shift to consider surveillance monitoring of rates of cases, hospitalizations, and deaths by vaccination status. CDC collaborates with 31 public health jurisdictions, representing 70% of the US population. These jurisdictions actively link case surveillance, immunization registry, and vital registration data to monitor rates of COVID-19 cases and deaths by vaccination status. CDC also tracks rates of COVID-19 hospitalizations by vaccination status using COVIDNet, which is a population-based sentinel surveillance system in 99 counties in 14 states, representing 10% of the US population. In addition, CDC's vaccine effectiveness studies allow for more robust analyses as compared with surveillance and a better understanding of how well vaccines are working. We also have detailed data on serious illnesses in vaccinated persons through COVIDNet, as well as electronic health records and vaccine effectiveness platforms. This slide shows the age-adjusted rates of COVID-19 associated deaths by vaccination status and receipt of booster doses. Unvaccinated people in all age groups have had higher mortality rates than people who received a primary series alone and people who also received a booster dose, including after Omicron became the predominant variant. In March, unvaccinated people ages 12 years and older had 17 times the risk of dying from COVID-19 compared with people vaccinated with a primary series and booster dose. This graph shows age-adjusted rates of COVID-19 associated hospitalizations by vaccination status and receipt of a booster dose. Hospitalizations for COVID-19 were higher among unvaccinated than vaccinated people over time, including after Omicron became the predominant variant. In March, unvaccinated adults ages 18 years and older had five times higher risk of COVID-19 associated hospitalization compared to those fully vaccinated with a booster dose. This slide shows age-adjusted rates of COVID-19 cases by vaccination status. In April, unvaccinated people ages 5 years and older had a 2 times higher risk of testing positive for COVID-19 compared to fully vaccinated people overall. Various studies have shown that severe COVID-19 illness is relatively rare among vaccinated people compared with unvaccinated people. Compared with unvaccinated people, fully vaccinated people with severe COVID-19 illness are more likely to be older, be long-term care facility residents, and have underlying medical conditions including immunosuppression, diabetes, and chronic kidney, lung, cardiovascular, and neurologic diseases. More than 75% of people who are fully vaccinated and get severe COVID-19 illness have multiple risk factors. In summary, CDC continues to monitor emerging variants like the BA.2 sublineage of Omicron, including their prevalence and impact on disease incidence and severity over time. Monitoring trends and rates of cases, hospitalizations, and deaths by vaccination status has been helpful for monitoring the impact of different variants. And finally, currently authorized vaccines offer protection against infection, severe illness, and death, so it's important to stay up to date with vaccinations, including receipt of first and second booster doses in eligible populations. Thank you, and I'd like to acknowledge those people for their contributions.
Thank you, Dr. Scobie. We have a few minutes for questions specifically concerning the presentation about where we are with COVID, with the variants, and with vaccination. And let's stick to those topics. Dr. Chatterjee.
Thank you, Dr. Arnold. Commander Scobie, my question is with regard to long COVID and whether you have any data to share with us on the impact of the vaccines on long COVID.
No, unfortunately, I don't have data on that today, but I might ask Lieutenant Commander Ruth Linkellis, if she's on the line, if she has any data on from vaccine effectiveness studies or any other related data that she wants to share. Are you there, Dr. Linkellis?
I'm sorry, who are you looking for, Heather?
Ruth Linkellis. Do you see her on the line?
Mike, can you
No. Go ahead, Prabha.
Okay,
Yeah,
but I think we'll just move on. I'm sorry, Dr. Chatterjee, but this will probably come up later again. Dr. Perlman.
Yeah, I just had a question about one of the first figures that you showed. The data showing the high mortality in 70 people over 75 with the Omicron. Were those? Do you know if those people were vaccinated? Would this be justification for different vaccines, or were those people mostly unvaccinated? Or were they just people who had many comorbidities? Do we know about antibody titers in them? Trying to get a sense for how the Novavax vaccine could fit in this.
Yeah, that's an interesting question. So the data I showed are actually surveillance data. So we don't have detailed information in that system on vaccination status. And we don't have like titers like you were asking about. There are studies like vaccine effectiveness studies that would have more detailed information. And the data that I showed you on vaccine breakthrough surveillance, the rates of cases, hospitalizations, and deaths by vaccination status, those data are collected by age group. And even in the older age groups, we do see a very large disparity in unvaccinated people having higher rates of, hospitalization and death, regardless of age group. So although it may be true that, as I was saying, that if you are older, of older age and you have underlying conditions and you happen to have a breakthrough infection, you will be more likely to have a serious event compared to people who don't have those risk factors. It's still very much the case that adults and children alike are protected against serious illness with these vaccines.
Thank you.
Thank you.
Dr. Reingold.
Hi, can you hear me?
Yes.
Yes.
Thanks for that, Heather. Reingold, quick question. So I know perhaps the most recent data you don't have quantified yet, but the eyeball test, looking at those graphs of rates of either hospitalizations or deaths by vaccination status would suggest they're converging more recently, and that the VE is in fact shrinking in the most recent time period. And I'm just curious if you have any thoughts about that. Whether the eyeball test.
I mean, so the eyeball test is challenging in this situation because the Omicron peak at the beginning of the year was so high. So it's really throwing the y-axis off, it's scaling everything down. And then in the data that I showed you on more serious outcomes, that was during a period of relatively low incidence in the US. So it's pretty hard to see what's going on in those graphs. But it is true what you're saying, that as different variants have come through, notably the Delta variant, and then the Omicron variant. We have seen evidence in our rate data that suggests a decrease in vaccination effectiveness. We don't choose to calculate vaccine effectiveness using those data because of our inability to control for other factors besides age. But it's definitely true what you're saying, that vaccine effectiveness has been reduced related to different variants. Ruth Link-Ellis, I don't know if you're able to connect her now, but she was my expert that I had on about VE, if she wants to say more.
Let's, for technical reasons, I think it's very difficult to link other people on. So let's go on, and Dr. Gellin has his hand raised.
Heather, thanks for that. Can you hear me?
Yeah.
Two things. You used the term fully vaccinated several times. We want to know how you define that. And maybe this is for later. When we think about epidemiology of COVID infection, will we be hearing either now or later background rates of myocarditis from natural infection? Thanks.
We have a talk coming up, Bruce, on myocarditis.
Yes, that's the next talk about myocarditis. Let's see. And fully vaccinated is a term that still exists, that's defined as vaccinated with a primary series. And it's challenging to understand, so we make attempts to not use it. But for whatever, it still exists in the literature and it still exists on my slides, so apologies for that. But it means it means vaccinated with a primary series. There's another term called up to date that means vaccinated with a primary series and whatever booster doses were indicated for the particular individual according to minimal intervals specified in the guidance. That term is challenging to implement from a surveillance and monitoring perspective. So it's often not what's used in our measurements for surveillance data.
Thank you. And Dr. Nelson, final question in this series.
Thank you, Commander Scobie, for that great overview and a very informative presentation. One of our considerations for an EUA authorization is the availability of treatments for the disease in which we are trying to prevent with the vaccine. So in scouring the agenda today, I didn't really see the impact of available treatments for disease on the epidemiology of the disease itself. And I wonder if you'd care to comment at this time or save it for later discussion. Thank you.
Yeah, so unfortunately, I plan to, I have a variant expert also on the line, but I'm not sure that that person can be connected, Dr. Natalie Thornberg. So I didn't prepare for certain questions because I believe they would be able to be connected. But it's true what you're saying, that this is definitely a concern when we're talking about vaccination and these variants, whether there are other treatments that can be used when people become infected to protect them against serious illness. And it's definitely true that when Omicron became predominant, this was a major thing that the healthcare system had to deal with, because there was essentially only one monoclonal antibody that was effective against Omicron, and there wasn't enough of it, and it was a major problem. And Natalie is writing me now, but are you able to connect her?
Dr. Natalie speak?
Well, yes, if she's available.
Yes, she is available. Thank you.
And then we'll have to move ahead. And Dr. Nelson, this is an important point, which I think you may wish Dr. Mark's group to weigh in on, but later on this afternoon.
Understood. Thank you.
Is Natalie available?
Dr. Thornberg, are you there?
Yes, Mike, can you connect Natalie, please?
Yeah, this is like, let me suggest that
she's
we not
muted.
have too many link-ins. The technology is not all that able to handle this. So let's go ahead.
Natalie, hold on a second. I will unmute you right now. Hold on. I just had to know who it was. Thank you. Go ahead, Natalie. Take it away.
I believe you may have unmuted Dr. Linktellis. It's Natalie
No,
Thornberg.
I am
Oh, you're
unmuted
there?
now.
Okay,
Thank
great.
you. Yes, I'm here.
Great. Thank
Yeah. So I believe the question was use of therapeutics. How could you sort of summarize the question? It was use of therapeutics in how that has impacted the variant with variants circulating. Is that the correct question?
Concerns about Omicron, probably specifically, and the use of monoclonal antibodies and other treatments.
Yeah, the monoclonal antibodies, it has use of those treatments and those prophylaxis. It's definitely, Omicron has, because there's so many changes in the receptor binding domain of Omicron. Heather said there's 15 in the receptor binding domain. That's the part of the spike protein that binds to the cell, and therefore that is also the same region that neutralizing monoclonal antibodies bind. So Omicron has indeed lost activity, or the monoclonal antibody therapeutics. Several of them have lost potent activity against Omicron, and we don't have as many of those available. Those same problems won't exist for small molecule inhibitors, fortunately, and new monoclonals can be developed. But when a variant emerges like Omicron that have a lot of changes in the receptor binding domain, it does reduce the toolbox clinicians have to use when people get infected.
I guess for committee's consideration and one clarifying question, would it be fair to state that the availability of these various treatments have had little impact on the overall course of the epidemiology of the disease in the U.S. at this time?
Well, since we know that, I think that the transmission of the virus, most people are most transmissible in the day or two leading up to symptom onset and the few days after symptom onset. Often they can't get access to treatment until their transmissibility is already beginning to wane. And therefore, vaccines are, a really key tool in reducing transmission. But we have to use all of the tools in our toolbox.
Right. Thank you, Dr. Marks, for a final comment before we go on to the next presentation.
I think it's very
This is going to continue for the whole day,
step
Doug.
back here for a moment and just recognize that vaccines are a unique public health tool that is relatively inexpensive. The safety of vaccines in terms of the benefit risk is often much better than the safety of some of the therapeutics that might be used post facto after one is infected. And so one of the really important things here about vaccines is they have been wonderful public health interventions, and that's why we use them. They can give protection to many, many, many more people than we can come up with courses of oral therapies or intravenous therapies. And the cost of actually, and the complexity and the potential complications of delivering intravenous therapies or even some of the oral therapies are much greater than the simplicity of giving vaccines. Not that vaccines have zero risk associated with them. We'll hear about potential side effects of vaccines later today.
Love you too, Lisa.
That overall, the benefit risk is quite favorable as a public health intervention.
Thank you, Dr. Marks. And now we are moving ahead to the next discussion, which is about myocarditis. And we will hear Captain
So this is important.
Puro from CDC giving us this update. Thank you.
Hi, can you hear me okay?
Yep, you're fine.
All right, next slide, please. Or I'm sorry, I'll control. So today topics I'm gonna cover is a background on classic myocarditis and myocarditis associated with mRNA COVID-19 vaccination. And then I'll give an update on myocarditis following mRNA COVID-19 vaccination with a focus on people ages 18 and older. That will include data from the Vaccine Adverse Event Reporting System, or VAERS, and the Vaccine Safety Data Link, or VSD.
Who is speaking?
So, classic myocarditis usually has an infectious cause, typically viral or presumed to be viral, although infection with a pathogen is frequently not identified. It can be due to direct microbial infection of the myocardial cells. I'm having some technical difficulties with the slides. They keep on reversing order here. I don't know if that's on my end or your end.
We're not touching your slide, so go ahead.
No, we're okay.
Okay. It can also be toxin mediated or in a setting of systemic infection or infection of non-cardiac tissue. Rare causes include autoimmune hypersensitivity or giant cell myocarditis. Incidence is higher in males compared to females starting after age 5 years. And as I mentioned, it's common to not identify a pathogen or possible infectious etiology for myocarditis. In some studies, when they do testing in a minority of cases, do they find a possible infectious etiology. So these are graphs showing the epidemiology of myocarditis with children on the right-hand side and adults on the left-hand side. This is from the published literature. If you focus on the left-hand side, with the exception of very early in childhood when there may be factors like genetic factors in play, incidence is relatively low in early childhood and then begins to increase in adolescence. And if you move over to the right graph, you can see peaking in adolescence and then gradually decreasing incidence with age. And most of these cases are male, and by the time you hit middle age, the male to female predominance goes away. So this is a table showing the characteristics of myocarditis associated with mRNA COVID-19 vaccination in a comparison with viral myocarditis. So for vaccine-associated myocarditis, mRNA COVID-19 vaccination is the inciting exposure. And then for viral myocarditis, it's viral, although many of these cases can be asymptomatic. For vaccine-associated myocarditis, most cases have been in adolescents and young adults with more cases in males compared to females. Then for viral myocarditis, incidence in males greater than females, male incidence peaking in adolescence and then gradually declining. Onset for vaccine-associated myocarditis has typically been within a few days after vaccination, with most cases occurring within a week. And then for viral myocarditis, onset is typically 1 to 4 weeks after viral illness. The next set of characteristics get at clinical severity, but just in general, the vaccine-associated myocarditis following mRNA COVID-19 vaccination has been relatively mild when compared to viral myocarditis, which can frequently be severe. So now I'm going to move on to data from the Vaccine Adverse Event Reporting System, which is the national spontaneous reporting system that's co-managed by CDC and FDA. The key limitation, VAERS is a passive surveillance system. We generally cannot determine cause and effect from VAERS data alone. So this is a flow diagram showing U.S. reports to VAERS of myocarditis after mRNA COVID-19 vaccination among people 18 and older following primary series and first booster. We have observed 1,836 reports in this age group. 11 remain under review, 504 did not meet case definition, and that leaves us with 1,321 reports in this age group that met CDC case definition. To put that number in context, there's been an estimated 491.9 million primary series and first booster doses administered in this age group. This is a figure showing time to onset of these cases, and you'll notice that there appears to be clustering within a few days after vaccination. Many of these cases occurring in the 1 to 4 day period. When we get to the vaccine safety data link, I'll show you some additional data that also supports this clustering within a few days of onset within a few days of vaccination. So of these 1,321 reports, verified reports that met CDC case definition, the median age in this age group, 18 and older, was 28 years. Median time to onset of symptoms after vaccination is 3 days. Most of these occurred after dose 2, and most occurred in males. This is a table of VAERS reporting rates of myocarditis per million doses administered after mRNA COVID-19 vaccination in the 0 to 7 and 8 to 21 days post-vaccination. I know this is a busy slide, but there's a few key takeaway points from this. If you look, the peach-colored slides are where the observed reporting rates to VAERS exceed the expected background rates based on what's in the published literature. So you can use that as a proxy of risk. That's where the O to E ratio exceeds background. If you look in the 8 to 21 days, you'll see that there are no peach shaded cells, and that reinforces that the risk is concentrated primarily in the 0 to 7 days. If you look at males versus females, you see that reporting rates are generally higher in females, and reporting rates for both males and females are higher after dose 2 compared to dose 1. I have the children in there for reference, but if you start at the 18 to 24-year-old age group, you see that the reporting rates decrease with time, and at least for males, by the time you hit 50 years old, we do not see an increased risk. So of these 1,321 reports, where we had information on healthcare utilization, most were hospitalized. And most of these reports that were hospitalized had a known outcome at the time of the report, and 73% of these had recovered from symptoms at the time of the last follow-up, according to the VAERS report. There were 21 reports of death involving myocarditis when we evaluated the reports and accompanying records. In one report, myocarditis was attributed to causes other than vaccination. In 4, potential alternate etiologies were present. In 15, cause of death was not attributed to myocarditis. And then 1, adequate information was not available to fully evaluate the case. So I just want to spend a little bit of time talking about CDC's enhanced surveillance of myocarditis outcomes. And this is currently in an age group 12 to 29 years. The purpose was to assess functional status and clinical outcomes among individuals reported to have developed myocarditis after mRNA COVID-19 vaccination. And it's a 2-component survey conducted at least 90 days after the onset of symptoms. It included a patient survey and a healthcare provider survey. When the analytic period closed in November 2021, VAERS had received 852 reports in this age group that were at least 90 days, that met case definition, that were at least 90 days post-myocarditis diagnosis. We're able to complete 360 patient surveys and 398 cardiologist or other healthcare provider surveys that these patients were seeing in aftercare. The main finding from the cardiologist or healthcare provider survey was that based on the provider assessment, most patients appeared to have fully or probably fully recovered from their myocarditis. Roughly 82% of patients, according to the cardiologist, were classified as fully recovered or probably fully recovered, but pending more information. And the majority of the remainder had improved, but did not report being fully recovered. So some key findings from this enhanced surveillance activity. On patient follow-up with the patient surveys, at least 90 days after diagnosis, most patients who were reached reported no impact on their quality of life, and most did not report missing school or work. As I mentioned, 82% of healthcare providers indicated that the patient was fully recovered or probably fully recovered. Notably, there was substantial heterogeneity in the initial and follow-up treatment and testing of these patients, and there did not appear to be a single test that was indicative of recovery. Some additional next steps we're doing is we're going to follow up on patients who are not yet fully recovered at the time of the survey, and further assess the recovery status at least 12 months after myocarditis. And we're also following up on children and evaluating myocarditis cases in children ages 5 to 11 years. So now I'm going to move on to data from our vaccine safety data link system, which is our electronic health record-based system for surveillance and research. We conduct rapid cycle analysis in the vaccine safety data link. The aims are to monitor the safety of COVID-19 vaccines weekly using pre-specified outcomes and to describe the uptake of COVID-19 vaccines over time among eligible VSD members. Here's a table of the pre-specified outcomes that we are monitoring in VSD and the settings in which we are monitoring them. So I'm not going to go through this slide. This is methods. I'll just mention that the primary analytic method for VSD rapid cycle analysis is a vaccinated concurrent comparator analysis. It basically compares vaccinated individuals to other vaccinated individuals, looking at cases in a risk interval compared to cases in a comparison interval. For the outcome of myocarditis and pericarditis, all cases were chart confirmed and verified using the CDC case definition. So here's the mRNA COVID-19 vaccine doses administered in VSD in the age group 18 to 39 years old, which is the age group that I'll be presenting data for. There were about 950 patients who received a primary series, dose 1 and dose 2 for Moderna, and about 1.5 million who received a primary Pfizer series. There's about 574 million people who received a Moderna booster dose 1 and about 812,000 people who received a Pfizer booster dose 1. This is the figure showing the day of onset of verified myocarditis and pericarditis cases in the age group. And you can see, similar to what I showed in VAERS, these cases following vaccination tend to cluster shortly after vaccination. In this case, statistically significant clustering in the days 0 to 3 and 0 to 4, reinforcing the biological plausibility of this 0 to 7 day risk interval that we use for our main analyses for myocarditis after mRNA COVID-19 vaccination. So this is a table showing verified myocarditis and pericarditis cases in the 0 to 7 day risk interval compared to outcome events and vaccinated comparators and risk. This is basically looking at the risk in the risk interval the risk in the risk interval compared to the comparison interval. The statistic is the adjusted rate ratio, and this table is for males 18 to 39 years old. And you can see whether it's a combined analysis of both vaccines or looking at the Pfizer vaccine or looking at the Moderna vaccine, the adjusted rate ratios are all elevated, many of them statistically significantly elevated, with the dose 2 rate ratios tending to be the highest. And then you see on the far right-hand side there how that translates into the excess cases in the risk period per million doses, which, depending on the analysis, range from about 40 to 60 additional cases in the risk period per million doses administered. So this is the same table, but for females. And you can see that there are the case counts are substantially lower. The adjusted rate ratios tend to be elevated, some statistically significantly elevated, and some of these adjusted rate ratios are comparable to those observed for males. But I want to caution you, this is based on relatively small numbers, and so these can be impacted by those small number effects. And then you see the excess cases in the risk period on the right-hand side there. I want to go back to the previous slide. You'll see that the excess cases in the risk period there, like I said, in the highest risk strata, ranging from about 40 to 60, and you'll see they're substantially lower here for females. So even though some of the adjusted rate ratios may be elevated in females, because of the lower case counts, the excess risk tends to be quite lower in females compared to males. So this is a table showing the level of care and status of the cases in VSD, and these are the cases after the primary series dose of mRNA COVID-19 vaccine. Most of these cases are admitted to the hospital. A relatively small minority are treated in the emergency department. The length of stays tends to be short. The median length of stay is 1, and the overwhelming majority of these cases have stays of 3 days or less, and 100% of these case patients were discharged home. This is the same slide, but it's a similar table, but it's showing the cases following the first booster dose of an mRNA COVID-19 vaccine. And this, I think, just demonstrates that the level of care status is similar for the cases following the booster dose compared to the cases following the primary series. So just to sum up, the current evidence supports a causal association between mRNA COVID-19 vaccination and myocarditis and pericarditis. Cases following vaccination cluster within the first week of vaccination. The risk is greatest in adolescents and young adults, higher after dose 2 compared to dose 1 of the primary series, and higher in males compared to females. Some risk estimates for females in VSD are comparable to males, but case counts are small and excess risk in females is substantially lower than for males. The risk appears to decrease with age, and the male to female predominance of cases attenuates with age. Reporting rates in VAERS are highest following dose 2. Reporting rates following dose 1 and first booster dose tend to be lower. Incidence rates in VSD of verified myocarditis and pericarditis 0 to 7 days following vaccination are generally highest following dose 2. In a minority of age and sex strata, notably males aged 16 to 17 years, the incidence is highest following the booster dose. And based on our follow-up of VAERS case reports, available information suggests that most persons with myocarditis after mRNA COVID-19 vaccination recover from myocarditis by 3 to 8 months after diagnosis. I'd like to acknowledge the following groups for their contributions, and I'll be happy to answer questions.
So, just a quick one over here. Here is a Seattle.
I'm sure that the topic will come back. Remember,
This is the report from Seattle Children's Hospital, which showed that the children had cardiac imaging abnormalities even up to 8 months after the injury. So he, in my opinion, he misspoke.
I'm going to have a couple of questions, maybe 2 or 3, and then we're going to be going on to the sponsor presentation. So if you are disappointed, my apologies. Dr. Rubin?
Hi, sorry about that. Thanks, Dr. Shimabukuro. Is there, one of the hypotheses is that it is the antigen itself and cross-reactivity that's leading to myocarditis. Is there any evidence, and I realize a lot of it might be international, of an association between myocarditis and the viral vector vaccines?
Their audio is way off. And by the way, Novavax trading is halted today.
No, I couldn't.
Oh, Tom just disconnected his audio inadvertently, so I'm just going to reconnect Tom's audio here. Here he comes. There he comes.
Novavax
There you go, Tom.
trading
You
is
there?
halted.
Sorry, I lost audio there for a second and missed it.
That's okay. Go ahead, Eric. Can you repeat that quick?
The quick version. Is there evidence that this is the antigen rather than the method of delivery? In other words, do you see the same thing with viral vector vaccines?
So there have been case reports after the Janssen vaccine, but the data are pretty sparse. There hasn't been much vaccine administered, and there hasn't been much vaccine administered in these high risk groups, namely adolescents and young adults. So I don't think we have sufficient evidence to rule out or establish a risk. And I'm not aware of any surveillance or epi data from the AstraZeneca vaccine that would indicate a risk. But I think right now the data are really not sufficient for Janssen to draw hard conclusions on that.
Thank you. Dr. Galen.
As sure, the voting will be at the end of this day. The whole day long, they'll be doing these discussions.
You have your phone muted again, Dr. Galen.
So this is about natural history of myocarditis and recovery from it. You talked about recovery and the vaccine-associated cases. Does that mean in the natural history is fully recovered, meaning people don't have to worry about it ever again, or are there long-term consequences that might come up later? Over.
I'm probably not the best person to talk about the natural history of myocarditis. I think that there can be long-term effects, residual effects of myocarditis. There is not, as I said, there does not appear to be. There appears to be a lot of heterogeneity both in the treatment and in the follow-up care, and not really a standard to determine whether a patient has recovered. I'll say there's a bit of a lack of standardization. What I can say about the cases that we have followed up on is that overwhelmingly, either when you survey the patient or you survey the healthcare provider, they report generally having favorable outcomes. There's a small number which have recovered or which have improved but have not fully recovered, and that's why we're going to follow up
Once
on
again,
patients that
this imaging study shows every child had imaging abnormalities 3 to 8 months.
Thank you. Dr. Meisner.
Dr. Shimabukuro, thank you so much for your work in this area over the past year or two. It's been very helpful. The question I have for you first is a follow-up to Dr. Gallen's question, and that is gadolinium uptake of children or adolescents who have had myocarditis has been a helpful marker to address this question of longer-term inflammation in the heart muscle. And do you have any more data regarding, in the groups that you're following, regarding the results of studies to look at gadolinium uptake? If I may, a very quick question. The numbers from Israel regarding myocarditis following the mRNA vaccines is a little bit different than from theirs, probably reflecting the means by which the data is collected. Can you comment on that? Over.
I don't think I'm going to be able to comment on the comparison between the Israeli data and the U.S. data because I'm not, at least off the top of my head, not that as familiar with the Israeli data. To get to your question on the recovery status, for patients that we followed up on that did receive an MRI during outpatient follow-up, some of those patients did have this late gadolinium enhancement on cardiac MRI. And from speaking to our cardiologist consultants, the clinical significance of the late gadolinium enhancement, especially in patients who report, having recovered their cardiac function and are otherwise feeling well, is unclear. So I think there still needs to be some more work in that area about what is the significance of that finding in these patients, months after the initial diagnosis.
So this is the study.
Thank you.
Collect the figures.
We're going to have to move ahead, Cody. Dr. Bernstein, the final question.
Yeah, Tom, a very clear presentation as always, and I was interested to know whether the number of cases following booster doses appeared to be notably lower than after dose 2. Can you comment about the length of interval between doses as possibly further lowering the rate of myocarditis in patients?
In many cases, the rate of myocarditis after the booster dose is lower. After the booster dose is lower than after dose 2, although I would say not in all age and sex strata. I believe there is some evidence in other countries where they have had some different recommendations, or at least operationalized the vaccination program. So there were longer spacing between vaccines, maybe not necessarily the booster, but maybe the primary series, and some evidence that the spacing may mitigate the risk of myocarditis. In the United States, there tends to be a fairly close following of the immunization schedule. So we don't have that kind of data, but it is an interesting finding that when you have that longer interval between that booster dose, we tend to see lower reporting rates or lower risk. I would just caution that it's possible that there could be some self-selecting out of the population. For example, if a person got myocarditis after dose 2, they may not get a booster dose. So that may also be impacting the findings as well.
Yeah, I just mentioned it because of the 21 days between the dose of Novavax as well.
Right, thank you. We're going to have to move ahead. Dr. Shimabukuro, I'm sure we're going to be coming back to these issues and ascertainment and rates later on, so please stick with us to the afternoon. Now I'd like to turn the floor over to the sponsor, to Dr. Dubovsky, who is going to take the lead in the presentations on the emergency use authorization by Novavax for a vaccine to prevent COVID-19 in individuals 18 years of age and older. Over to you.
Thank you, and I'll just wait for the slides to load up.
So while he's waiting for the slide, this is the study. It says, in a cohort of adolescents with COVID-19 mRNA vaccine-related myocarditis, a large portion have persistent LGE abnormalities, raising concerns for potential long-term effects. Similarly here, notably in our cohort, although there was significant reduction in LGE at follow-up, abnormal strain persisted for the majority of patients at follow-up. This is 3 to 8 months.
It should be coming from...
Yep. We gave the share to Justin. There you go, sir.
So this is Novavax company representative.
They're up on our end, sir.
Excellent. Good morning. After that delay, my name is Philip Dubovsky, and I'm the Chief Medical Officer at Novavax. We're pleased to have this opportunity to present our data for NVX 2373. Our vaccine provides an important new approach in the fight against COVID-19. We believe its authorization will improve vaccine availability and accessibility with the ultimate goal of increasing vaccination rates in the US and throughout the world. As we will show you today, NVX 2373 leverages a well-defined platform offering a different vaccine option to fulfill an unmet need within the US and globally. Our vaccine is a recombinant protein subunit vaccine formulated with a natural novel adjuvant. It induces robust immune responses and provides high levels of protection against mild, moderate, and severe COVID-19. The vaccine is proven to be generally well tolerated and has a positive benefit-risk profile across a large and diverse patient population. Our COVID-19 vaccine is based on Novavax's platform technology, a recombinant protein antigen formulated as a particle, and our Matrix-M adjuvant, which is a Sapponin-based adjuvant. Our recombinant proteins represent a tested and well understood vaccine technology. Currently approved examples include influenza, Hep B, HPV, MenB, and shingles. There are also approved vaccines for malaria and shingles that include Sapponin-based adjuvants. Here's a brief overview of our vaccine. The NVX 2373 antigen is a recombinant SARS-CoV-2 spike protein. The spike protein is based on the full-length sequence from the original strain, including the transmembrane domain. We've engineered changes into the sequence to inactivate the pure nucleotide site and stabilize its structure. It's manufactured in the baculovirus SF9 insect cell expression system, a well-defined approach which has been used for decades. And then the full-length protein is expressed and self-assembles into a trimer, which is locked into its native conformation. These recombinant protein trimers are purified and further processed to form nanoparticles around a polysorbate core shown on the slide in blue. The adjuvant, Matrix-M, is purified from the silbark tree grown in South America. During processing and purification, the sapponin forms cage-like structures. The antigen and adjuvant are co-dispensed into a vial where they form a ready-to-use suspension. Now, let's talk a bit about the mechanism of action of the Matrix-M adjuvant. The mechanism of action of sapponin-based adjuvants has been evaluated in a number of animal models. Matrix-M exerts a transient effect at the injection site and a more sustained immunostimulatory effect in the draining lymph nodes. It does not contain alum, it does not form a depot at the injection site, nor does it engage the toll-like receptor pathway. Injection of Matrix-M increases the magnitude and breadth of the immune response. It induces a rapid transient activation of innate immune cells and increases cytokine and chemokine production at the injection site. This peaks at 5 to 48 hours after injection and rapidly drops by 72 hours. This results in a local influx of activated antigen-presenting cells to which the antigen is delivered and triggers an antigen-specific immune response. Subsequently, there's enhanced antigen presentation by MHC class I and MHC class II molecules in the draining lymph nodes. The result is the induction of high levels of neutralizing antibody, polyfunctional CD4 T cells, and a Th1 biased cell-mediated immune response. Now let's shift to the vaccine's presentation and profile. Our vaccine has key attributes that support the increased access and ease of use. It's dispensed in a 10-dose vial as a preservative-free, ready-to-use liquid suspension. It can be transported and stored at refrigerator temperatures, making it easy to ship, store, and administer. Each dose contains 5 micrograms of antigen and 15 micrograms of Matrix-M adjuvant. 2 doses are administered 21 days apart as a 0.5 ml intramuscular injection using standard needles and syringes. The proposed indication under discussion for today's meeting is for adults 18 years of age and older. Our clinical development program includes 4 studies. These studies constitute the body of data used for global regulatory approvals. The initial phase 1, phase 2 study established the 5 microgram dose level, confirmed the utility of the adjuvant, and a 2-dose schedule in both younger and older adults. The study also defined the immunologic phenotype and described the preliminary safety profile. Subsequently, our phase 2 study in South Africa that included a small subset of medically stable participants living with HIV evaluated preliminary efficacy and safety. Pivotal safety and efficacy was initially evaluated in a phase 3 study in the UK, followed by an even larger study in the US and Mexico to enable licensure within the US. As part of the US-Mexico phase 3 study, effectiveness and clinical efficacy was established in adolescents 12 to 17 years of age. Now, after discussion with the FDA, today's efficacy presentation will focus on our largest study, the U.S.-Mexico Phase 3 study, Study 301. Study 301 is most relevant for today's discussion because it was conducted in a diverse U.S. population, and the vaccine used in the study was manufactured at the commercial scale that is consistent with the vaccine we are distributing globally. Relevant safety data from all studies will be discussed. Okay, let's briefly review the 301 top-line results. Our U.S.-Mexico Phase 3 study met its pre-specified primary efficacy endpoint. Study 301 demonstrated an overall efficacy of 90.4% against protocol-defined symptomatic disease and achieved 92% efficacy in high-risk populations with medical comorbidities and 100% efficacy against moderate and severe disease. All events of moderate and severe disease and hospitalizations occurred in the placebo group. Additionally, matched strain efficacy was 97%. Matched strains are strains that are considered genetically similar to the original virus for which the vaccine is based. As far as variants go, our vaccine demonstrated high levels of protection against the alpha variant, as well as against all of the variants of interest and concern that circulated at that time. A number of variants circulated and caused disease during our study. In fact, the majority of cases were caused by variants of concern and variants of interest. The most commonly observed were alpha, epsilon, and iota. Displayed here are our global regulatory approvals. As of today, our vaccine has been granted authorization for use in over 40 countries for individuals 18 years of age and older, including those over 65. In December 2021, we received our first authorization in the European Union, granting access to all 27 countries. This was followed by the World Health Organization granting emergency use listing for global use. Subsequently, approval was achieved in the countries listed on the slide. I should note that the indication in India and Thailand includes adolescents and adults, and our vaccine has been approved as a booster dose in Japan. Our clinical development plans are ongoing. We will continue and will continue following EUA. We've submitted the results from the adolescent expansion of Study 301 and are completing safety follow-up in our Phase 2 and Phase 3 studies to support a full BLA. And we have additional ongoing studies in adolescents, and soon we will initiate a pediatric HD escalation study beginning in school-age children. We believe one approach to optimizing protective efficacy may include the use of vaccines engineered against emerging variants. We're collecting data on homologous and heterologous boosting to support continued vaccine use. And finally, we have post-authorization plans, which include additional studies for real-world effectiveness and safety monitoring. Here's the agenda for the remainder of our presentation. Dr. Rayburn Mallory will review the immunogenicity and efficacy data. Dr. Denny Kim will present the safety data, followed by Dr. Gregory Poland of the Mayo Clinic to provide his clinical perspective for NVX-2373. I will then return to conclude the presentation, and my colleagues and I will be available to answer questions from the committee. We also have additional experts with us today. All outside experts have been compensated for the time to prepare for today's meeting. Thank you. I'll turn the presentation over to Dr. Mallory to review the immunogenicity and efficacy data.
Thank you, Dr. Dubofsky. I'm the Senior Vice President and Head of Clinical Development at Novavax, and I'm pleased to review our efficacy data for the EUA application today. As I'll share with you, our data demonstrates that the vaccine induced high levels of neutralizing antibodies in both younger and older adults. The vaccine was highly efficacious in preventing COVID-19 illness and showed a high level of efficacy for the variants of concern and variants of interest that circulated during our Phase 3 study. The vaccine also completely prevented moderate and severe COVID-19 in the study. Before I describe our clinical data, I'd like to briefly summarize our non-clinical results. We conducted a number of non-clinical studies during the development of the vaccine. In immunogenicity studies, the vaccine induced high levels of functional antibodies and induced a strong Th1 biased cellular immune response. In animal challenge studies, the vaccine effectively suppressed viral replication in both the upper and lower airways. And this was an important milestone, as it suggested that the vaccine could be highly protective in humans. In addition, reassuringly, there was no evidence of enhanced disease in these studies. We performed a comprehensive toxicology program, and there were no adverse findings seen in these studies. There were also no adverse findings in our developmental and reproductive toxicity studies. So these results, combined with the absence of safety findings in all of the studies, supported moving the vaccine into clinical development. Turning next to the results from our US-Mexico 301 study, which is also called PREVENT-19. This was a randomized, observer-blinded, placebo-controlled study. We randomized participants in a 2-to-1 ratio to receive vaccine or placebo. And we did this so that we could gather additional safety data on the vaccine from a larger number of participants. The study initially enrolled adults 18 years of age and older, and we then amended it to also include adolescents 12 through 17 years of age. About 4 months after the initial vaccination period, participants remained blinded and were crossed over to the opposite treatment arm. And we did this so that all participants in the study could receive active vaccine. Participants are now being followed for up to 2 years following primary vaccination. The primary endpoint for the study was mild, moderate, or severe disease occurring 7 days or more after the second dose of vaccine. In terms of the definition of success, in order to meet the primary endpoint for vaccine efficacy, the lower limit of the 95% confidence interval needed to be greater than 30%. One of the main secondary endpoints we were interested in was how effective the vaccine would be in preventing the more concerning cases of moderate and severe COVID-19. As Dr. Fox mentioned, a large number of variants of interest and variants of concern circulated during the study. Sequence data were available for 75 of the endpoint cases, and 61 of these, or about 80%, were due to variants of concern or variants of interest, with the alpha variant being the most common one isolated. These strains are now classified as variants being monitored, as they aren't circulating to a large extent. However, efficacy for these variants remains relevant, as some of them contained escape mutations, like the L452R, seen in the Omicron subvariants, that could also be seen in future variants. Turning now to the results. Demographics and baseline characteristics were well balanced between the two arms as shown. 13% of participants were 65 or older. It's important to note that enrollment in older adults was somewhat limited. This is because COVID-19 vaccines became authorized and recommended for those over 65 while we were enrolling. Black or African American participants made up about 12% of the study population. 7% were American Indian, and 22% were Hispanic or Latino, representative of the overall U.S. population. In line with one of our study goals, about 95% of participants were considered at increased risk of COVID-19, either due to underlying medical conditions, their occupation, or living conditions. About 7% were seropositive at baseline, and we excluded these individuals from the immunogenicity and efficacy analyses. In terms of immunogenicity, in study 301, there was a robust neutralizing antibody response at day 35, 14 days after the second dose of vaccine. As you can see, this was true for both younger and older adults. In fact, there was an 123-fold increase for younger adults and an 87-fold increase in older adults from baseline. Brings the efficacy results.
The audio is gone. It's not.
Dr. Drbeen, we can't hear you.
So audio is gone for the time being?
I'll start this slide again. So looking at the Kaplan-Meier curve, we can see that cases began to diverge between the placebo and vaccine arms at around the time of the second dose, at day 21, and that there were very few cases in the vaccine arm through day 98. achieved our primary efficacy endpoint in the study with 90% protection from mild, moderate, and severe disease. In fact, there were only 17 cases in the 17,000 or so vaccine recipients, compared to 79 cases in the 8,000 or so placebo recipients. And as a reminder, we randomized participants 2-to-1 to receive vaccine. We also observed 100% protection against moderate or severe illness, a key secondary endpoint. As I showed previously, a number of different variants of interest and variants of concern were circulating during the study, and were responsible for about 80% of the cases where sequence data are available. Our vaccine showed 93% efficacy against these PCR-confirmed variants of interest and variants of concern, including the alpha variant. Some of the cases that did occur in the vaccine arm were mild in severity. Notably, efficacy was 97% for strains that would be considered more closely matched to the vaccine. When we looked at vaccine efficacy based on race, the vaccine provided a consistently high level of protection across all groups. The efficacy estimate for Hispanic or Latino participants was somewhat lower than that seen for the overall study, though with broad confidence intervals. In order to evaluate this in more detail, we looked to see if lower immune responses might have contributed to this finding. However, what we found was that IgG and neutralizing antibodies in Hispanic and Latino participants were actually slightly higher than those seen in non-Hispanic Latino participants. As a result, this efficacy estimate may reflect the chance finding. However, we will continue to gather additional information about the effectiveness of our vaccine in this subgroup in our planned U.S. post-marketing effectiveness study.
So guys, are you seeing that Hispanics have a lower
The vaccine
efficacy?
also provided a very high level of protection against severe disease, and for individuals who had baseline comorbidities that put them at increased risk from COVID-19. As you can see, the number of cases in older adults was limited. As you may recall, older adults made up 13% of the overall study enrollment, but they were clearly practicing social distancing measures during this time, as they only made up 6% of the cases in the study. The estimate for vaccine efficacy in older adults was 79%, so this was based on a total of 6 cases, and we believe this estimate is likely attributable to the very small number of cases that occurred. In the next slide, I'll provide some additional data on vaccine immunogenicity and efficacy by age that indicates that the vaccine continues to be immunogenic and efficacious in older adults. On this slide, I'm showing the relationship between geometric mean neutralizing antibody titers, efficacy, and age. When we look at adults 18 to 64 years of age, they had a neutralizing antibody titer of approximately 1,300, and this was associated with 91% efficacy. Within this group, we then evaluated whether vaccine efficacy might be decreasing with age. However, this did not appear to be the case, as vaccine efficacy remained high at 91% for the 50 to 64-year-olds, even though their antibody titer was somewhat lower at 979. We also saw an efficacy estimate of 89% for adults 50 or older, with a corresponding neutralizing antibody titer of 922. Finally, for those 65 or older, the efficacy estimate was 79%, so with a broad confidence interval that overlaps with the primary efficacy endpoint. When we looked at the neutralizing antibody titer for this group, which was 900, it is quite similar to, and in fact non-inferior to, the titers for the 50 to 64-year-olds and for those 50 or older that were associated with around 89 to 90% efficacy. These data, along with supportive efficacy data in older adults from a large phase 3 study conducted in the UK, not presented today, provide assurance that the vaccine is efficacious in older adults, supporting this indication in all countries in which the vaccine has been approved to date. So to conclude, our vaccine was highly efficacious in preventing COVID-19 in a large phase 3 study. This efficacy was demonstrated against all the variants that circulated during the study. The vaccine also provided complete protection from moderate or severe COVID-19 in our pivotal study. Our vaccine also demonstrated consistently high efficacy across subgroups, including race, gender, and in individuals with comorbidities. I'll now turn the presentation to my colleague, Dr. Kim, to present the safety results. Thank you.
Thank you, Dr. Mallory. I'm the Chief Safety Officer at Novavax, and it really is a pleasure for me to be able to present the safety results for our Novavax COVID vaccine. While our presentation will focus on the U.S.-Mexico Phase 3 Study 301, we will also take into consideration the other studies from our clinical development when there is safety data that adds additional insight. The total safety database includes nearly 50,000 people enrolled across 4 studies. More than 30,000 received our vaccine, and more than 19,000 of these individuals received the vaccine in our Phase 3 U.S. and Mexico Study 301 in the pre-crossover portion of the study. This is a large data set that can provide us confidence that we have a well-characterized safety profile that supports positive benefit risk and a favorable reactogenicity profile across the diverse populations that Dr. Mallory described. Now, taking into account follow-up time, during the placebo-controlled period of Study 301, we have more than 5,500 person-years of follow-up in the vaccine arm alone. The median follow-up was 89 to 92 days. And study compliance was high. Actually, more than 96% of study participants received 2 doses. I'd like to briefly review now how our safety follow-up was conducted. Beginning in the pre-crossover phase, participants received 2 doses 21 days apart at day 0 and day 21. On the day of each vaccination in the pre-crossover phase, participants recorded local and systemic reactions using an electronic diary for 7 days. Unsolicited adverse events were collected through day 49. Approximately 4 months after the initial vaccination, participants entered a blinded crossover and received 2 doses 21 days apart. Local and systemic solicited reactions were not recorded post-crossover. However, unsolicited adverse events were collected through day 49. Participants were then followed by visits occurring at 6-month intervals, in person or by phone, until the end of study. In addition to the nasal swab that was collected prior to each vaccination, continued monitoring for COVID-19 is ongoing with active and passive surveillance and prompt PCR testing when warranted. This study includes long-term follow-up with SAEs and AEs of special interest collected through 2 years following initial vaccination. Let me begin with a summary of our solicited adverse events collected through e-diary entries for 7 days following each vaccination. Shown here are the local reactogenicity events after the first dose. Participants 18 to 64 years of age are represented in the top panel, and those 65 and older in the bottom panel. Within each event column, on the left are vaccinated participants, and placebo participants are on the right. Pain and tenderness were the most commonly reported events, and those 65 years of age and older experienced fewer local events compared to those 18 to 64 years of age. While many participants did not report reactogenicity events, those who did mostly reported events that were grade 1 or 2, which is mild to moderate in severity, shown in blue. Grade 3 and higher, shown in yellow, occurred at low rates. Overall, these events resolved quickly with a median duration of 1 to 2 days. As expected, events occurred more frequently following the second dose. And again, as expected, more participants in the vaccine groups experienced these symptoms. And most events remained grade 1 or 2 in severity, with low numbers of grade 3 and higher events. Now, I'd like to turn to systemic reactogenicity. On this slide are systemic events after the first dose. Malaise, fatigue, muscle pain, and headache were the most commonly reported, And as well for systemic events, we also see lower frequency in those 65 years of age and older. Again, events were reported as grade 1 or 2 and resolved quickly with a median duration of 1 to 2 days. Notably, the rates of fever were quite low in less than 1% of participants. Following the second dose, while higher overall, most systemic events remained mild to moderate in severity as grade 1 or 2. Rates of grade 3 and higher events were low and occurred in relatively few participants. And even after the second dose, participants reporting fever continued to remain low. Moving to an overview of unsolicited adverse events. Overall, the frequency of unsolicited adverse events was comparable between vaccine and placebo groups, and severe adverse events were reported in few participants, both pre and post crossover. Medically attended adverse events, as well as potential immune-mediated conditions, were similar between groups. SAEs were also balanced across vaccine and placebo groups. And as you can see in the last row, deaths also occurred at similar rates between treatment arms. This figure shows pre-crossover unsolicited AEs by system organ class occurring at a frequency of at least 1% through day 49. This was our primary safety collection window through 4 weeks after receiving the second dose. Frequency between treatment groups was similar, and the overall percentage of participants reporting adverse events remained low. Here, we see more data on potential immune-mediated conditions. These all occurred with low frequency. Individual events occurred with less than 1% incidence and without any obvious patterns that would suggest associations with vaccination. There's a lot of data on this slide, so let me give you a moment to review the table.
So this is interesting that neuropathy, there were 3 cases out of 19,000.
Overall, events were balanced between both vaccine and placebo arms. Shown here are pre-crossover serious adverse events by system organ class with frequency of at least 0.1%. SAEs by system organ class were infrequent and comparable between vaccine and placebo groups, with the exception of the infection system organ class due to COVID-19 cases in the placebo arm. When we looked at individual preferred terms, there was a numerical imbalance driven by events reported as cholecystitis. And I'd like to provide you a little more of our analysis on this topic. Because we saw an imbalance in cholecystitis cases, we looked at the totality of the data, including a deep dive into individual cases. We do believe that the weight of evidence does not suggest a causal link. In study 301, the overall frequency of cholecystitis in the vaccine group is low, 0.05%, which is consistent with the expected background rate. In the UK study 302, there was one additional event in the vaccine arm, as well as another in the placebo arm. No events occurred in studies 501 and 101. All these events occurred in participants with known risk factors for cholecystitis. And all participants had gallstones at the time of event onset. A broader look at related terms with a standard MEDRA search did not reveal any additional findings. Importantly, there was no clustering or temporal relation to treatment. And we have not received any post-authorization reports with more than 740,000 doses administered. On this slide, we've plotted time to onset of cholecystitis following vaccination. The y-axis is the patient's age, and the x-axis represents the number of days from the first dose to when the event was reported. As you can see, we did not observe any temporal patterns and see a pretty random spread over a long period following vaccination. Because of the importance of myocarditis, pericarditis, we wanted to provide you a complete summary of our clinical data. For this analysis, we will be presenting from our entire pooled safety database. For a little context, and as Dr. Shimabukuro reviewed in detail, with the numerous investigations into the myocarditis findings of the past year with messenger RNA vaccines, I think we've learned that we can expect to see natural background events of myocarditis in any sufficiently large database. We've also learned that young males are at higher risk for both vaccine-induced myocarditis and other forms of myocarditis, most often caused by non-specific infections. COVID infections can also cause myocarditis. It's important to note that our studies were largely conducted during this time of heightened awareness for myocarditis. And so for our data, overall, in our placebo-controlled phase of our clinical development program, the rates of myocarditis were balanced between the vaccine and placebo arms at 0.007% for vaccine and 0.005% for placebo. No pericarditis was reported. In study 301, one case occurred in the active arm and one case in the placebo arm. As a reminder, there was a 2-to-1 randomization in study 301 in order to increase the sample size of vaccinees. And one case occurred in the active arm of study 302.
John.
Of the 2 cases in vaccine recipients, one 67-year-old male also had a concurrent severe COVID infection after dose 1. The other case from study 302 occurred in a 19-year-old male 3 days after the second dose of vaccine and was without a definitive alternative cause. In the post-crossover portion of studies 301 and 302, where all participants had been exposed to our vaccine, events of myocarditis or pericarditis occurred within the expected background rates as determined by the EMA access study. This study was specifically designed to determine background rates of interest for COVID vaccine. There were 3 reports of myocarditis or pericarditis, and all had plausible infectious alternative causes. One notable case occurred in a 16-year-old male 2 days after the second crossover dose of vaccine with a viral diagnosis that was diagnosed by a healthcare provider. One 20-year-old male had strep throat preceding the events of pericarditis, diagnosed by EKG findings and normal troponin levels. While the cases in the 2 teenage males, one during the placebo-controlled phase and one post-crossover, have characteristics of vaccine-induced myocarditis, we believe that the totality of the clinical evidence here is not enough to establish an overall causal relationship with the vaccine. I wanted to mention that we did not include here a case that the FDA has included in their briefing document of a 28-year-old male who had features of myocarditis, but was diagnosed by a cardiologist with non-ST elevation myocardial infarction. We also, a few days ago, received a follow-up report of a cardiac MRI that did not show evidence of a recent episode of myocarditis. Our latest monthly summary safety report with post-authorization data includes more than 740,000 doses administered and was submitted in May with a data cutoff of April 30th. We analyzed a cumulative 35 spontaneous reports of potential myocarditis or pericarditis received from passive surveillance systems. These reports often come with very limited information. Because of the general limitations of spontaneous reports, we carefully adjudicated these reports and applied the Brighton collaboration case definition. Out of the 35 potential reports, none met a definitive case definition. One report was a probable case of myocarditis in a 47-year-old male with an unknown time to onset. There were 10 reports of probable pericarditis. Of these, 7 were in males and 3 were females with a median age of 42 years. The time to onset was 2 to 14 days from vaccination. One of the 10 probable cases of pericarditis was in a participant with a history of messenger RNA vaccines and pericarditis. Illustrating the limitations of this type of spontaneously reported data, we just recently received confirmation by the Australian Health Authority that 2 pairs of the pericarditis cases are duplicate reports, bringing the 10 reports of probable pericarditis down to 8. It's worth noting that as of April 30th, all of the probable cases were reported from Australia. despite the fact that the doses administered in Australia account for only 17% of global administration of the 744,000 doses administered worldwide. No reports of probable cases had been received from other regions, including the EU and South Korea, which also have robust surveillance systems. And those regions also account for the majority of doses administered. We take all reports of adverse events seriously. As we examine the accumulating data and continue our collaborations and discussions with global regulators, we will get a better understanding of the nature of the cases and a more precise and stable estimate of the rates. We then expect to have more clarity on whether or not this important safety risk is related to the vaccine. We do consider myocarditis an important potential risk, and we are very carefully monitoring our post-authorization data. Additionally, we attempt to follow up each reported case with targeted questionnaires, and these data are being communicated in our analyses and our monthly summary safety reports. Our close monitoring will also include safety studies, which will cover large populations in administrative claims databases and electronic health records. For these other specific events of interest in our clinical development program, there were no reports of anaphylactic reactions or TTS in our integrated safety data. While our integrated safety data from our EUA submission did not have any cases of Guillain-Barré, a recent update to an SAE of neuropathy from study 302 has provided information that meets the Brighton collaboration case definition for Guillain-Barré syndrome. We will, of course, continue to carefully monitor for these events in our post-authorization surveillance activities. Because pregnant women were excluded from all our studies, there is limited information on pregnancy. For all women of childbearing potential, a negative urine pregnancy test was required at screening and prior to vaccination. But as it occurs for all large studies with long follow-up, we did have some reports of pregnancy. As of March 15th, 2022, a total of 147 pregnancies were reported across the entire clinical program. 56 of the pregnancies were still ongoing, and 41 resulted in live birth. 25 experienced miscarriages, 13 women elected to have voluntary termination, and 1 had an ectopic pregnancy. There were no fetal deaths or stillbirths recorded. Because pregnancy data was not systematically collected and there are inherent reporting and ascertainment biases, you can't make direct comparisons to background rates and draw definitive conclusions. But overall, this data does not indicate a potential risk for the mother or fetus, and there are no specific restrictions for pregnant women in our global label. In order to continue safety surveillance for very rare events that may not have been seen in clinical development, we also have plans and strategies in place to address potential safety concerns following emergency use authorization. Our post-authorization pharmacovigilance investigates potential risks such as vaccine-associated enhanced disease and myocarditis. Novavax supplements our routine monitoring with monthly summary safety reports and targeted follow-up questionnaires. Qualitative and quantitative reviews using multiple data sources for signal detection are conducted on a daily, weekly, and monthly basis. Additionally, we plan to conduct 5 post-authorization studies. They include 2 effectiveness studies and 2 safety studies using administrative claims and electronic health record databases to robustly characterize the safety profile in the post-marketing setting. And Study 405 is a global registry that will provide us with important data in pregnant women who receive our vaccine. So, in summary, the Novavax COVID vaccine safety data supports positive benefit risk and a favorable reactogenicity profile. Our vaccine is well characterized with exposure in more than 30,000 recipients across the entire clinical program in the pre-crossover placebo-controlled portion of the study. Local and systemic reactogenicity events were generally grade 1 to 2 in severity and resolved within 1 to 2 days. Grade 3 and higher events were infrequent. Importantly, we saw low rates of fever post-vaccination and most AEs were mild to moderate in severity. When we look at our long-term post-crossover follow-up, where more than 40,000 recipients received the vaccine, rates of SAEs were low and comparable to placebo. And for the important potential risks, we will continue to monitor for these events with our ongoing and future safety studies. Thank you. I'd like to invite now Dr. Greg Poland to share his clinical perspective on the Novavax COVID vaccine.
Thank you. Good morning, everybody. I'm very pleased to be here to provide my clinical perspective on the Novavax vaccine. I've been a practicing internist for 40 years and have served as a PI of over 40 vaccine clinical trials, and I'm the editor-in-chief of the journal Vaccine. I've spoken to this committee before about the need for COVID vaccines, and I'm here today to discuss why the Novavax vaccine is an important addition to what is already authorized. As we are all well aware, 2 years into this pandemic, the SARS-CoV-2 variants continue to challenge and re-challenge us. And a major reason for the continuing pandemic is that despite the availability of safe and effective COVID-19 vaccines and the constant efforts of our public health officials to increase vaccination rates, millions of Americans today are still unvaccinated, as we've heard. While I expected some of the challenges we're seeing today with this pandemic, I'm still surprised to see how this virus continues to unfold and what we're learning about the long-term and multidimensional impact the virus is having on individuals and the public health. There's no question that our ability to quickly develop vaccines has been impressive. However, the complexity and dynamic nature of this virus emphasizes the need to have multiple vaccine platforms to fight it. For those individuals who are not fully vaccinated and are waiting for another option, having a vaccine platform that multiple stakeholders, including regulators, physicians, and the public are familiar with, can help mitigate some of the challenges we're facing today. Indeed, a recent Ocugen Harris poll found that 73% of Americans would like additional COVID-19 vaccines developed from a more traditional method. Perhaps this is no surprise considering what we've witnessed during the pandemic when people become concerned about vaccine safety or tolerability. The latest CDC data reports that 89% of the U.S. population over the age of 18 have received one dose. And then we see the uptake of a second dose and booster shot fall precipitously. Only 77% have gotten a second dose and only 50% the first booster. There are many indications that decrease is linked in part to concerns people have about vaccine safety, reactogenicity, and efficacy. I certainly see that in my own practice. In the last year, I've received innumerable requests from physicians asking how to treat patients who've had a reaction to one of the currently available COVID-19 vaccines. Reactogenicity is a real problem and one that prevents a significant number of people from being fully vaccinated. So what is the benefit of the Novavax vaccine platform? The data show that combining the SARS-CoV-2 spike protein with an immune-enhancing adjuvant stimulates robust antigen-specific immune responses and provides high efficacy. Importantly, the vaccine is not highly reactogenic and compares favorably with other vaccines. We saw that borne out in the sponsor's clinical trials, where most events were mild to moderate and resolved in just a day or two. And as a reminder, the vaccine was able to deliver 90% efficacy with this favorable reactogenicity profile. This well-defined recombinant protein platform demonstrated safety and efficacy in 2 large phase 3 clinical trials against numerous variants, the combination of the immunogenicity data showing robust antibody responses across multiple variants with clinical efficacy data from the phase 3 trials signals broad cross-protection. This will be vital as we head into an era where we simply don't know what the next variant will be. Simply put, it's important to have choices in vaccine platforms in a pandemic that is constantly evolving. It's also important to make it as easy as possible to get vaccines to the people who need them. While many of us think of logistics in the cold chain and increased access as an issue in the developing world, there are also many healthcare providers in the United States who will find the ease of storage and administration of the Novavax vaccine to be a significant benefit over current vaccines. Every day, we're learning more about just how important it is to remain vigilant about trying to control this pandemic in the longer term for both the individual and the public health. Someone who is unvaccinated has a 4-fold greater chance of getting infected, is 23 times more likely to be hospitalized, and has a 20-fold higher chance of dying than a vaccinated person. The fact that we're still seeing more than 300 people die every day in America from COVID-19 is simply unfathomable to me. In fact, there are far more than the 100,000 new cases being reported each day. This is clearly an undercount due to the amount of home testing. These cases are resulting in almost 3,000 new hospitalizations per day. And this represents a significant opportunity to protect health with vaccines. And this is just the impact from the immediate acute infection. One aspect of this pandemic that is just starting to be understood is the long-term impact. And this includes both the individual and our healthcare system. A study published in Nature this February showed that 1 year after recovery, people with COVID, whether mild or not, had a substantially higher risk of 20 different cardiovascular conditions than those who did not have COVID. These conditions, like heart disease, vascular disease, and heart failure, are likely to negatively affect the health and life expectancy of people for years, if not decades to come. In addition to physical ailments, the new research is also documenting the mental toll COVID is having on people. A study also published this February, this one in the BMJ, reported that people who had COVID and were hospitalized were more likely to experience anxiety, depression, suicidal thoughts, and to experience opioid disorders. And these physical and mental health issues due to COVID are preventable if we get a handle on this pandemic and offer people choices that they may be more likely to use, thereby encouraging them to get vaccinated. In summary, as a clinician, I believe the Novavax vaccine offers benefits to multiple stakeholders. Patients and providers will find the Novavax vaccine an easy and logical option based on its efficacy, safety, and tolerability, especially the millions of Americans who say they are waiting for another option. Pharmacies and distributors will find this an easy and logical option for logistical reasons, because as we've heard, it's easier to ship, store, and administer. Employers will find this an easy and logical option to encourage people to get vaccinated. And finally, policymakers will find this an easy and logical option because it's a vaccine that's easy for people to access, easy to explain, and a choice that people want. One last note, while we're here today to discuss only the COVID vaccine, the clinician in me is also hopeful about the continued potential of this vaccine platform. By design, it's inherently amenable to combination vaccines, including influenza, RSV, and other respiratory illnesses. We've had remarkable success increasing vaccine compliance, utilizing combination vaccines in children, and ultimately, this can be true in adolescents and adults. As I conclude, I want you to understand that speaking to you today is both personal and professional for me. I've dedicated my career to researching and fighting infectious diseases. I've taken care of patients for over 40 years, and I've seen firsthand the miracle of what vaccines can do. We have an opportunity and a need to be proactive and continuously vigilant as the challenge and the fight against COVID is likely to continue for the foreseeable future. Authorizing an effective vaccine with a different mechanism of action is not only important for Americans, but will have an impact on global health. Our goal should be to have the right vaccine for the right person, for the right purpose, at the right time. And having more vaccine options with different platforms is a key component of achieving just that. Thank you for your attention. I'll now turn the presentation over to Dr. Dubovsky to conclude.
Did we need this sales pitch?
Thank you, Dr. Poland. The results from our clinical development program strongly support the emergency use authorization for people 18 years of age and older. Our vaccine is based on a differentiated platform that is well understood. The recombinant protein vaccines have been used globally for decades. Our adjuvant, Matrix M, is a natural sapponin product, and sapponin-based adjuvants are used globally. Importantly, our vaccine achieved 90% efficacy in our phase 3 study in the US and Mexico, despite the majority of cases being attributed to variants. Our vaccine offers a favorable reactogenicity profile, with most symptoms resolving after 1 or 2 days, and our safety data that was collected in a diverse American population supports a positive benefit-risk profile. In summary, NVX-2373 can be a useful tool in addressing the ongoing pandemic, providing a different option that may be helpful in increasing the incomplete vaccination rates in the US. Thank you, and I'll turn it over to Dr. Monto.
Thank you. We have just a few minutes for some specific questions. I want to remind the committee that we will have a much more less time constrained discussion after we hear the FDA presentation. And I want to remind you that yes, we do know there have been other variants. Yes, we know there are booster shots being given, and there are mix and match strategies, but that's not what we're going to be talking about in the next few minutes of questions. We're going to be talking about the presentation of the data on the clinical trial that is being considered for our evaluation right now. Dr. Levy.
Hello, thank you for that presentation, which was very interesting. I had 2 quick questions. One is regarding the apparent lower vaccine efficacy, or VE, in Hispanic or Latino individuals. The presentation pointed out that this was puzzling because the immunogenicity appeared to be similar to other groups, and that maybe this was a chance observation. Another interpretation may be that we don't understand the correlative protection well enough, and that what's being measured for the immune response doesn't capture what is protecting. So does Novavax have a comment on that? The other question regarding safety, are there any lessons to be learned from looking at safety data of other studies with other Sapponin-based adjuvants? Thank you.
Yeah, so as far as your first question goes, there's emerging data now from our phase 3 study, 301 study, that was supported by the US government. And there's a correlates of protection analysis that's emerging now. And the best correlate that was in fact identified appears to be linking it to neutralizing antibodies. Now, we looked at those patients, the Hispanic participants, very closely. We were interested to know if they were from the US or Mexico. And in fact, all the cases were in the US. And when we looked carefully at their other risk factors, we didn't identify anything specifically which seemed to have pointed toward increased risk. So right now, our best estimate is that it may be a chance finding alone. Now, as far as other sapponin-based vaccines, the largest database for our particular version of sapponin is clearly the studies we presented today. We have data from multiple antigens that we've tested previously in pre-licensure studies, and that includes influenza, which we took to a phase 3 study, and the reactogenicity profile looks comparable. And certainly we didn't see anything that looks like any of the events of concern that we talked about previously. The other Sapponin-based vaccines are largely, they're distributed both in the U.S. as well as globally. And I'm not sure that there's anything specific we can learn from them, because clearly they are given with different antigens. So that leads to a different biological profile.
Thank you. Dr. Pergin.
Okay. I think I was unmuting and somebody unmuted me. I apologize. So I had a question just about the incidence of COVID. It seemed as though the benefit was primarily after the second dose of the vaccine, which was around 3 weeks that second dose was given. Does the company have any data on antibody responses following the first dose? Knowing that the data that Dr. Poland presented, not everyone does get a second dose of vaccine. And is there evidence, or do they have additional data on those who only received one dose of vaccine and the antibody responses in those?
Yeah. So the Kaplan-Meier that Dr. Mallory showed the rates diverging at day 21, which was the day the second dose was administered. So obviously it takes time for that second dose immune response to mature. So we would posit that some of that benefit we're seeing is really from the first dose. We have looked at efficacy following the first dose, and I think I'll need to bring that data to you after the lunch break. I don't seem to have it readily available right now.
That's perfect. Thank you. Let's go on to Dr. Berger.
You don't have to wait till your camera pops up to speak. Go ahead, Dr. Berger.
We see you.
Dr. Berger, did you mute your phone? Yeah, did you mute your own phone? There you go, sir.
Okay, sorry. I just wanted to come back to the question around vaccine efficacy in Hispanic populations again, and just see if you've been able to conduct any subgroup analyses to look at the 18 to 64 range and the 65 plus to evaluate vaccine efficacy in each of those subpopulations by themselves.
Yeah, there were very few number of cases, if you remember. There were a total of 27 cases, and in the elderly there were really very few cases. As Dr. Mallory presented, there were only 6 cases total. So I'm not sure that would be an informative analysis to look at, but we can try to do a 2x2 table over the break.
Thank you, Dr. Meisner.
Thank you, Dr. Montel. I have one comment which will lead into my question. That is, there's an interesting editorial this week in the New England Journal about whether the world needs additional vaccines. And the article points out that there have been 11.5 billion doses of COVID vaccines that have been distributed, which sounds like a big number, until you remember there are almost 8 billion people on the planet that we need to vaccinate. And the distribution, obviously, of those vaccines have been unequal between high income, middle income, and low income countries. So clearly there's a need for additional vaccines. And furthermore, it may be that a protein platform vaccine, such as the one you're using, offers advantages over the messenger RNA vaccine, which is what leads to my question. And that is, Dr. Mallory mentioned that there was a reduction in upper airway viral numbers among people who had received the vaccine, suggesting that there may be some mucosal impact from this vaccine, and it might have perhaps a better effectiveness at reducing infection in addition to severe illness. Can you quantify that? Have you looked at mucosal IgA, or do you have a sense of how well this vaccine might protect against infection versus severe disease? Over.
Yeah, the data that Dr. Mallory referenced was primate work, and there what we demonstrated and is published now is that the vaccine is capable of generating sterile immunity in the upper and lower airway. We don't have IgA data from the studies. Those are difficult to collect, and during the pandemic, we weren't actually able to do that. What we do know is the vaccine is capable of preventing infection from our clinical studies. And we measured this by looking at seroconversion to N protein, as well as being PCR positive. And perhaps I'll be able to briefly share that data with you. But what we saw is that in the UK study, we were able to prevent infection in 82% of the people who were vaccinated. And obviously, if you don't get infected, you can't transmit, you can develop emergency variants, and you can't have long COVID because you're not infected.
These questions
Thank
seem
you.
like
Finally,
setup
Dr.
questions.
Marasco.
Yes, thank you, Dr. Mallory. So I have Dr. Mallory, I have a comment on Dr. Mallory's presentation and a question on study 301. So if I have the data right, you looked at neutralizing antibody responses 35 days after their second boost, and you followed the patients for roughly 50 days. Is there a time dependent? Is there a time dependence for breakthrough infection? Do you have enough data to know that? Because with your high neutralizing antibody titers that were measured at day 35, and then your follow-up period, is there a time-dependent risk in carbon infection?
Right. So it's an excellent question. As we described for all of our studies, we had to institute a crossover into the design, and that's because emergency use vaccines became available. And to maintain integrity of the study, we had to provide everyone a vaccine. What that did is it took away our ability to have placebo-controlled data beyond the crossover period. The issue with looking at the breakthrough cases for our specific vaccine in study 301 is there were very, very few cases. So, you could see from the Kaplan-Meier curve kind of where they fell out. And there wasn't a specific uptick as time went on. It was relatively flat, if you remember the data that Dr. Mallory showed.
So he's saying that the breakthrough cases are fewer.
We're going to be trying to catch up by taking a break now. Why don't we stick with the 15 minutes we had before, so we will reconvene at 11:30 Eastern Time. And then we're going to shorten our lunch to half an hour so that we can fully catch up and start the oral public hearings on schedule.
So they're going to take a 15-minute break. They'll come back in 15 minutes, then half an hour lunch after some more work, and then they'll go for the further. I'm going to mute my speakers a little. So here is my understanding of what we saw so far. Number one, it seems like the vaccine company, Novavax, is saying that FDA is wrong in pulling together. This is my interpretation. In pulling together all the myocarditis data, they do not even have some of that data. They did not even consider one case. There is another case that was followed up later. So to me, it seems like there is a hassle between the company and the FDA. FDA did something which I think is, compared to the other vaccines, and the other briefings, they did something underhanded, and that is they published this first, the briefing. I think they tried to sway or create a public opinion, and they provided their commentary, and they, for the first time in their document, they had added the cause and relationship to myocarditis, which they didn't do for the other vaccines. So that was interesting. So the company is coming back saying, no, you're taking the data, which is more than what we have in this trial. And secondly, that data does not seem to be because of the vaccine. So the problem is, whoever is right, there still is data of myocarditis, and this could be a risk for younger men. And so there is a problem there. So if the vaccine company is misinterpreting, then we have a problem. If FDA is misinterpreting, then we have a problem. So it's a weird situation. Somebody asked me yesterday, would you go get this vaccine? This is what I said yesterday. I am now confused about it from a mechanism point of view. It was an interesting vaccine. It is an interesting vaccine. The data manipulation and the fight that is happening, to me, it seems incorrect of FDA to do. Today's meeting is a fair game, but creating this opinion beforehand was strange. So that is one that so they're fighting over myocarditis. The second thing that is interesting is the upper respiratory and lower respiratory tract infection prevention. And so it is interesting that the company's representative or the chief medical officer said, well, we prevented 80% of the infection. Well, all vaccines did that. That is how they come up with the efficacy to say our vaccine prevented so many infections. But we also know now that the efficacy is really not long enough in so many cases. So that was a good question that, hey, what is a breakthrough infection rate and duration? And they said we had a crossover, so we don't really know, but we think it is very less. Then their answer that there was no IgE measured, which I understand. So if you give me a vaccine in a trial, now you're not going to take my saliva every day to say what is IgA level. And so this claim of preventing the infection seems to be an incorrect claim. I think we should say protection from the infection or reducing the severity may be a good answer. So I see both sides. And then this doctor for 40 years of experience, that was just an unnecessary sales pitch, which I thought that today should be more of a data day. Here is the data, and let's try to figure out an answer to vote yes or no, instead of vaccines are important, we should all have vaccines, and without vaccines we'll have this, and we have 8 billion people and we need to vaccinate them all. That just is a unnecessary sales pitch and wasting our time. This should be data. Here is the data, here are our questions, here are the answers, and then let's vote. Unnecessary wastage of time. So that is my takeaway. One, lots of, even the questions, those 3-4 questions, other than the breakthrough infection question, other questions just seem like setup. What do you think about the efficacy of the vaccine? Why do you think you are puzzled about the Hispanic cohort vaccine efficacy is less? Well, they said they're puzzled about it. So are they going to say now, in response to your question, that now we are not puzzled? They were puzzled. So it's weird. Overall, from 6 to now, 8:21, majority of the time was wasted. There were just a few interesting things. One thing that I really, Dr. Shimabukuro, and so I want to put this in front of all of us. I have done this discussion before. So let me share the screen for a second. Look, this is a study that I discussed. When did I discuss? Let me bring my discussion here. I discussed this study on March 28, 2022. This is a study by Seattle Children's Hospital. So it's not some fringe group as they like to um, label anyone who doesn't go with the mainstream message or their message. So this is a Journal of Pediatrics. This was from Seattle Children's Hospital. The only thing is the age is 12 to 17. This vaccine, Novavax, is 18 plus. The interesting thing in this one, they kind of brought it up a little bit about gadolinium in radiation images, gadolinium images. They brought it up. They knew this. Still, they were spinning it. And they were saying that everybody became okay. Yes, everybody clinically, from a symptoms point of view, became okay. But this is the study that 3 to 8 months later, children still had these signs of damage. Their hearts had not recovered. So that means it is possible somewhere in the future, when the heart is under more stress, there may be a less ability of the heart to work appropriately. So look, this is from their study. This is the section about discussion. In here they say, notably in our cohort, although there was significant reduction in LGE at follow-up, abnormal strain persisted for the majority of the patients at follow-up. In a cohort of adolescent with COVID-19 mRNA vaccine-related myopericarditis, a large portion have persistent LGE abnormalities, gadolinium, raising concerns for potential long-term effects. So it is not that these are not known. And it is actually interesting that FDA had put that message in the Novavax document, but not for other. Let me open up that document as well, which I thought was interesting, that all of a sudden FDA had found out that there is a, check this out. This is about FDA about Novavax. And if I search for that short term here, so I had shared this yesterday as well. This is the briefing document that FDA had made available beforehand. And see here, available data from short-term follow-up, they're talking about myocarditis, suggest that most individuals have had resolution of symptoms, although information is not yet available about potential long-term sequelae. So they're saying this about Novavax. Check this out. This is now the messenger RNA vaccine. In cohort of adolescent with COVID-19 mRNA vaccine-related myocarditis, a large portion have persistent LGE abnormalities, raising concerns for potential long-term effects. And did you hear that in today's discussion, they kind of were wishy-washy about that part of it? That is what I do not understand, that why do they do that? Really, I think the easiest is to put the data in front of us and let us figure out what data is saying. Then they do the voting in front of us based on that data as well. But this salesy, I didn't feel great about what I was hearing. So with this, give me a few minutes. I will take a break as well. I'll put this one on. So what did you think? And my apologies, this weird person with those porn site messages, I think they make many users with the same similar name, and you block one and the other one posts, and then you block that and the next one posts. And so it is YouTube that has to fix this. You cannot block keywords or words in a username. You can block them in the message, but you cannot block them in the username. So these guys have learned to create usernames with these weird site addresses. Carolyn, thank you. And I kept blocking them, but they just keep showing up. Guy Telfer says, if histones are released, does that mean DNA breakup? Yes. If histone is where the DNA, like if DNA is like a rope, histones are little, balls around which the rope is wound to kind of put a lot of rope in one small area without making it jumbled up. So if histones are found outside of the cells or even in the cytoplasm, then yes. During the cell replication, histones can be present in the cytoplasm as well because nuclear walls are torn down. But if they are found outside, then yes, nucleus has been breaking. Julia says, I'm confused about cell detection of infected area. The immune response to the vaccines was in the shoulder, not on where the COVID is. So original immune response is going to be where the vaccine is and in the local lymph nodes as well. But then when the infection occurs, so these immune responding cells, the memory cells, would circulate everywhere in the blood afterwards, and then they would go and live in various lymph nodes. They'll spread throughout the body. So then, as the infection appears from any route, they would respond. I hope I answered your question. I hope I got your question correctly. So give me 1 minute. I will take a quick break and we will continue. America Channel says, are you a doctor? Yes. Sorry, guys. 1 second.
Or offers the opportunity to cross over from the originally assigned study treatment to the other study treatment, the vaccine to placebo and placebo to vaccine. The primary efficacy endpoint was assessed until the participant received the first blinded crossover vaccination or until the data cutoff of September 27, 2021, whichever came first. And there was also a assessment of humoral antibody responses assessed in a subset of participants. The safety assessments included the following. The solicited, systemic, local and systemic adverse reactions during the 7 days after each vaccination, unsolicited adverse events and medically attended adverse events through 28 days following second vaccination in both the pre-crossover and the post-crossover period, and through the duration of the study, medically attended adverse events attributed to study vaccines, serious adverse events, and adverse events of clinical interest. Efficacy was assessed through daily surveillance of symptoms suggested of COVID-19 throughout the study follow-up. Symptoms of COVID-19 experienced by participants during the post-vaccination follow-up prompted an unscheduled illness visit in person. A nasopharyngeal swab was collected and sent to the central lab for processing. Additionally, participants were also given a kit to begin daily nasal swabbing within 3 days of symptom onset and collected for a total of 3 days. The swabs were also sent to the central lab for processing. Molecular confirmation of SARS-CoV-2 infection by the central laboratory was required to meet primary and secondary efficacy endpoint case definition. The primary efficacy objective was to prevent PCR-confirmed symptomatic COVID-19 illness diagnosed 7 or more days after completion of the primary series. Primary efficacy endpoint was the first episode of PCR-confirmed mild, moderate, or severe COVID-19 assessed up until the blinded crossover period. The primary objective would be met if the point estimate of the vaccine efficacy was 50% or more and the lower bound of the 95% confidence interval was greater than 30%. And below, some of the secondary and exploratory efficacy objectives are also shown at this slide. These are the case definitions for mild, moderate, and severe COVID-19. And the data sets to support the EUA. The FDA conducted independent analyses of data sets with different cutoffs and extraction dates. And these included data, efficacy, and safety data with the data cutoff of September 27, 2021. These were clean data sets. And then safety data was requested from FDA to review clinically important safety events. And this was an extraction date of February 17, 2022. And these were from data sets that were not post-clean. This slide presents disposition of all randomized participants as of the data cutoff of September 27, 2021. A total of 29,945 participants were initially randomized in a 2-to-1 ratio, with 19,963 vaccine participants and 9,882 placebo participants who received sampling. 96.8% of the vaccine group and 95.7% of the placebo group completed the 2-dose primary series. Then 77.7% of participants who were initially randomized to the vaccine group and 64.8% in the placebo group elected to participate in the crossover portion of the study. This slide presents the efficacy analysis population. Per protocol population for efficacy was comprised of participants who received 2 doses of the vaccine or placebo at the pre-specified time points and had no major protocol deviations prior to the first COVID-19 positive episode, no confirmed SARS-CoV-2 infection during the surveillance period or prior infection due to SARS-CoV-2 at baseline, and were not censored prior to the start of the observational period. 78% of vaccine participant recipients and 73.1% of placebo recipients completed at least 2 months of follow-up after dose 2. And then the second per protocol efficacy set included all participants regardless of baseline SARS-CoV-2 status. Here's the per protocol efficacy population. They were balanced in terms of percentage of male and female. The median age was 47 years, with 12% of participants 65 years and older. In terms of race and ethnicity, 11% of participants were African American, 6% were American Indian or Alaskan Native, 4% were Asian, and 22% were Hispanic. The main comorbidities were obesity and chronic lung disease. The primary efficacy endpoint was assessed until the participant received the first blinded crossover vaccination or until the data cutoff of September 26, 2021, whichever came first. In the per-protocol efficacy set during the pre-crossover period, 21.7% of participants who received the placebo were unblinded with the intention to receive a COVID-19 vaccine under EUA, compared to 13.2% of participants who received the vaccine. And these participants were censored for the primary efficacy analysis at the time of unblinding. So the results, the primary endpoint for 18 years and older as a whole was met. The vaccine efficacy for the first episode of PCR confirmed mild, moderate, or severe COVID-19 was 90.4%. And of the 17 cases of COVID-19 in the vaccine group, all were mild in severity. And there were, in the placebo group, there were 66 cases which were mild, 9 which were moderate, and 4 cases that were severe. There are no hospitalizations or deaths due to COVID-19 among the 96 primary endpoint cases. In an analysis of the primary efficacy endpoint provided for participants who were SARS-CoV-2 positive at baseline, among the 300 participants who were SARS-CoV-2 positive at baseline, there were no COVID-19 cases that occurred at least 7 days after the second dose. So the vaccine efficacy, regardless of baseline SARS-CoV-2 status, was 89.8%. And in the age group 65 years and older, the lower limit of the 95% confidence interval for the vaccine efficacy estimate was negative 16.6. There were 6 cases, 2 in the vaccine group and 4 in the placebo group. And the vaccine efficacy, or the 95% confidence interval for the estimate was wide. To provide supportive data for the effectiveness in adults 65 years and older, a post-hoc analysis of the vaccine efficacy among participants 50 to 64 years of age was conducted at FDA's request. And the neutralizing antibody titers in this age group was compared descriptively to those participants 65 years of age and older. And the table on the left summarizes the results of the immunogenicity comparison between the 2 age groups. The GMT for the participants 65 years of age and older was a little lower than the GMT for the age group 50 to 64 years of age. And but the GMT ratio was 0.91, with the lower bound of the 95% confidence interval that would have met FDA's usual success criterion for immunobridging. And on the right, the vaccine efficacy estimate for age groups 50 to 64 years of age was 90.7%, which was comparable to the overall vaccine efficacy for ages 18 years and older, which was 90.4%, and for the age group 18 to 64 years of age, which was 90.1%. These are the results of the secondary and exploratory efficacy analyses. First, the efficacy against COVID-19 among covariants of the 96% cases in the primary efficacy analysis. 75 cases had sequencing data available, which were mainly the alpha variant. And these were classified in the sponsor's presentation according to the CDC classification during May 2021. So currently, as of June 2022, none of the variants identified in the primary efficacy analyses were considered variants of concern or variants of interest. The second analysis was vaccine efficacy against moderate to severe COVID-19. Of the 13 cases, there was a total of 13 cases in the placebo arm and none in the vaccine arm, resulting in the vaccine efficacy estimate of 100%. Third, the vaccine efficacy by race was comparable to the overall study population, and as discussed previously, there was a lower vaccine efficacy estimate for participants of Hispanic ethnicity. For the participants in the safety analysis set, these participants were enrolled from a total of 119 sites in the US and Mexico. In the pre-crossover period, as of the cutoff date, September 27, 2021, the median duration of follow-up during the pre-crossover period was 2.5 months. And the safety analysis set included 19,111 participants in the vaccine group and 9,416 participants in the placebo group. And 77.8% in the vaccine group and 72.8% of placebo recipients completed at least 2 months of the safety follow-up after the second dose. In the post-crossover period, the median duration of follow-up after the fourth dose was 4.4 months, and 99% of participants in each study group were followed for at least 2 months after the second crossover dose. Third, the sponsor provided, at FDA's request, additional safety data through the extraction date of February 17, 2022 to assess clinically important adverse events. And at the time of the extraction date, the median duration of follow-up was 8.4 months after the completion of the crossover series. So the demographics of the safety analysis population in the vaccine group and the placebo group were similar. And the
conference call is now ending.
So just said conference call is now ending and she stopped. Oh man, her phone number is just... That is not nice. Her phone number is opened up to everyone. I cannot imagine that they're so less managed. This is like a... Have you seen relays that are so broken? Yep. So they're still working it out, the sound. And my apologies, my keyboard is here and the speakers are here. So when we put the mic near the keyboard and the speakers, then the sound.
Joining your meeting, you are muted by the host. 27 other participants.
Or go ahead with your microphone.
We did not detect any digits.
Yeah.
Can you still hear me now?
Yep, I can hear you now. Go ahead.
Okay. So the safety analysis population. Okay. So the baseline so the demographic and baseline characteristics. I think I'm hearing an echo.
Yeah, go ahead and turn off your speaker. Turn down your speaker. Again, if you want to just reconnect your audio, ma'am, right now you're on speaker, but on your microphone on your computer. If you want to dial back in, it's up to you. Otherwise, you can continue, but just turn your volume down. I'll help you.
Okay.
We were not able to automatically recognize you. Please enter your assigned code.
Yeah, let me, ma'am, I'm gonna dial you in a different way real quick. If you could look at the chat pod, just give us a quick momentary break, and I'm gonna help our speaker here.
So while they figure that out, I was apologizing for my keyboard and the speaker over here. And the other thing, I usually do not criticize folks who block those folks who criticize me for my opinions or my discussions, but I do block those who become insolent towards me, towards other fellows here, or start making up shit. This is what I do not like. So I just blocked someone who was just making up things about me or others. And I cannot stop them from making up stuff, but at least not sitting within my own home and then lying about me. So that is just my general approach, that I block such folks, because it just tells me they have no values. If they are going to lie, they know that they are lying, because I know they're making it up. So then I just block them. All right, just starting again.
So the demographic and baseline characteristics in the safety analysis population in the vaccine group and the placebo group were similar in the pre-crossover period, and also the pre-crossover period, the demographic and baseline characteristics were similar to the post-crossover period, and the safety analysis set was similar to the per protocol efficacy set. This slide shows the overall rates of reactogenicity.
It is still
You
not
still there, ma'am?
working.
Lucia, are you still there? Ma'am, did you mute your own phone? Oh, you dialed back in. Here we go.
Okay.
There you go. There you go, ma'am. Go ahead.
So the solicited adverse reactions were reported in higher proportion of the vaccine recipients than the placebo recipients, and more frequent after vaccine dose 2 than dose 1. So in the interest of time, I'm going to skip the details of this, but in general, the vaccine, the local and systemic adverse reactions were mild to moderate and lasted about 1 to 3 days. So this slide shows unsolicited adverse events and the frequency of non-serious unsolicited adverse events occurring through 28 days after dose 2 by time period are shown here. In the pre-crossover period, the percentages of participants reporting at least one non-serious unsolicited adverse event were comparable between the vaccine and the placebo group. And in the post-crossover period, the percentage of participants reporting at least one unsolicited adverse event was lower than the pre-crossover period, but slightly higher in the vaccine group than the placebo group. In terms of grade 3 reactions and grade 3 reactions considered by the investigator as related to the study product, all those percentages in both the vaccine group and the placebo group were low. The key findings in the pre-crossover period included that there were no adverse events by preferred term reported by more than 1% of participants in either study group. There were imbalances in the system organ class of general disorders and administrative site conditions and blood and lymphatic system disorders, which were largely due to reactogenicity and lymphadenopathy, respectively. Lymphadenopathy was reported by a higher proportion of participants in the vaccine arm for dose 1 and dose 2, which were 0.06% and 0.2% respectively, than in the placebo group. The most commonly reported severe unsolicited adverse event in the vaccine group was fatigue. The percentage of participants reporting serious adverse events was similar in the placebo group and the vaccine group in both the pre-crossover and the post-crossover period, and ranged from 1% to 1.4% across study groups. The percentage of participants reporting SAEs related to study vaccination was 0.1% in both the vaccine group and the placebo group in both time periods. The percentage of deaths reported in the vaccine and placebo groups was less than 0.1% in both time periods. This slide shows the number and percentage of deaths reported in the pre- and post-crossover periods by study group and the causes of death. For participants with fatal cardiac arrest, there were 5 in the vaccine group during the pre-crossover period compared to 3 in the placebo group and 1 in the post-crossover period in the vaccine group. For most of these participants, they had underlying factors, conditions which were risk factors for cardiac arrest. However, at this time, there was limited information available to assess the cause of death, as some of the autopsy data were not available. And participants in this study were randomized in a 2-to-1 ratio, which could account for more events in the vaccine group. Additional data presented through the February 17, 2022 extraction date. All of these deaths had a time to onset of 140 days or more following the dose 4 in the crossover period. None of these deaths were considered by the investigator or FDA as related to vaccination. In the pre-crossover period, the most common serious adverse events that occurred at higher rates in the vaccine group than the placebo group were cerebrovascular accident, acute cholecystitis, atrial fibrillation, pneumonia, aspiration pneumonia, and spontaneous abortion. In the post-crossover period, the most common SAEs occurring at higher rates in the vaccine group versus the placebo group were ischemic cardiac events, which included myocardial infarction, cholecystitis, both chronic and acute, and pneumonia. In terms of events of clinical interest, which included potential immune-mediated medical conditions, there were numerical imbalances observed in the following categories of cardiac, neurovascular, embolic and thrombotic, and biliary events. terms of cardiac events, there was an imbalance of events of cardiac failure and cardiomyopathy with 0.5% in the vaccine group and 0.02% in the placebo group. Almost all of these participants had underlying conditions that were risk factors, and the time to onset was comparable between the two groups. In the post-crossover period, there was an imbalance in events consistent with myocardial infarction, and the time to onset was comparable between the two groups. In terms of neurovascular events, in the pre-crossover period, there was an imbalance in events consistent with stroke, and 3 of the events occurred within 15 days of the most recent vaccine dose, and both events in the placebo group occurred within 15 days of the most recent placebo dose. Cumulatively, through February 17, 2022, there were a total of 19 neurovascular events consistent with stroke that were reported in the vaccine group in the pre and post crossover period. The time to onset from the last vaccine dose for 11 of the 19 cases, they occurred greater than 61 days after the last vaccination. In terms of thrombotic and embolic events, in the pre and post crossover period, the non-cardiac, non-neurovascular, thrombotic, and embolic events were balanced in the pre and post crossover periods. However, there were eight participants in the vaccine group that experienced events within 21 days of the most recent vaccine dose without plausible alternative etiology. And cumulatively, through February 17, 2022, there was an imbalance of pulmonary embolisms that occurred during the post-crossover period. However, most of the events in both study groups had an onset of greater than 90 days after the most recent dose, and the proportion of events with onset less than 2 weeks were comparable. In terms of biliary events, in the pre and post crossover period, there was an imbalance in cholecystitis, and the 18 events in the vaccine recipients in both time periods, 6 of those had an onset within 30 days of the vaccine dose. In terms of other events of clinical interest, those included Bell's palsy, and these were all in the pre-crossover period. There was one case of Bell's palsy within 30 days of vaccination in each of the placebo and vaccine groups. In terms of uveitis, there were 3 participants in the vaccine group with new onset uveitis within 3 weeks of vaccination, one of which recurred with rechallenge. And in the placebo group, there were 2 events of uveitis, one of which had onset within 1 week of placebo, and a participant that had a history of uveitis. And lastly, there was one event of angioedema and urticaria that occurred 2 days after dose 2 in the vaccine group, which was potentially related, but the participant also started an antibiotic concomitantly. And review of additional safety data from studies 101, 301, and 501, which were conducted in Australia and the US. 302 was in the United Kingdom, and 501 was in South Africa. The FDA reviewed serious adverse events and adverse events of clinical interest. These studies were conducted with the vaccine produced by an earlier manufacturing process than the vaccine evaluated in 301. So out of the 3 studies, there was one event, Guillain-Barré syndrome, that was reported by a 65-year-old female in the vaccine group who experienced progressive neuropathy starting at 9 days after dose 1. And other than this event, there were no new serious adverse events of special interest, or potentially immune-mediated conditions in these studies that were considered at least possibly related by FDA that were not already previously identified in Study 301.
I think so too.
So in a total clinical safety database of about 40,000 vaccine participants to date, 6 vaccine recipients reported myocarditis and or pericarditis, including 5 events that occurred within 20 days after the Novavax vaccine. So these are the cases that a little bit more detail. And these cases were concerning for the following reasons. The temporal relationship for 5 out of the cases occurred within 20 days after vaccination, and the event was, and only one of the events among the vaccine group had a clearly identified alternative etiology associated with myocarditis. And the other events had only circumstantial evidence of potentially plausible alternative etiologies. 4 of the events occurred in young men, which is a subject population known to be at high risk for mRNA COVID vaccine-associated myocarditis. Now, continuing to the sponsor submitted post-marketing safety data. And as of April 30th, 2022, there were about 700,500 doses administered in Australia, Canada, the European Union, New Zealand, and South Korea. The sponsor reported a potential safety signal for myocarditis and pericarditis listed here. And the observed to expected rate ratio for all doses was 4.95. So in summary, the known and potential benefits include that the vaccine was efficacious with an estimate of 90.4%, and the efficacy estimates from Study 301 were generally consistent among subgroups stratified by demographic variables and for the risk of severe COVID-19. The uncertainty in the benefits include vaccine effectiveness against currently circulating SARS-CoV-2 variants, long-term effects of COVID-19 disease, effectiveness in certain populations at higher risk of severe COVID-19, and the duration of protection. The known and potential risks associated with vaccination include local and systemic reactogenicity, myocarditis and pericarditis, and Guillain-Barré syndrome. And there are uncertainties in the risk in certain populations and for adverse reactions that are uncommon and that require longer follow-up, which include biliary events, neurovascular events, cardiac events, and uveitis. Sponsors submitted a pharmacovigilance plan to monitor safety concerns that could be associated with the Novavax COVID-19 vaccine. The FDA recommended adding myocarditis and pericarditis as an important identified risk, and the sponsor considered as important potential risks vaccine-associated enhanced respiratory disease, myocarditis and pericarditis, and anaphylaxis. The sponsor will conduct several post-marketing activities, which include active and passive surveillance activities, periodic aggregate safety review of safety data, and 5 planned surveillance studies. So the pharmacovigilance activities include adverse event reporting, which come from vaccine recipients, vaccine providers, or the sponsor themselves. First, the vaccine participants will be notified that adverse events can be reported to VAERS through the fact sheets for recipients and healthcare providers or from the VSafe program. Adverse event and these events, this reporting is voluntary. For the vaccine provider and the sponsor, these adverse event reporting is mandatory. For both the vaccine providers and the sponsor, They report to payers the following information: serious adverse events, irrespective of attribution to vaccination, cases of multi-system inflammatory syndrome in adults, and cases of COVID-19 that result in hospitalization or death. In addition, the sponsor will also conduct periodic aggregate safety review of safety data and report newly identified safety concerns. Both the FDA and CDC take a collaborative and complementary approach to reviewing adverse events. In the initial stage of post-authorization surveillance, FDA will individually review all serious adverse events on a daily basis. FDA will also examine other sources of AE, such as in the literature, and perform data mining to determine if the adverse events are disproportionately reported for the candidate vaccine compared to other vaccines and VAERS. And other potential safety signals will also be investigated. In addition to passive surveillance, FDA will also perform active surveillance studies for safety outcomes. These studies will be conducted using the Biologics Effectiveness and Safety System, which obtains safety outcomes from various healthcare settings. And active surveillance will also be performed using data from the Centers for Medicare and Medicaid Services. The sponsor also proposed 5 post-authorization surveillance studies. The first is a pregnancy exposure registry, and the second and third are 2 active follow-up safety studies, one in the US and one in the UK. And the last 2 are real-world effectiveness studies, one in the US and one in Europe. Lastly, the sponsor was requested to include certain safety outcomes in active safety surveillance studies, which includes the assessment of cardiac, neurovascular, embolic and thrombotic, and biliary events. The sponsor will also perform enhanced pharmacovigilance activities for safety outcomes of GBS and uveitis. And this concludes my presentation.
Thank you, Dr. Lee, and apologies for the interruption. You did very well, considering. We do have a few minutes for questions now before the lunch break. And I don't see any hands raised. Is that the system's fault, or?
No, there's no hands raised at the moment. I was looking to.
No hand raised. I can't believe it.
No. Oh, wait, here we go. We got our first one.
There we go. Okay, Dr. Gallen.
I also wanted to mention that Dr. Brandon Day will answer questions pertaining to the pharmacovigilance plan.
Thank you. Dr. Gallen.
Okay, thanks. Maybe we're going to get into it later, but thank you for that great presentation. There's a lot to detail, and you had to deal with the system, so none of that is easy. So thanks for that. Are we going to get into sort of the real world part of this? We heard the study data is through the end of September. A lot has happened since then. I'm guessing study participants had real lives and done other things, like gotten other vaccines from whomever, maybe had other experiences. Is that going to come into play at some point?
Dr. Day, do you want to take that question?
Who did you want to take that question again?
Brandon Day.
Brandon, go ahead and unmute yourself.
Or anybody else who wants to answer that question.
I don't see Brandon in here right now. I'll call Brandon back in just to be safe. So go ahead. Let's go to the next question real quickly.
I don't see another question. Can't believe this.
If you give us a moment, we'll bring Mr. Day back in here. So,
or anybody else who can answer that question. Might be Dr.
So while they are trying to figure that out, Texas Meg and DDS, there are some folks who want to be moderator. They would join Discord. Can you please talk with them, see if they are good for this work, and then we can go from there.
Can you restate the question?
Hi, we hear you. Go ahead.
Want me to go again?
Bruce, why don't you go again, Dr.
Yeah,
Dr.
thanks.
Fink's, Dr. Fink's
all right,
here
thank
as
you.
well, so,
All right. Can you hear me? Yes.
I also wanted to.
Yes, we hear you.
We did review data through the safety data through February 2022.
So that's the good news, is that somebody's continue to follow these patients, as you said, for safety past the end of the study. The question is, what else are we going to learn about the vaccine recipient, about the trial participants since the study? Durability, other vaccines that they may have received, did they get boosters of Novavax as well? Because this is entering into the real world of other vaccines. And while it's not our purview to figure out how they're going to be used if and when they're available, that's going to be an important consideration. And so any data about that, and including any data about Omicron, which is missing from this discussion because it wasn't present in September, but it's quite present now, and hopefully people are still being followed in an Omicron environment. Thank you.
So the study was conducted quite a while ago, and so there were the cases that accrued were not during the time that Omicron was circulated. And in terms of, we tried to focus mainly on the primary series, which is the topic of this VRPAC. So we were not prepared to further discuss the topics of participants who got boosters and those related topics.
Dr. Fink, are you in the position to straighten things out?
I will try, although I have to admit I'm having a lot of problems on my end with the system figuring out on or not. Can you hear me?
Yes, we hear you.
Yes. Okay, great. So I think that the main points, as Dr. Lee summarized, are that we do have rather long-term safety follow-up that we were able to review in detail for all of these study participants. There has been, some use of the vaccine worldwide in post-authorization settings, although we really don't have much data to report on that beyond what has been summarized in the slide. If this vaccine is authorized for use in the U.S., clearly we will need to have the same intense level of post-authorization safety surveillance as we have had for the other COVID-19 vaccines that have been authorized, some of which have gone on to be approved and fully licensed. And again, I just have to reiterate, I know that there is intense interest in the potential for using this vaccine as a booster dose in individuals who might have received a primary vaccination with some other COVID-19 vaccine. We don't have the capacity to discuss that potential use today or data related to that use. But if this vaccine were to be authorized for use as a primary series, we could take an approach similar to what we have for the other authorized COVID vaccines for considering use as a booster dose, and we would evaluate study data to inform the safety and effectiveness of a booster dose as it comes to us. So,
thank you.
And we have Brendan Day also on as well. Brendan, do you have anything to add?
No, I think they covered it. Thanks.
Okay, this will be a persistent item of discussion as we go through the rest of the day, is my humble prediction. Dr. Lee.
Yeah, thank you for that presentation. I think one of the questions that I have, and I don't know if you're exactly the person to ask, this actually was designed as a crossover so that individuals were randomized to either receive the vaccine first, followed by placebo, and then placebo followed by vaccine. So far, obviously, the primary endpoint was based on the original randomization to vaccine versus placebo before the crossover, although we have seen some safety data after the crossover period. Is there any plan to analyze? I mean, typically the crossover, you have a washout period and then you do the second comparison. And the reason I ask that is it would seem as though those that had started with a vaccine and then went to placebo, that there might actually be a carryover effect that might actually give us some indication as to waning immunity or not. And I didn't know if there were any plans for that analysis to be done to separate the pre and post crossover in terms of efficacy, not just safety.
I think the sponsor can add, but the study, since the study is still ongoing, there are provisions to collect samples to look at the duration of protection.
Okay.
Hey,
All right,
why don't we park that question, Dr. Lee, and this is something we can come back to when we have a broader discussion with the sponsor online as well after lunch.
Okay, thanks.
Dr. Meisner.
Dr. Lee, thank you for the presentation. I would like to ask you about the FDA's experience with baculovirus insect cell protein expression systems. I don't think it's been used very often. Do you have any other specific issues regarding safety with that eukaryotic protein expression system that the FDA worries about?
I'd have to defer this question to Dr. Fink or others from the FDA.
Yeah, hi. So we do have an example of a recombinant protein-based seasonal influenza vaccine, Flublok, that has been approved and is manufactured using a similar expression system. We don't have any safety concerns attached to that vaccine, specific to that manufacturing platform.
Thank you.
Not seeing any further hands raised to my amazement. We will be able to start lunch a few minutes early. Thank you, Dr. Lee, for your careful presentation and handling the technical issues in the middle. So back for the open public hearing at 1 o'clock.
Thank you.
All right, so everyone, just give us a moment as we pull up the lunch and nobody log off and take break yet. So let me.
Okay, so while they are on lunch, I wanted to discuss something that I saw here in the comments. And then I responded to that as well. So once again, I'm going to present some of the answers to the initial accusations. So there was somebody who said that somehow I'm not truthful about the data. And they also said that somehow Paul Bork is my data analyst and he is associated with some industry and we are not telling that. So one, he's not my data analyst. Paul Bork is a cool bean who prepares data because he's a Six Sigma, about COVID, and I had actually requested him to see if he can present that data. So he prepares it, he presents it. There is no data analysis other than what he presents. So that is one. So I actually reached out to Paul Bork and I said, are you associated with this company called Dow? So here is his response. He said, nope, I don't work for anyone. I'm retired. My last employer was the local school district, the job so I did work for Dow for a long time before I retired, before COVID started. That's where I became a Six Sigma black belt. I think we talked about this when I was first introduced. Perhaps I just said I worked in the industry. I don't really remember. I saw the comment before it was deleted. I'm also not your data analyst, just a repeated Cool Bean guest. The comment had so many things wrong. A Google search will find some of the things I did for Dow, perhaps on EPA servers. Nothing ever put on the internet may actually disappear, or very little does. So thanks for the question. The fact that you asked shows your concern for truth and accuracy. I like that. So then he said that, so this is from Paul. That is one. Second, yes, I am actually a software engineer and a medical doctor. And yes, I have always kept it open that I do not work in the US. And it is not that I was licensed in the US and somehow then I was not licensed or I got license canceled. I actually never sought my license in the US. So I don't practice here. And I never have tried to ask anyone in the US. Even when somebody reaches out and says, can you please guide me for this? The first statement I say is, I don't practice here. I don't advise here. Majority of my lectures here, medical lectures, contain this, that this is not a medical advice. So I have a very, very open process. Now, here, let me very quickly. This is actually my medical degree. So if you can probably read Bachelor of Medicine and Bachelor of Surgery, and somewhere over here, you would also see my name. Anyway, so this is my medical degree. And sorry, just to read a little more, I need to now open this and readjust it. But it says here, Syed Muhammad Mubin, King Edward Medical College, Lahore, Bachelor of Medicine and Bachelor of Surgery in this university at the examination held in March 1994, and that she is authorized to practice medicine, obstetrics and surgery, and then they have given my marks. So that is my my degree, medical degree. And then there are other degrees as well. If you see, there are two more. One is my bachelor's in sciences, BS degree, and one is my this one software engineering degree as well. So I am a software engineer too. And yes, I used to actually work at PayPal. I don't work there anymore. 2015 is when I quit PayPal. After that, medical teaching is what I do. And I had been doing medical teaching since soon after my college. I was actually teaching medicine even during the college because that is how I used to pay my fees. We were from a broken family, not very rich. So one way that I used to pay for our fees and our livelihood was by teaching tuitions in medicine. Then after I finished the college and residency, then I kept teaching. And so teaching is going on since 92, 93. And so why do I block folks, for example, shrimpy-like folks, who the only thing is when somebody makes up shit right in front of me about me and it is incorrect, then that is not good. And the reason is this: I know the truth. Here is my degree. Majority of the cool beans know the truth that I don't practice here. Now, if somebody is new and they don't know it, I do not know how every 5 minutes I can repeat that, but I try to repeat it as much as possible. So when somebody makes up things about me, and I know that I'm sitting right here and they are making it up for me, that means they don't have the values. Values are very important. Truthfulness. They themselves are not truthful to themselves. A person who is making up lies about another person is not truthful for their own self, because what they do to others is what they do to themselves. They would even lie to themselves as well. To have such person around in my home is not a good thing. Even if they were my physical friends, I would part way with them. Not because they said something about me and I was hurt or not, but because they don't have values, they don't have morals. So when somebody doesn't have morals, then they don't have a place with me. They can go stand out on the road, make up whatever they want to. I cannot stop that. That is their freedom of expression. They can make up shit, they can talk whatever they want. I have no control over that. But not when they're sitting in my home. And that is exactly what I also do for you. When you are here, so let's say there are 426 people right now over here. You are communicating, chatting. You are my guests. So I respect you for being my guest. So I don't need any fake respect. I do not need any fake credentials or approvals. But I also don't want someone to make up lies and then to say, and I also understand that this will happen. The more popular somebody gets, and by the way, that was never my intent to become popular. I just started to teach medicine to general public and learn to draw to teach medicine so I could teach more effectively. That was what it was. So I understand when somebody is more exposed, then there is going to be more attacks as well. However, do you think you would like to have somebody who is attacking you or abusing you? You would have them in your own home? So that is my point of view. So once again, I am a medical doctor. I don't practice in the US. I teach medicine. I'm a software engineer as well. I'm very proud of it. Now I have almost become an artist too. So not the best. If Meeple Art would be here, he can probably judge me. But I love art. And then if I have Paul or others, neither do I have any commercial interest with them, nor do I have any information interest with them to say provide some information in a specific direction or leaning. Little Ink says, I'm an artist, I approve. Thank you very much. Christine says, jealous because you're a hottie. Thank you. Lisa says that Dr. Peter McCullough says Novavax no longer works. So that is an interesting question. The question is, and you saw that question being asked a few minutes ago, Novavax trials were done on the original previous variants. Is it still efficacious for Omicron or not? That is a question. And there is no answer yet. This is a very good question. So Standing Mannequin says, does practice make perfect? So it makes perfect whatever you're practicing. If you're practicing incorrectly, then the incorrect things will become perfect. So for example, doing art, my line work or the line weights or the flow or the gesture was not the best, but I just kept doing it and I became more perfect in non beautiful art, and then I had to unlearn and relearn. Christine says, love you, dog, been such a beauty. Thank you very much. Craig says, you've tuned up my cell biology understanding. My original professor was brilliant but didn't teach well. We all had, that is true. Tony, the reason I explain these things are not for shrimpy like folks or whoever was here before, because what I have learned is somebody who's not going to believe or trust or like you cannot make them believe you or trust you or like you. They are in a group that is not going to do it. And they're going to keep searching for reasons to strengthen their position, even if these are false reasons. Similarly, somebody who likes you, trusts you, is with you. They're going to be with you regardless of how you are. Of course, not criminal, but meaning they're fine with you, with your goods and bads, with your faults. So when I explain this, it is really not for to go out and say, Shrimpy, please be convinced. So that's not the point. Or whoever else was there before. The point is that the audience, the cool beans that are here, if they have any doubt, they should at least be able to see where I come from. Jody says, I'm an artist. Anyone can learn to draw, but it's like basketball. You can learn, but you need talent to be NBA. Correct. So KB Usenets, no, they're not done. So here is where they are. So if you see, they are on a half-hour lunch meeting. Actually, I should use this time as well to quickly go have my breakfast. But I wanted to use this time to share a bit about myself. Craig says cats are good judges of character. So cats, Luffy loves to be with us. He would not be anywhere than to be on my table. I was just, his hair are all over me. And so he is just with me, my wife, my son. Kyrie would not even come near us. So within one family, one cat behaves differently than the other one. Yes. And I have actually never tried to use my credentials to, for example, in the beginning when the channel started taking off, there were many people who knew me who said, start wearing a white coat, start putting your stethoscope somewhere over there, start putting your stethoscope with you and have a little mannequin of a doctor on the side. And there were so many such things, but that all, or wear a shirt and a tie, and that all, whoever does it, it's great. But it just seemed so not me that I didn't even want to have any props to add further to who I am. This is who I am. Good, bad, ugly, better, whatever it is. But if you look at the purpose that is coming from this shell that you see, the purpose is, can we educate ourselves together? Can we learn more together? And this is why you know that there are people who get upset that I talk about vaccines and their benefits, and then I talk about, others become upset when I talk about vaccines injuries and protest about those as well. So the point is, how can we study together? KB Usenet says, I prefer your style. It is easier to learn with someone talking with me than to me. Correct. Correct. We explore together. Thank you. And then one more clarity, now that we have this time we're discussing. Sometimes people think that I don't respond to them, and that is a disrespect. There are times when there is a question which is, somebody is asking about themselves, saying, hey, I have this situation, what should I do? And it's not something I can answer. And even if somebody was a licensed doctor within US and practicing, even for them to do this kind of practice online is not fair. So just like a doctor should not do it, similarly, pundits and politicians should not do it as well. It is not correct to do. So I then ignore such messages. Similarly, there are messages where there is clearly something that is incorrect, or they are asking me to reach out to someone who I feel I don't have a great respect for. And then I don't respond to them, because that starts a debate. If I respond and say no because I don't respect this person, and they say, why don't you not respect this person? And then that becomes a debate. And I don't have time to debate every single person. So that is why there are some questions who I just not engage with. That is not a disrespect. That is not my cup of tea. T says, I've been with you since early 2020. You're a kind and humble hero. Thank you. Thank you. M says, I'm sorry, my first time here. Welcome. I'm listening because it's very interesting and you don't speak down to us. Absolutely. Look, if the intent is to tell how much I know, then I can wear a coat and I can have a stethoscope and I can speak in all jargons of medicine, and I can draw or take those difficult diagrams as medical professionals use. But that is not the intent. The intent is to say, can I take the knowledge, medical knowledge, make it accessible and explore it together with the audience? So I don't want to regurgitate. You can read that from a book or an article or a study. The idea is, can we work on it together? Can we explore it together? And it is so fun for me that even after exploring it together, I'm actually fortunate that people go away with varying impressions of that information. And that is the fortunate part, that you don't bias people so much that they just go with what you want them to go with. Instead, it is actually the success of providing information that somebody listens to that and says, okay, I have this impression of it. Somebody else listens to that and says, I have this as my takeaway, or my opinion is formed this way. That is a success of neutrality, although me being a human being cannot always be neutral. For example, I will choose those topics that speak to me. So that right there is a bias and a filter, right? And then I would, within the articles, I would choose those parts of the articles that speak to me. So that is another bias. So I cannot say that what you get from here is biasless, but at least I keep opening up my biases like a surgical procedure, so that you're not blind to them. You are very welcome. This is true. So I'm thankful. So Helene says, I cannot understand the virtual present in the world right now. I'm sorry, some of this. It is actually interesting, and I worry about this a lot. And I say it in my talks, that an underlying pandemic is the psychological well-being. Generally, I think with the pandemic, one, we have created, I mean, look how many people got affected. Daily death counts, lockdown, away from the friends and family. I was talking with a patient, and she was saying that I used to be very outgoing and extrovert. And since I've been 2 years in home, I have become psychologically upset. So I'm so glad that we can start going and meeting. And so, how many psychological scars have we developed during this time? It's not even something that is seen. If somebody's arm is broken, you can see it. But if somebody's psychological state is broken, we cannot even see that. And so I see everyone on edge. Somebody is financially in trouble. Somebody is emotionally in trouble. Families are in trouble. So a lot of uncertainty, a lot of loss of control, a lot of distrust. There was just a lot that happened. And I was talking with Dr. Paul Marik yesterday, and I was saying, I don't even know if our generations will recover from these scars, or how long will we take to recover from this impact. Yes, people were weaponized against each other. Yes. Christine. Yeah. Yeah, correct. Lockhart says we don't have a control group. Jason says, who cares what people think? You're correct. But at the same time, if somebody is in my home, in my living room, so let's say if I have an event, and then there are 6 friends of mine, And one of them just sits down there and just starts, you know, doing things that may not be for that little event, then they may not be a good fit for that time. Exceptional human being. Thank you very much. Thank you, lady. George, in the competition, not carefully considering audience, creating an excellent way for learning. Yes, you're a Howard too deferential to your cat. That is true. Luffy would. Yes, Luffy does a lot of controlling. Susan says, I hope you get a breaky break. So I've had some breaks and I just haven't had a breakfast yet. So once they come back, or maybe I should just quickly run and go get breakfast. Kathleen, thank you very much. Christine says, go eat. Okay, so I'm going to put this on so that if they come back in my absence, it is not stopped. I'm going to put this on. It is going to be some music now. Are you okay if I left it like this? Okay, cool. So I will be back. Let me just go eat a little bit.
Like, tell us when we're live. All right, and welcome back to the 173rd meeting for Vaccines Related Biological Products Advisory Committee meeting. I will now hand the meeting over to our chair, Dr. Monto, and our DFO, Dr. Prabha Atreya. Take it away.
Thanks, Mike. Welcome to everybody to the open public hearing session. Please note that both the Food and Drug Administration and the public believe in a transparent process for information gathering and decision making. To ensure such transparency at the open public hearing session of the advisory committee meeting, FDA believes that it is important to understand the context of an individual's presentation. For this reason, FDA encourages you, the open public hearing speaker, at the beginning of your written or oral statement to advise the committee of any financial relationship that you may have with the sponsor, the product, and if known, its direct competitors. For example, this information may include the sponsor's payment of expenses in connection with your presentation in this meeting. Likewise, FDA encourages you at the beginning of your statement to advise the committee if you do not have any such financial relationships. If you choose not to address the issue of financial relationships at the beginning of your statement, it will not preclude you from speaking. Prabha, over to you.
Thank you, Dr. Monto. Before I begin calling on the registered speakers, I would like to add the following FDA guidance. FDA encourages participation from all public stakeholders in its decision-making processes. Every advisory committee meeting includes an open public hearing session during which interested persons may present relevant information or views. Participants during the OPH session are not FDA employees or members of this advisory committee. FDA recognizes that the speakers may represent a range of viewpoints. The statements made during this open public hearing session reflect the viewpoints of the individual speakers and are their organizations only, and are not meant to indicate agency's agreement with the statements made. With that guidance provided, I'm going to first call on the first OPH registered speaker, Mr. Benjamin Newton. You have 5 minutes to speak. Thank you.
Thanks so much for your time. I really appreciate it. So the question we have here, right, and this is always the question, is how can we save the most lives? This is the question that constantly faces this committee and all of the FDA. The question before VRBPAC today is easy to answer. Authorize Novavax as vaccine. It's highly effective, something we knew more than a year ago. Plus, it hurts less than the mRNA vaccines. It appears from the limited data that the FDA has shared the myocarditis issues are likely a result of data mining errors, but we will only know with more data. From my view, there's a clear benefit to a vaccine that hurts less. For example, I have an acquaintance who recently got sick with COVID because he didn't want to get boosted because the mRNA vaccines hurt too much. All of this is really an aside to the rest of my presentation. Next slide. So really the thrust here is about how we can better protect people, right? The question we have to ask ourselves as a regulatory body is how best do we serve the people of this nation? Should we provide people with options, or should we stand in the way of people protecting themselves? Today, children under 5 still have no access to vaccine nearly a year after the American Academy of Pediatrics recommended approval using sero-bridging data. Today, Omicron-specific boosters have not yet been approved 6 months after we knew that they were required. Today, we all suspect that antibody half-life long-term will be about 90 days. So we will need boosters about every 6 months. However, it is very challenging for people to get access to needed boosters. The question we are trying to answer is not if these vaccines are safe, but rather, are they safer than the alternative? They are clearly safer than COVID. Next slide. So why was Novavax delayed? Novavax's vaccine demonstrated efficacy superior to J&J's vaccine before J&J was approved. Instead of approving Novavax in January 2021, the FDA required a second phase 3 trial, one of the best vaccines, delaying approval. At this point, Novavax's vaccine has been authorized for use in more than 100 countries, courtesy of the WHO. Like all approved vaccines, this could have been approved sooner. What went wrong at the FDA? What can we do better next time? These are questions that only the FDA can answer. Next slide. The FDA prevents access to life-saving medicines. Why? In fact, that is the entire point of the FDA. Block access to drugs to prevent dangerous drugs from entering the marketplace. This is a constant balancing act, one that is impossible to get right, but the FDA must constantly work to improve. Some reasons for delayed approval are likely to be incentives. No one gets fired for being conservative with drug approval. FDA committee members derive revenue from illness and or clinical trials. This is to be expected as it's populated by doctors. You are all likely familiar with the trolley problem. People feel differently about having agency in an outcome versus just letting people die by preventing access to medicines. The FDA does not quantify the mortality and morbidity associated with both action and inaction. We count the people injured taking bad medicines. We don't count the people injured by lack of access to good medicines. Diversity matters, and it's highly important for effective decision-making. There's almost no diversity on VRBPAC, partially by design. And someone is not on mute. So we know that there are problems. It would be surprising if there were not. So what can the FDA do to improve? You can set clear guidelines for approval of vaccines for all known pathogens today. You can create data standards and automated data feeds for clinical trials. You can publish this data in real time, redacted of personally identifiable information, allowing for real-time analysis. You can quantify and assess the risks of both action as well as the cost of inaction. You can increase diversity on FDA committees. You can create a process to authorize vaccines in 30 days for all ages. Then name a pathogen and let the FDA and the industry practice. We are going to have another pandemic. It's only a matter of time. And this group is the group that can best prepare us for that eventuality. Next slide. On slide 6. Our future is bright. So this may have seemed like all doom and gloom, but it's not. We have this bright future. There are a few hundred viruses that cause disease in humans. It's a tractable problem. What is nice is that vaccine development is actually very cheap, less than $1 million per vaccine. Unfortunately, clinical trials cost hundreds of millions of dollars, with most of the cost occurring during phase 3. With modern statistics and a highly effective vaccine, phase 3 trials are a measure of how effective and safe a vaccine is, not if it is safe and effective. This allows regulators the ability to streamline and standardize trials and data flows, reducing costs, increasing the speed of innovation. If we want people to stop getting sick and dying from viruses, the safest course of action today is vaccination, a process impeded by a current regulatory environment. I really thank you for your time and attention to this matter. You've done so much work over the last couple of years getting these vaccines approved. Thank you.
Thank you. The next speaker is Ms. Kathy Cavaney.
My name is Kathy Keaveney. Thank you for the opportunity to speak. East Korea, Japan, Australia, New Zealand, India, Switzerland, Thailand, Indonesia, the Philippines, the United Arab Emirates, 4 countries of the United Kingdom, 27 countries of the European Union, Canada, and the World Health Organization have all approved the Novavax vaccine. A vaccine developed by a company in Maryland funded with $1.8 billion of United States citizens' hard-earned taxpayer money, and yet it has still not been approved for the American citizens who paid for it. Why not? Why in the United States, bastion of freedom and personal choice, are Americans served up only one type of vaccine option, mRNA, while the rest of the world gets choice? When Operation Warp Speed was initiated, it funded 7 companies. Practically speaking, we are now down to 2, Moderna and Pfizer, and both are mRNA platforms. The FDA's response to the delay in approving Novavax has seemingly been 2 issues: the amount of trial data to be reviewed and the need to inspect Novavax's manufacturing. Question both. Your contemporaries on review boards across the world have managed to review Novavax's manufacturing data and expeditiously approved this vaccine. What is it about this data that is somehow more onerous for the FDA to interpret? A Wall Street Journal article attributed the delay to manufacturing inspections, quoting a source as saying, pandemic safety protocols made it more difficult for FDA inspectors to get to Novavax's overseas manufacturing sites. I fail to see why, in the midst of a global pandemic with millions dying, the FDA couldn't put on a mask, get on a military plane, and fly to India to inspect Novavax's one manufacturing site. Certainly many inspectors from other countries did. Throughout the last 2 years, I've learned a great deal about why others want Novavax. I've learned that there are American citizens who, after 2 years, still haven't left their homes because they have medical reasons that won't allow them to take mRNA vaccines. I've learned that many Americans have been fired or quit their jobs rather than be forced to take the one type of vaccine that their employers mandate. I've learned that many have had adverse reactions to mRNA vaccines. At this same meeting 2 months ago, this very committee heard from American citizens, many in tears, who described their adverse reactions to mRNA and pleaded with you to help. I've learned that Americans with dual citizenship have resorted to going to other countries in hopes of receiving the Novavax vaccine where it has been approved. Americans paying to go to other countries to get a vaccine that's headquartered 20 minutes from your office. I've learned that while we deny our own citizens the Novavax vaccine, we allow citizens from other countries who have been vaccinated with Novavax there to freely travel here. I've learned that Americans who want Novavax choice are not vaccine hesitant or anti-vax or afraid or whatever other terms the media likes to use to marginalize us. We're simply informed Americans who believe that this is a better, safer vaccine, and we want what our $1.8 billion paid for. And where is America's leadership on this? Full disclosure, I voted for President Biden, but I don't understand how our country can invoke the Defense Production Act to manufacture and import baby formula, yet in a time when millions are dying, can't figure out how to get Novavax from India, or better yet, help them produce it in the United States. A year ago, President Biden was quoted as saying, the problem right now is that we have to make sure we have other vaccines like Novavax and others coming on. Also a year ago, Ashish Biden's COVID czar had this to say about Novavax. Quote, I realize that the Novavax vaccine results won't get the same attention as we heard from Moderna, Pfizer, and Johnson & Johnson, but for vaccinating the world, this is huge. Very, very good news. Novavax is essential to vaccinating the globe. The fact that Novavax has 90% efficacy is awesome. And yet, still nothing. Awesome news for the globe, just not for the United States. An emergency use authorization based on today's meeting will certainly be a step in the right direction, but it would be largely meaningless if you do not follow quickly behind that with an EUA to allow people to choose Novavax as a booster to the Moderna and Pfizer mRNA vaccines already received. A week from now, on June 14th and 15th, the same committee will review Moderna and Pfizer's applications for immunizing 6 months to 4-year-old children. For some Byzantine reason, Novavax can't advertise in any of the countries where it's already been approved until it's approved by the FDA. Without advertising or marketing or media coverage or governmental promotion, parents aren't informed. Many don't know that elsewhere in the world, vaccine choice exists, and they should exist for they and their children. Parents don't have the time or energy to sit around and read up on all this, and you know it. Instead, they will rely on your recommendations. Because they are scared and wanting to protect their kids, they will rush to vaccinate our most vulnerable with the only option you've allowed them. You won't put the brakes on this, but you should, until they're given a choice. Parents should be able to make informed decisions about their children's health, and you should inform them. Which brings me to the media. You are complicit. I'm a mother, not an investigative reporter. I'm tired of this battle and have 8th grade math homework to focus on. Do your job. Ask questions. Quit regurgitating what you were fed. I challenge you to verify or discount every detail of what I've said here today. We have 1 week until this committee meets again to determine approval for mRNA in infants and toddlers. Again, parents should be informed. You should inform them. Members of this committee, you are charged with evaluating the benefits of this vaccine, but you also have the awesome responsibility of ensuring American citizens continue to have freedom of choice. Thank you for allowing this mom for Novavax to speak.
Thank you. The next speaker is Mitchell Goldstein.
Yes, I'm Dr. Mitchell Goldstein. I'm a professor of pediatrics at Loma Linda University Children's Hospital. The emergency use authorization request by Novavax for a vaccine to prevent COVID-19 in individuals 18 years of age and older should be granted. As reported by Dunkel et al. In the New England Journal of Medicine, data from 2 separate studies involving over 30,000 participants demonstrated a composite efficacy of approximately 90% in preventing significant infection. Although this data was collected prior to the presence of Omicron and other subvariants, this vaccine product is the first traditional protein-based vaccine to achieve this level of protection. The vaccine has been authorized for emergency use by the World Health Organization and can be used in over 170 countries worldwide. Current mainstream United States immunization regimens for COVID-19 involve the use of mRNA technologies. These immunizations have resulted in decreased morbidity and mortality when measured against the demographic considerations of an unprotected population. However, the frequent need for boosters and the broad public perception of these technologies as untested and thus untrusted demonstrates the need for a traditional protein-based technology that mirrors those of the more trusted traditional vaccine products currently on the market for other viral diseases. Further, the need to provide effective protection to pregnant women and their particular concerns regarding the use of mRNA immunizations and potential consequences to their unborn babies, as described by Hageman and Goldstein in Neonatology Today, provides further corroboration of the need for an effective traditional protein-based vaccine product. Please make this vaccine available to provide additional protection for those most at-risk groups. Thank you.
Thank you for your comments. The next speaker is David Charles.
David, you're unmuted. Make sure your own phone isn't muted.
Thank you and good afternoon. Thank you for allowing me to address today's Food and Drug Administration Vaccines and Related Biologics Products Advisory Committee. My name is David Charles. I'm a practicing physician in Tennessee, and I'm here today on behalf of my role as founding member and chief medical officer of the Alliance for Patient Access. The Alliance for Patient Access is a national network of policy-minded healthcare providers who advocate for patient-centered care. The Alliance is supported through associate memberships, grants, and donations from a diverse group of organizations, including Novavax and other vaccine manufacturers. The Alliance supports health policies that reinforce clinical decision-making, promote personalized care, and protect the physician-patient relationship. Motivated by these principles, Alliance members participate in clinical working groups, advocacy initiatives, stakeholder coalitions, and the creation of educational materials. On behalf of the Alliance for Patient Access, we would like to commend the FDA and the Advisory Committee on the important work that you have done in ensuring safety and the efficacy of COVID-19 vaccines throughout the pandemic. As we know, access to FDA-approved vaccines has slowed the spread of COVID and undoubtedly saved the lives of untold numbers of Americans. However, despite the work that the FDA and the emergency use authorization process has done in approving COVID-19 vaccines, there continues to be vaccine hesitancy among the American population. The World Health Organization has listed vaccine hesitancy as one of the top threats to global health. They further noted that the reasons some are choosing not to vaccinate are complex, but of lack of confidence in the vaccine is a concern. This lack of confidence can stem from a lack of understanding about the newer or unknown vaccine designs and technologies. While the mRNA and viral vector vaccine designs are available, they're still not well known to the general population, which makes additional vaccine options valuable. Having another vaccine design introduced, such as a recombinant-based design, may encourage those who are vaccine hesitant to finally become vaccinated. There continues to be many unknowns about the virus, variants, and long-term effects of contracting COVID-19. It is imperative that Americans have access to a variety of safe and effective vaccines to ensure greater uptake and to protect individuals and the public. As the virus evolves, additional vaccine options are important to meet the needs in keeping the Americans safe. The benefits of more COVID-19 vaccine availability, especially with those who are vaccine hesitant, greatly outweighs the risk of declining to authorize the use of safe and effective vaccines. We're asking the committee to strongly consider emergency use authorization of safe and effective COVID-19 vaccine applicants that use different, historically well understood vaccine designs. It is to the benefit of every American to have additional vaccine options other than mRNA, including protein-based vaccines if available. Thank you very much for allowing me to participate.
Thank you for your comments. The next speaker is Charles Ritter. Chad Ritter.
Okay, thank you very much. My name is Chad Ritter. I am a professor of nursing at Chatham University in Pittsburgh and have been promoting public health and universal vaccination of adults for many years. Thank you for this opportunity to address the Vaccines-Related Biological Products Advisory Committee meeting today to discuss the issuance of a emergency use authorization request for the Novavax vaccine to prevent COVID infection in individuals 18 years of age and over. I would like to put forward 3 reasons to support this EUA. First, there are currently approximately 258 million Americans who have received at least 1 dose of vaccine, and 2/3 of the population can be considered fully vaccinated. That's 221 million. Accepting these statistics, at approximately 1/3 of the population is still skeptical of the COVID-19 vaccine. How can we persuade those reluctant Americans to get vaccinated? The Novavax vaccine produces an exact replica of the spike protein of the COVID-19 virus that prompts our immune system to produce antibodies against the virus. This may help those who are hesitant, who are not supportive of the messenger RNA technology that comprise the first two vaccines currently under EUA. This vaccine does not involve utilizing any of the genetic functions in the human cell. Specifically, there is no production of messenger RNA to make new proteins within the cell. The vaccine uses proven technology by presenting antigens to the immune system, resulting in production of antibodies. Additionally, the Novavax COVID-19 vaccine includes a special adjuvant, matrix-M1, bonded to the particles in the vaccine. This matrix very strongly boosts immune responses similar to the adjuvant used in the Shingrix zoster vaccine. With this boost, even people over 80 years of age who typically have weak immune responses to vaccines can respond. Secondly, I have been actively working to promote vaccines to enhance public health in America for close to 20 years. My first publication promoted universal vaccination against pertussis was the result of my doctoral research project addressing an outbreak within a school district that resulted in over 70 cases in multiple age groups. The paper was published in 2010. Vaccines have been proven to help to prevent disease in all age groups, and the meta-analysis described the effectiveness of pertussis vaccine, showing that patients were more than twice as likely to contract pertussis. The vaccine doses containing pertussis antigen were missed or administered late. Third, during the past 2 years, I have been closely monitoring the COVID-19 pandemic while attending ACIP meetings as the ANA liaison representative and conducting research doing an academic sabbatical in fall of 2021. One result of that study was publication of an article titled COVID-19 Vaccine Hesitancy and How to Address It, published early this year. Points discussed included vaccine hesitancy describes an unwillingness of citizens to accept vaccines that are accessible and available. Currently, 1/3 of the population has not received any dose of vaccine and are at risk for significant disease, hospitalization, and death. We need another tool in the arsenal to address the concerns of this significant segment of the population. Secondly, not all healthcare workers are acceptance of COVID vaccine. A Reliance Media report documents that 20% of nurses refuse the initial vaccines due to questions about safety and efficacy. A proportion of the population, a third point, are grouped as in-betweeners, those adults who have taken a wait-and-see attitude. This group typically includes women, younger adults, and an ethnic minority background with less education. Common concerns include vaccine safety, skepticism about the risk of COVID-19, belief they are already immunized from prior exposure, and reservations about efficacy. There are many approaches to educating this hesitant population. It is key that we make another effective vaccine available that may be more acceptable to this broad and complex population. Hesitancy crosses many boundaries between gender, ethnic group, and socioeconomic classification. Let's not forget that social determinants of health can also influence vaccine acceptance. Some of those factors include political belief, education, low trust in scientists, where they live, where they work, where they get their news information and how they evaluate health risks. Just because someone in the family had COVID will not necessarily influence acceptance of the vaccine. I strongly urge the Vaccines and Related Biological Products Advisory Committee to approve this emergency use authorization to make another vaccine tool available to help achieve universal vaccination against COVID. With another choice available to doctors and nurses, we will have a better chance of convincing adult citizens to accept COVID-19 vaccine more readily. The Novavax COVID-19 vaccine could help break down some of those barriers of which many of us are aware and help achieve the World Health Organization's achievement of 70% coverage with COVID-19 vaccine throughout the world. Thank you very much.
Man, they should just talk.
Thank you. Thank you for your comments. The next speaker is Ms. Sophia Phillips.
Hello. Thank you for the opportunity to speak today on behalf of the National Center for Health Research. My name is Sophia Phillips, and I am a fellow at the center. We analyze scientific data to provide objective health information to patients, health professionals, and policymakers. We do not accept funding from drug or medical device companies, so I have no conflicts of interest. Today, the panelists are asked to evaluate if the benefits of the Novavax COVID-19 vaccine outweigh its risk for use in individuals 18 years of age and older. While this vaccine demonstrates similar levels of efficacy as compared to vaccines approved for COVID-19, the data suggests additional safety risks. As was stated in the FDA materials, there was an elevated risk of myocarditis and pericarditis demonstrated in Study 301. Further, this risk could be higher in the Novavax vaccine compared with mRNA COVID-19 vaccines. There were 6 cases identified pre-authorization of Novavax, while no cases were identified before the authorization of mRNA COVID-19 vaccines. Although these serious complications were also identified for mRNA vaccines, that was only when the much larger numbers of people were vaccinated, not the original mRNA study participants. Data from passive surveillance in other countries where the Novavax vaccine is authorized also indicate a higher than expected rate of myocarditis and pericarditis associated with the vaccine. As a result, the FDA requested that the sponsor change myocarditis and pericarditis to an important identified risk on the pharmacovigilance plan. The design of Study 301, which is the basis for today's discussion, initially resembled that of the 3 COVID-19 vaccines granted in EUA. They were similarly phase 3 randomized placebo-controlled trials with a similar number of vaccinated participants. However, when the study design transitioned to a blinded crossover due to the availability of EUA vaccines for certain populations, it weakened the value of the data. Efficacy of the drug compared to placebo could only be determined in the pre-crossover period after dose 2 for approximately 2 months before the opposite treatment was given to each participant. Therefore, it is not possible to assess sustained efficacy over a longer period of time. It remains unclear how long protection lasts. And while the FDA remains hopeful that Novavax will provide some meaningful protection against Omicron, that is also uncertain, since the vaccine was primarily studied on the Alpha variant. Additionally, very few of study participants were immunocompromised, pregnant or lactating, or at risk of severe COVID because of cardiovascular, chronic renal, and chronic liver disease. That made it impossible to meaningfully evaluate the vaccine's efficacy for those populations. Few cases of PCR-confirmed COVID-19 were analyzed for participants over 65 years of age, limiting the value of the efficacy data for that age subgroup. For those that were studied, there was a 12.5% dip in vaccine efficacy for individuals 65 or older, which is also typical for the mRNA COVID-19 vaccines. What would be the value of this vaccine compared to the 3 COVID vaccines that have already been approved? If it is less safe than the other 3 vaccines, it does not provide additional benefit to make up for that. Even if it is not proven to be less effective than the other COVID vaccines, it lacks long-term placebo-controlled efficacy data, and there's very little safety or efficacy data for the most at-risk patients. When we already have vaccines on the market that are FDA approved and based on much better data, why would the FDA authorize this vaccine? Wouldn't it just add to the controversy surrounding COVID-19 vaccines? Thank you.
Thank you for your comments. The next speaker is Martha Dawson. Ms. Dawson.
Good afternoon, everyone, and thank you so much for allowing me to speak today. I'm here today to support another technology to fight COVID-19 and the many growing variants. When one is at war, many different approaches are used by land, air, and water. Although the public is exhausted and ready for this pandemic to end, there really is no light at the end of the tunnel. As a nurse for 45 years and the current president and CEO of the National Black Nursing Association, representing over 500,000 registered nurses, licensed practical and vocational nurses, and nursing students nationwide. I am also fatigued from educating, testing, vaccinating, and addressing other health and social determinants that place Black and Brown population at risk during this pandemic. And we know that more of them have died. In addition, NBNA nurses have been on the front line. We have been serving every day. From March the 20th, when this first variant of the COVID hit our nation. Therefore, I encourage the FDA to give us another more traditional medical intervention in this fight and approve this vaccine. African Americans and Black nurses that I represent and give voice to are on the front line of this pandemic. And they continue to watch more of our population die. They have lost colleagues, spouses, partners, parents, children, and other relatives and friends. Let us be very clear. COVID-19 is a public health crisis. However, it has been politicized with myths and disinformation. Therefore, some people will never become vaccinated, putting others at risk. And unfortunately, people are not following public health policies and best practices. It appears that the world is just tired of wearing masks and washing their hands, isolating and being social distant. However, through the lens of public health, this is exactly what is still needed today. But again, since the majority of the population is not leaning in that preventive and health promotion direction, we have to look for other measures. And I do believe that having another vaccine will give people options. Maybe some of those that are still sitting on the sideline saying, well, let's just wait until a few more are vaccinated. But that put us at risk. And it brings to mind of me talking to a close friend this week. I mean, just this week. With another under 40-year-old relative ended up with COVID pneumonia. How many of our young people are we going to allow to die? Or we're going to allow to become sick? So again, it's unfortunate that people are not following public health policies and best practices. So we have to look for other options and give people more options. Many believe that these measures did not work and are not going to work. However, we see cases still increasing. Yes, again, we have a breather. It's now summertime. We can be out. We can have more fresh air. But make no mistake, within another 8 to 10 weeks, we're going to move back into our kids going into the school system. And we will probably see another spike if we don't do something. So as I stated, I represent nurses and nursing students, and the future workforce for this profession is not good. It is projected that over 1 million nurses will be needed in the United States, and over 6 million will be needed worldwide by 2030. My colleague, it takes 3 to 4 years just to educate one nurse. So if we continue to lose nurses because they are fatigued and they're tired and they just can't see one more patient expire from this COVID pandemic, then we're going to continue to have them leave the occupation and look for other things to do. So I want to just say, let's think about this, because yes, I know nurses and physicians and others in the healthcare space, they're also refusing to be vaccinated. But we need to continue to provide options to reduce excuses and as many excuses as we possibly can. This is why I strongly support and encourage the FDA to approve the Novavax vaccine. So I say, and I would like to close with this, in the art of humanity and public safety, since we as a country have not been able to lean in and accept that this is a public health crisis and that we should not only protect ourselves, but those around us, then we need to have other options. So let's look at this and say, is it going to do more harm, or does we lean that pendulum towards it could be the next thing that saves your life or your loved one life? Thank you.
Thank you for your comments. Last but not least, Mr. Kermit Kubitz.
Hello, can you hear me?
Yes, we can. Go ahead, please.
Okay, thank you. I'm a graduate of Caltech, the Harvard Law School, and the Harvard Business School. I have participated in BRB, PAC, and ACIP meetings, including the October 22nd, 2020 VRB PAC meeting on developing and licensing vaccines, the December 20th Pfizer EUA meeting, and the September 17th, 2021 VRB meeting on boosters. And I have no conflicts and no fiscal interest in the company being considered. The question to the ACIP is, do the benefits outweigh the risks of a 2-dose series of vaccination for persons 18 years and older for NVX CoV 2373? I look at this in the evidence in the form of a structured benefit risk table of the form previously adopted by the FDA. 1, the condition to be treated. 2, available alternative therapies. 3, the benefits of the proposed drug, including uncertainty. 4, the risks of the proposed drug, including uncertainty. And 5, the summary conclusion in view of all available evidence, including uncertainty, about the benefit risk and is it positive. Point 5 is similar to the question presented to the ACIP meeting. In my analysis, the condition to be treated is prevention of COVID-19, mild, moderate, or severe, including hospitalization and death. The alternatives are other vaccines, Moderna, Pfizer, as well as Janssen and AstraZeneca, and treatments such as Paxlovid. As one of the initial questioners on this panel asked, why EUA if there are other available vaccines? As other speakers before me have discussed, vaccine hesitancy is a serious problem. Some people may not be able to take mRNA vaccines, and we need as many tools as we can get to control and eliminate this pandemic. The benefit of the Novavax vaccine is elimination in the treatment arm of 17,000 patients of moderate to severe COVID-19. The risks of the Novavax vaccine are significant adverse events of about 1% for Novavax and 1% for placebo. The Novavax vaccine versus the placebo showed about no moderate or severe cases versus 11% moderate cases and 5% severe, or about 16% severe or moderate COVID-19 cases, which were not occurring with the Novavax vaccine. This demonstrates significant efficacy. In addition, the Novavax vaccine has now been administered to hundreds of thousands of patients outside the U.S., so there is available data on its effectiveness. See the study published in Cell by the La Jolla Institute of Immunology, Professor Daniel Weiskopf and Shane Crotty, who found antibodies after 6 months were highest with the Moderna, Pfizer, and Novavax vaccines. All participants retained a similar percentage of memory CD4+ helper T cells. It's important to have multiple vaccines ready and approved to fight COVID-19, to provide initial protection, to provide protection against variants, and to provide protection against boosters. As the CDC has noted, heterologous booster vaccination may provide significant benefit even if mixing and maxing vaccines. Finally, I'd like to thank the ACIP and the FDA. You have saved 1 million lives by your providing vaccination through all the vaccines, despite all the time it's cost you to attend these meetings. Thank you very much.
Thank you for your comments. Thank you for your comments and the presentations. And this concludes the open public hearing session. And we're going to be moving on to the next items on the agenda as we have finished with the pre-registered OPH speakers. Thank you. And I hand over the meeting to Dr. Manto. Manto, take it away, please.
Thank you, Prabha. Question is, do we have a break scheduled and reconvening at 10 minutes past 2? Is it possible to begin the meeting at 2 instead? Prabha? Dr. Marks? Or should we go to 2:10 as we are scheduled?
Let me just check with our technical people, but as long as they say that we can move ahead, well, we will move ahead.
Yep, we're ready. We can move ahead if you'd like to.
I believe in that would be wise to do. Thank you, Dr. Manto. Go ahead and proceed.
Okay, well, we're moving ahead now to the additional question and answer session, which regards both the presentations of the sponsor and the FDA. And Dr. Marks, would you like to say a few comments before we go ahead with this session?
Yes, thanks. Yes, thank you. Thanks very much. So there have been some questions about how the Novavax vaccine will fit into the other vaccines. And I think what we need to say here is that we are here to consider the authorization for the primary series right now. That means that this is the initial step. There will be additional submissions, I am sure, and additional consideration by FDA of both booster doses, additional populations, as well as potentially the activity of this vaccine or variant vaccines using this technology submitted in the coming, that will be submitted or considered in the coming weeks to months. So I think we just so that the advisory committee meeting members think about this. There will obviously be some evolution of this. I believe it's fair to say that, and you can certainly feel free to ask the company that question as well over the coming weeks to months to essentially make it consistent with the vaccine paradigms that we are using now. Dr. Monto, is that helpful to the committee?
That's very helpful, and perhaps I could start the discussion by asking the sponsor if there's anything further that they would like to tell us, which might help in our deliberations, given the fact that all the testing was done in the era of Alpha, and we're now preparing to launch a vaccine in the era of various Omicrons. So does the sponsor want to give us any additional information that might be valuable to us?
Sure. And then, Dr. Monto, should I start off with that topic, or do you want me to cover some of the questions that we deferred from previous week?
It's up to you. We have more time than I anticipated, so we can have an in-depth discussion, which is very helpful.
Can I make a suggestion? Why don't we stick to that? Since I just mentioned that, maybe it would be good, if you don't mind, just to address that question now, so we take care of that issue before moving on.
Okay, sounds good. So let me parse it apart. So a couple things we talked about. One of them was expanding our indication beyond adults greater than 18 years of age. And as we've already talked about, we completed studies in adolescents 12 to 17 years of age. And those studies have been the basis of approvals in other territories. And certainly, as soon as we achieve EUA in the US, our intent is to file that and to seek regulatory approval to expand that indication. We've also have data on boosting, both homologous and heterologous boosting. And once again, that's the sort of data which we are going to bring to the FDA to seek approval for the booster indication as well. So it's true that the efficacy studies we conducted were conducted in the era before Omicron emerged. What we do know is the data that we've showed you is that the vaccine works well against the variants that circulated during the conduct of the study. And there were a broad number of variants. And, this is a feature, we think, of our technology. So the recombinant proteins that are made in insect cells, which give benefit to antigenic spread along with the adjuvant system. And this is proved to be true in our influenza vaccine, where the immune responses were shown to recognize a broad array of H3N2 drifts as well as ancestral strains. And it's true in the efficacy data we showed you that showed that the vaccine worked well against the variants that circulated. We additionally have immunologic data from our studies, which look at how they respond to the Omicron variant. And perhaps we can show some of that now. This is data from the U.S. adolescent study. And I bring this up because we don't have the comparable data for the adult data. But what you can see is the immune response against the original prototype on the left-hand side, and the immune responses against the various variants that circulated, including Omicron on the far right-hand side. And this is the BA1 version. Now, what we can also look at is data from our previous study, our 101 study. Once again, what this is looking at is immune responses, IgG, after 2 doses and after 3 doses. 2 doses in dark blue, 3 doses is in light blue. And you can see we get a nice boost against all those variants with the 3rd dose. Importantly, if you look at Omicron and look at the values achieved after 3 doses, it's really comparable to what we saw after 2 doses to the original. And those were the kinds of immune responses that were comparable with 90% protection in our efficacy study. Now, this is binding. How about neutralization responses? Here, what I'm showing is a comparable graph, original on the left-hand side, Omicron on the right. And you can see there's a good boost, once again, between 2 doses and 3 doses. But importantly, between Omicron, after 2 doses, there's only 3.6-fold difference than from the original. And this is data generated by the Matt Fryman lab and the University of Maryland. So we have good confidence that not only are we generating binding antibody, but at least in this assay, induction of neutralizing responses. So overall, it's factual that we don't have efficacy data against Omicron, but what we do have is a technology that we think generates a broad immune response, demonstrated against a broad array of variants.
Thank you. And now that we've had that question answered, you had some other information that you wanted to give us. So let's go on to that. And then we've got hands raised. We've got questions from the members.
Okay. So there was a question asked by Dr. Meisner about IgA responses. And what I'm showing you here now is data from rhesus macaques. And what we're looking at is IgA titers in the vaccine group versus placebo group. And on the left-hand side of the panel, we're looking at upper airway. So these are nasal IgA. On the right-hand side, lower airway, so bronchial alveolar lavage. And you can see that the vaccine does, in fact, induce IgA in both the upper and lower airway. And this was associated with sterilizing protection in this animal model in both the upper and lower airway system. Now, I mentioned some data earlier about the ability that we have to stop infection, whether it be symptomatic or asymptomatic infection. And I wanted to bring a complete read of that. And what I'm showing you here is the 302 data from the UK on the left-hand side I mentioned, and comparable data in the US on the 301 study. So this is the ability for the vaccine to block all infection, whether it be symptomatic or asymptomatic. And the only difference between the 302 and the 301 study is the time, the median time of surveillance in the UK study was longer. It was 100 days versus 60 days in the US study. And the point, once again, being is the sterilizing protection and the IgA that we saw in the animal models may be a signal. This is what we're seeing as far as the ability for this vaccine to protect against all infection. And like I mentioned, obviously, if you stop infection, you have the ability to prevent long COVID and transmission. Now, there was a question asked by Dr. Pergan about efficacy after dose 1. And what I'm showing here is data from the US study, the 301 study. And you can see that after dose 1, there was totality of 133 cases in the vaccine group and 156 in the placebo group, 2-to-1 randomization. And that gave a efficacy of almost 59%. And after dose 2, it was 86% in this analysis. And this is a FAS analysis, so it's like an ITT analysis. So it doesn't take into account the observation window, which starts 7 days after post dose 2 in seronegatives alone. Now, the efficacy that you see at 58%, that includes the time frame after dose 2. So a lot of that efficacy is attributed to the time period after the second dose is administered. There was a third question asked about by Dr. Brian about the Latinos and the proportion that was in those greater than 65 years of age. And in fact, there were no cases in the Latino group in those that are greater than 65 years of age. And I think that's what I did think that perhaps you'd be interested in the immune responses we talked about during the main presentation. On the left-hand side, you see the day 0 values. On the right-hand side, you see the day 35 values. In dark blue are the Hispanic Latino participants, and in light blue are the non-Hispanics. And you can see that the Hispanic population, in fact, had a slightly higher IgG titer than those in the non-Hispanics. And when we look at neutralizing responses, we see a very similar pattern. Once again, a slight increase in the dark blues representing the Hispanic population at the peak immune response at day 35. So I think those tidy up the questions that were asked prior to the break.
Right. And now we're going to be moving into the discussion. And I want to remind the members that not only is the sponsor here to answer questions, but also the FDA representatives. So, Dr. Offit, you're up next.
Yeah, thank you, Arnold. This is directed, I guess, to both the FDA and the CDC presenters. I agree with the FDA's assessment that handful of cases of myocarditis that occurred within 3 or 4 days of receipt of the second dose of vaccine in young men is consistent with what was seen with the mRNA-induced myocarditis. So I think that is likely a causal, not coincidental association. It's also interesting in the document that the FDA handed out to us, that they referred to a 2020 paper where there was suspected molecular mimicry between SARS-CoV-2 spike protein and the heavy chain of alpha myosin on cardiac muscle cells. If that's true, then you would argue that really all COVID vaccines, as well as COVID itself, should cause myocarditis. But that may well not be true. And this gets to Dr. Rubin's question. We really need to know whether or not this is true for the vectored virus vaccines like J&J or AstraZeneca. We really need to know whether this is true for a whole inactivated viral vaccine like Sinopharm's vaccine, which has now been administered to millions of people, or whether it's true, I think most interestingly, for the Corbevax vaccine, which is a receptor binding domain vaccine, in other words, a truncated protein vaccine that's been given now to many people in India. I think it's incumbent upon us to know this, so to know whether it's about the protein itself or whether it's about the way the protein is being processed, so that we can use that knowledge to make safer vaccines for a disease that is going to be with us for decades, if not longer. So I think this is a real opportunity to learn something, and I hope that it's not lost. We need to get the data, the kind of data that Dr. Rubin was referring to earlier. Thank you.
Who's up to answer this rather critical question? I think the FDA, I think in the briefing document, you raised the issue of mimicry. So it's, why don't you try to answer it first?
I'm sorry, who you're calling upon, Arnold?
I'm calling on FDA since it was in the briefing document, the reference to the issue of mimicry.
There's Dornan. Dornan, I'll unmute you. There you go.
Okay, thank you. Yeah, so Dr. Offit, I couldn't agree with you more that this is a critical question to understand whether vaccine-associated myocarditis is a class effect related to S protein antigen, and if so, whether there are other features of specific vaccine platforms that mitigate either positively or negatively toward a risk of vaccine-associated myocarditis. I think that the situation is clear for mRNA vaccines. We have some preliminary evidence from the clinical trials of this vaccine from Novavax that raises this concern, although I think we need more data from post-authorization use in larger numbers of individuals to really get at what the rate of myocarditis associated with this vaccine is and what exactly the risk is. As you heard earlier from Tom Shimabukuro from CDC, as we accumulate more experience with the Janssen vaccine, which has been used to a much lesser extent than the mRNA vaccines here, as well as outside the U.S., we are continuing to evaluate the occurrence of myocarditis after that vaccine. And of course, there's the AstraZeneca vaccine and other platforms that you mentioned as well. I couldn't agree with you more that we really need to look closely at these events and also to do the work necessary to understand what the mechanism might be. And so I guess from an FDA perspective, we would endorse that viewpoint strongly, and of course are here to assist vaccine manufacturers and the research community in addressing this very important issue.
Any further comments from CDC on this or from the sponsor?
Yeah, a couple of points to what Dr. Offit said. I mean, it's curious in the data we saw that the third boosting dose, there wasn't as big an increased risk after dose 2. And that makes me wonder if there are other mechanisms at play than that has been proposed. And we also know about other vaccine-associated myocarditis from the smallpox, monkeypox, and they don't even have the spike antigen. So I think the story isn't completely written here, and we do need to more fully understand what's going on before we can think about a class.
Hi, this is Dr. Shimabukuro from CDC. Can you hear me?
We can.
Dr. Offit, I would just reinforce what Dr. Fink said about the importance of getting the data that you mentioned, and I'm not so familiar with some of the other vaccines that are used outside of the United States, but I'm not aware of a similar association observed with the AstraZeneca vaccine, which was widely used in Europe. Similar association as is seen with the mRNA vaccines. And with respect to disease, my understanding is that isolated myocarditis following COVID disease is pretty rare. And the adverse cardiac outcomes that you see after disease are often in association with MIS, which may be a different mechanism altogether. But with respect to disease and just pure myocarditis or isolated myocarditis, it's a fairly rare occurrence after COVID.
Thank you, and thanks all for a discussion of an important topic that we need more information about. Dr. Levy.
Yes, I wanted to thank the sponsor for showing the additional data a few moments ago with some slides about antibody responses. And I wanted to know, those data were elegant, they were helpful, and yet without knowing what level or concentration of antibody correlates with protection, it's a little hard to draw any conclusions as to whether this vaccine, how it would perform against Omicron. I mean, it seemed like there were lower responses, lower binding and neutralization of Omicron than the other variants, yet there was some. But without, maybe I'm stating the obvious, without a correlative protection, it's hard to draw a conclusion one way or another. The sponsor made some comments about their impression of the correlative protection earlier in the day, and I'm wondering if they can make some further comments in terms of where they would see the correlative protection to be on those graphs, and does Omicron reach up? I realize there's an element of speculation here, but it is the elephant in the room, isn't it?
Yeah.
Dr. Dubrovsky, do you care to speculate?
I will always speculate, but I agree that without definitive data, we won't know. Listen, we think that we simply don't know if an Omicron-based vaccine is required, right? Those studies are ongoing. They're ongoing we sponsor a study in Australia, but we're looking both at an Omicron vaccine as well as a bivalent format to see if that offers any advantage. What we do know is what we showed you, is this technology in general does a good job with antigenic spread and having a broad response. And we do know that the binding and nuke responses we see are relatively favorable. And the best I can do to compare, and I understand this is very, very fraught with potential error, is to try to compare it back to the levels we saw after dose 2 to prototype. And as you saw for prototype in the US study, we had 97% protection. So where is the cutoff isn't clear. The signals we're getting right now is in our view favorable, but we'll know for sure when the study reads out in Australia.
Thank you. Dr. Pergam.
Thanks, Arnold. I had a question about the adjuvant in particular. I know that the adjuvant comes from a particular tree. I believe it's in South America. it is pretty, my understanding is that it's a highly regulated supply chain. Do you guys have any comments about the ability to get this on a regular basis to make the vaccine available? Because I know that's an issue.
Yeah, so we took steps early on to secure the supply chain. There is a zero supply problem with the adjuvant.
Great, thanks.
Thank you. Dr. Lee.
So thank you for that presentation. I think one of the questions I had earlier, and I think this is really for the sponsor, had to do with the current design of the protocol, which actually was a crossover. Most of the data we're seeing for efficacy is obviously related to the first the first part where they randomized obviously the vaccine and placebo. And I do understand the study is ongoing for the crossover part for the vaccine to placebo and placebo to vaccine. And I think the comparison of the second part after the crossover would be very illustrative in terms of what kind of carryover effect you might have from those that started with a vaccine and then were getting placebo. Do you have that data, or when do you anticipate we would be able to see that?
Yeah, so you're right. The crossover complicated or eliminated our ability to look at placebo-controlled data at the crossover time. Furthermore, we've taken the opportunity to boost those participants with both dose 3 and some with dose 4, which complicates the story even more.
Yeah.
Now, without a good comparator and with new variants emerging and the different courses of infections across time, it becomes extremely difficult to do anything but make model-based assumptions. Now, that work is ongoing, and we'll have that available in due course. I'm not sure how trustworthy it is because I'm unconvinced we know how to guess the efficacy against the variants until we get real data on that.
Okay, thank you.
Thank you. Dr. Galen.
Thanks. So thanks for all this. I want to start by thanking the public who commented, and then the many, many more of the public who commented in writing that didn't get a chance to speak. This is an important part of these conversations. Philip mentioned a couple of times something that doesn't always get mentioned about sterilizing immunity. So I'd like to hear some more about that. He showed us a little bit of data about it, but the degree to which you believe this vaccine can lead to sterilizing immunity, and any data that you have from anywhere that might have insights about its limited or its ability to dampen transmission. And then finally, we hear a lot about its authorization in other countries. It'd be interesting to know about how much it's being used in other countries and what other data you might have that's relevant to these discussions today from other country experiences.
Yeah,
very broad question.
I think I got these, so we'll see. So as far as the sterilizing protection data, it's a broadly feature we've seen in the animal models we've tested. And our best data for what we saw in humans is the data I showed you. So there's no direct measurement that we have in hand of transmission. You need to make that leap of faith or logic that if you don't get infected, you can't transmit. The durability of that period where you're protected from infection is also variable. In the UK study, like I mentioned, that was measured across those cases were accrued over 6 months with a median of about 101 days observation in those groups. So I think it's speculative, frankly, but we're hopeful. As far as real-world evidence from the doses that are administered, it's still early days for us. We are shipping doses; they're being used. Right now, you heard from Dr. Kim from the pharmacovigilance side. We have good line of sight to about 770,000 doses having been administered. We have imperfect visibility into this. Our customer are the governments. The governments deploy those. So we, to a certain degree, rely on the governments to feed that data back to us so we understand how many are used. For us to get into real-world effectiveness, which we want to do, and we will do, we're committed to doing it, we need to get the vaccine usage up and up in certain areas where we can do a test narrative control design. Without adequate doses being deployed, we can't do those studies.
Good response. They should not overcommit.
Does that answer all your questions, Bruce?
It did. Thank you.
Okay. I just wanted to interject a question of my own for Dr. Dubovsky, and that is we've heard that there are differences in the vaccines that are being authorized for use in other countries versus the vaccine that we're now considering for the United States. Would you speak about that and how different are they? Was it just a question of where they were manufactured? What's the story there?
Right. So all vaccines that are being distributed globally, commercially, are being made in a single facility and by our partners in Serum. That includes the vaccines which are being deployed around the world, as well as the ones that will be initially deployed in the U.S. As far as the previous studies that were done, all the clinical and commercial lots were released after being tested to assure they met a set of critical quality attributes. And this includes the lots that were used in the early studies in Australia, US, UK, South Africa, as well as in the US-Mexico study. And it's normal for these specifications to tighten as experience is gained with the manufacturing process. Now, we've completed a comparability program that we believe demonstrates the comparability between the early lots and the lots used in Study 301 and the commercial lots. We acknowledge the FDA has a perspective on this that's different from ours. But the quality of that material and the results of all the studies are really the basis of our global licensure. So I think that's where we stand. But importantly, all the vaccine which is being deployed commercially comes from a single facility.
Thank you. Dr. Meisner.
Thank you, Dr. Monteau, and thank you, Dr. Dubovsky. I feel like you're carrying a heavy load here. You're facing all of these questions. But let me, I have a question about the adjuvant, two questions about the adjuvant. The first one, and maybe you simply don't know, but what happens if a person gets at the same visit or the same day 2 Sapponin containing adjuvants? For example, if someone were to get the shingles vaccine that contains AS01 on the same day that your vaccine is administered, are you worried about an increased risk of adverse events that might occur? And then secondly, a little bit, one of your earlier studies with respiratory syncytial virus that I thought was very interesting because you really broke new ground with that publication. And but during the study with the same platform, I think, using RSV fusion glycoprotein, used a different adjuvant. You used an aluminum, an adsorbed aluminum adjuvant. And I think that was because there was some concern about using the Sapponin-based adjuvant during pregnancy in a person who might be pregnant. And so I'm assuming you no longer feel that that's a concern, and that's why you haven't expressed any reservation in that setting. Over.
Yeah, so the RSV maternal program, which was a study done before my time, was also a study done before the company had the Matrix-M adjuvant in its portfolio. The immune responses induced by alum were thought to be quite good at that time. We don't have any specific concern with Sapponin. The reproductive tox studies and have been clean, and certainly in the data that Dr. Kim presented, we didn't see anything that looked concerning to us. And I'll remind you that the amounts of, just back to your previous question, the amounts of adjuvant we're deploying are very low. We have 50 micrograms. We've previously tested doses that are higher, up to 75 micrograms for our quadrivalent influenza vaccine program. We know that there are a small number of people who received Shingrix in our study, less than 10, but those probably weren't co-administered. Those were just given close by, but we didn't see any concerns with that.
Thank you.
Thank you. Dr. Perlman.
Yes, so I have two questions. One,
what will it take for this to obtain a full licensure as opposed to an EUA? Because we keep hearing about why it should be an EUA instead of a full licensure. But what more would be needed to obtain full licensure? And the other thing is, with the adjuvant, is there any? I know you looked for things related to autoimmune disease, but was there any hint of exacerbation of pre-existing autoimmune disease with this adjuvant?
Yes.
Okay, I'll handle the first question, and perhaps I'll turn to Dr. Kim to talk about the second question. The long pole in the tent, the thing that takes the longest to get a BLA, is to do a lot-to-lot consistency study. And this is a requirement that's unique in the U.S. And to do that study, we need to generate lots which are deemed to be comparable and appropriate for such a study by the FDA before we do the study. So that's really the thing which is going to take the longest. We have some additional data requirements for length of follow-up, and those we'll come to terms with in discussions with the FDA before we bring it to them for the full BLA. Dr. Kim, do you have a perspective on enhancement of autoimmune disease?
Yes, can you hear me?
Yes, we can.
Yes.
And seems like my camera's not on.
There you go. I'll turn it on for you, sir. I'll turn it on for you, sir. There you go.
All right, thank you. Yeah, so we've seen very low frequencies of potential immune-mediated conditions. And so whether it's new onset or potentiation of existing comorbidities that some participants had in our study population, we didn't really see any patterns that suggested a worsening of conditions.
Thank you.
Dr. Sawyer.
The point has been made that one of the main audiences for this vaccine are vaccine hesitant people who, by now, many of whom have been infected with natural disease because of their reluctance to get vaccinated. And we've also discussed the fact that this vaccine has been used in other countries for quite some time. Perhaps I missed it in the presentation, but I don't think we heard much about vaccine reaction in people who had prior exposure to the natural virus. I think the 301 study specifically was in seronegative individuals. So I'm wondering if you could either remind me or share any information that we have about the experience with the vaccine in people who've already had infection.
You might as well stay on, Dr. Dabrowski. I think you're in the hot seat. Please go ahead.
Yeah, I mean, you remember correctly that 7% of the people in study 301 were positive at baseline, and while they were excluded from the efficacy analysis, we do have data, both immunologic as well as safety data on what the vaccine does. And let me see if I can get that data pulled up. But in general, if I were to summarize it, what we see is we see really quite a nice boost of the immune response in people who were previously vaccinated. Let me start with a slide that shows you what the neutralizing responses were by age group in those that were seronegative. And you can see overall, in those that were greater than 18, it's a value of about 1,000. And those greater than the younger group, 18 to 64, it was 1,200. When you compare it to the values that we see in those that were seropositive, what you see is an increase of roughly 3 to 4-fold. So they're getting a nice priming response from the natural infection. This vaccine boosts really quite well. From a safety perspective, we didn't really see any difference in the reactogenicity of the vaccine when it was delivered in the seropositive versus the seronegatives.
But
Thank
why
you.
would you recommend a vaccine to somebody who was infected and recovered successfully?
Quickly to tell us how many people, I know it was 7%, but what is that total that you had experience with?
So in this particular study, it was 7% in both groups, so 7% of 30,000. So you can do that math.
Thank you. Thanks.
We have additional exposure data in people who are seropositive in our other studies. The attack rate was very, very high in South Africa, for instance, where a much larger proportion were seropositive. And we see the same pattern. We don't see any increased safety signals, but we do see an increased immune response.
Thank you. Dr. Fuller, did you have your hands raised? I thought you did.
Hi there. So I have two questions. Can you hear me? Okay.
Yep.
I have two questions. As an allergist, I would be remiss if I didn't ask whether you observed any allergic reactions to the vaccine. After all, it is a protein-based vaccine rather than mRNA, so the risk of having an allergic reaction might be higher. I guess you can answer that one first, and then I have one more question.
Yeah, I'll give a scientific spin to it, and then I'll pass to Dr. Kim, actually. So our adjuvant gives the Th1 bias. So in a sense, it's slightly anti-allergic as far as that goes. So I think that plays in our favor. Dr. Kim, do you want to review the hypersensitivity and we know anaphylaxis, as has been mentioned in the pre-licensure database? Dr. Kim?
Yes. So as to confirmed anaphylaxis, we had no cases of anaphylaxis in our clinical development program, pre-crossover or post-crossover. Certainly, we do a broad search to look for any type of allergic type reactions or hypersensitivity type reactions. And I can show you some of that data here. And so as you can see, the pre-crossover, and we used a standard measure query, and that's a very broad search. And so any sort of events that could possibly be related to allergic type reactions. And we saw a minor imbalance, a numerical imbalance. And so you can see 0.77% in the active arm compared to 0.57% in the placebo arm. And the most frequent preferred terms or events was rash, as you can see there. And we have that in exposure-adjusted incidence. So 0.93 events per 100 person years versus 0.90 events per 100 person years. That's fairly balanced. And the reason we did that is because there's differential follow-up, oftentimes, especially when you consider post-crossover between placebo and active arms, or those who receive vaccines, because everyone will eventually have received vaccines. That small numerical difference is mostly driven by urticaria and dermatitis. And so we didn't see clinically significant sort of patterns and associations here.
So presumably the patients would be advised to do the standard 15-minute wait after the vaccine and not a prolonged wait or anything like that. My other question is you're applying for emergency use authorization. This is kind of a continuation of Dr. Perlman's question, and yet we already have two vaccines available that are highly effective and relatively safe. I haven't seen, your vaccine seems to be comparably effective and comparably safe to the other ones, but you didn't show that it was superior in any particular way. Since so many of the people in the United States have already been vaccinated, I assume that the it's going to be promoted largely to the vaccine hesitant individuals who might adapt a more conventional vaccine that's protein based rather than these other technologies. Do you have any information from vaccine hesitant individuals suggesting that they might be more willing to consider getting this vaccine as opposed to one of the other vaccines? Have you talked to vaccine hesitant people, or have any sense of whether they would be willing or more interested in using this vaccine than one of the others?
That's not
Yeah, so I'm going to ask Dr. Poland to step in and give his perspective. But I have to say that 1 in 10 Americans has yet to be vaccinated, and we haven't given up on them. We heard in the open public comment period that there seems to be a desire to use this product And that's what we want to bring to the U.S. population. It's another option, a choice. Now, whether the proportion that choose to be vaccinated from a primary series, that isn't clear. We'll find out. We do know that in countries where the vaccine is being deployed, it is being used both as a primary series as well as a booster. And those are choices that people are making in those countries to choose our vaccine. Dr. Poland, do you have any other perspective?
They shouldn't have this discussion. It's really up to a person to decide. EUA should not be based on this, that somebody would be vaccine hesitant and they would like it. Yes, it's another choice.
Arnold?
Hello.
Yeah. Are we waiting on somebody?
We're waiting on Dr. Poland, who was called on.
Oh, there we go.
No, I don't know that he's not coming on. So maybe we'll take that as the sponsor's answer for the time being.
Okay, I see Dr. Reingold. He's not in my regular list. He's up among the presenters, so I don't know how long he's been waiting.
Hi, can you hear me?
We can.
Good. So one of the problems of coming late is lots of questions that have been answered, particularly the one Dr. Sawyer asked. But I do have one other question, building on what Dr. Meissner mentioned. Hey, will we fall soon? We'll be giving a lot of flu vaccine to people. And I don't know about other people, but I got my flu shot and my booster dose of COVID in different arms on the same day. And I'm just curious what you know about the administration of this vaccine at the same time people get a flu shot.
Yep, that's a question that we have also been very curious about. In the UK study, we actually included a cohort of participants who received with the first dose a dose of licensed influenza vaccine. And what we saw there is that we didn't negatively impact the hemagglutinin responses. However, what we did see is a decrease in the anti-spike responses in that cohort. Now, we still maintain efficacy. Efficacy was maintained at pretty much exactly the same rate as the overall population, but it did drop the anti-spike response. This isn't unique to our platform. There are publications that show that with other platforms, when you give flu vaccine, it tends to drop those responses, including against mRNA vaccines. We furthermore conducted a combination study with our flu vaccine and our COVID vaccine, and this was made public a few months ago. And we recapitulate the same finding. We do, in fact, impact the anti-spike response, but you can overcome this response by minimally decreasing the hemagglutinin while increasing the spike antigen. And that's a combination product that we're taking forward as well.
Thank you. Dr. Marasco.
This is a question for Dr. Drbeen. So I wanted to follow up on a question I've asked before in a different way, and it has to do with your comments about antigenic spread. So, your titers look pretty reasonable going across the lineages, and but there is some drop-off. So my question, it's really two questions. One, do you know that the vaccine is not, just because you're adjuvanted, and that's going to have some impact on this, do you know that you're not getting epitope shift? I mean, some of the more conserved regions of the spike are in the S2 domain, for example. So do you know that the reason you're getting less of a particular drop-off is because there's a difference in the antibody response that you're eliciting? And related to that, when you look at the studies that have been published on immune serum from people that have been vaccinated with the Wuhan strain versus hybrid immunity, it's pretty clear that it's both and Dr. Marks commented on this, it's both your peak response, your breadth of response, and the sort of rate of decay. So do you know, for example, that the rate of decay is not lower because you're adjuvanting? I mean, this would be a very important point for the public who recognizes now that the vaccine response is waning
And my real question is, because this is adjuvanted, do you know anything more about your rate of decay? I mean, it would really take you 3 time points to know that, or antigenic shifts in terms of subdomains of the spike that you may be eliciting antibodies to.
These are good questions.
Yep. So let me show you the data we have on decay. And since it takes time to develop those studies, they're from our earlier studies, although this is data that we're developing in the 301 study as well. What I'm showing here is IgG responses in the first instance. You see they peak at day 35, they decay over the subsequent 6 months, and then they take a nice boost up to 4 or 5-fold higher with the boost. So I'm not. When I look at the comparable data, I'm not seeing there's any specific advantage in the length of decay. This is IgG. It seems to be dropping at about the same rate. What we just as far as the boosting. What we do know is that those titers that were achieved are quite high. So what I'm showing you here is that same IgG with a third dose, and showing you that the levels we achieved are much higher than was achieved in the 2 phase 3 studies, and that gives us some assurance that a third dose boost is going to be quite efficacious. And if you prefer newts, although our newts and IgG correlate extremely well. Once again, you can see a 5.5 to 5.6-fold increase in newts with a third dose compared to the levels achieved in the 2 phase 3 studies. So that's as far as decay and boosting. As far as your question about what parts of the antigen we see or don't see. So we know we recognize parts of the spike domain that are distant from the RBD. Right? We've mapped that out, and some of the common epitopes, including the original SARS epitope, are found by this vaccine and are utilized. The extent of that and how that matures is something we're working on right now. Maybe I'll stop there, and if there are further questions, I'll need to call into my bench, and perhaps Dr. I'll call my colleagues if we have further questions.
Oh, that's good. Thank you.
Who would you like to call in? Here we go.
Thank you, Arnold. So I just had two questions. One is, can you remind me of Novavax study plans in the pediatric population, and specifically, what experience do you have with the use of the adjuvant in the pediatric population in younger age groups? That's the first question.
So we've concluded the study in adolescents 12 to 17 years of age, and that was in 3,000 adolescents in the U.S. And that's the basis of the licensure we're going to be requesting from the FDA subsequent to our EUA. Our further plans, we have further plans to study this vaccine in school-age children, and then age de-escalating down to children as young as 6 months of age in the first study. Our colleagues in Serum have done this study down to 2 years of age, taking the same adult vaccine dose. And what they found in that study is that the reactogenicity profile stayed very solid. The only small uptick they saw was in fevers, but less than 1% were grade 3 fevers. And the immune responses were favorable. They were much higher than what's seen in adults. This adjuvant is in a phase 3 study being studied in West Africa for malaria. And in that study, the doses have been taken down to children as young as 5 months of age. And once again, they're not seeing a safety problem, although it is a different antigen, obviously, since it's against malaria. But overall, this appears to be quite favorable, and we'll know more as we develop more data.
Thank you.
And I should say that we have agreed upon a pediatric investigational plan and a pediatric study plan with the FDA as well as the EU.
Thank you. My second question is, and I may have missed this, but can you explain? You published the UK data in September of 2021 and the US-Mexico data in February of 2022. Is there a reason that we're talking about this in June of 2022 as opposed to earlier request for EUA?
Yeah, so our first approval was in December of last year, but when the pandemic started, this company didn't have a manufacturing base. So we had to build it from scratch, build a manufacturing network from ground up. What really took the longest time, however, wasn't the manufacturing of the product. It was the generation of the assays to demonstrate that we could make the product over and over again the same way, and to deploy those assays against the multiple lots. But, all of them need to achieve those critical quality attributes. So our approach then was to settle in a single facility in India, because they're the world's largest vaccine manufacturer by dose, and that's a single process we've taken forward. And that's the one that's the basis of licensure globally, as well as the EUA request here in the U.S.
And do you have a concern about having a single manufacturing plant?
There's always risk there, and we have a network, and the subsequent sites are being brought on now. They're done as variations. So first you need to be approved with one, in the instance, the serum. And we're bringing on our sites, one that we own in Europe, and then one of our partners in South Korea. And those are being now applied for in various locations globally. And I have to say that also Takeda and SK are licensees of ours. And they have a different relationship. They're not manufacturing for us. They're manufacturing for themselves. And they're licensed in Korea and Japan, respectively.
Thank you.
Thank you. That helps in some of our considerations. Dr. Fuller.
Yes, thank you. Yes. So a question that I think the public will ask, in trying to ask it in a way that the public will understand, for those who have not been vaccinated, as well as those who may want to use this in some other way, this baculovirus express protein in an adjuvant, we get asked all the time with other vaccines, well, how long does it stay in my system? Could you just share in sort of general language for people who may be listening, how long they can expect this particular baculovirus with adjuvant of the S protein to be in the system to get the response that we want from the immune system? And secondly, if they get this, how long will it take you to make something else, if indeed something else is going to be needed later, as far as a strain of SARS virus?
That's a good question.
Just some practical questions.
Right. And just to be clear, even though we use baculovirus in the manufacturing process, the vaccine contains no virus whatsoever. In fact, the process has been specifically designed to eliminate all virus from the final container, from the final product we generate. All that's in there is the spike protein, the viral spike protein. Now, what we talked about is that the adjuvant effect seems to peak right about 72 hours locally, with a longer protracted effect in the lymph nodes later on. What we saw from our immunogenicity data is it generally kind of peaks at 2 weeks and then goes lower after that. I guess as far as the variants question, we're manufacturing Omicron right now. It isn't clear to us it'll be needed. It isn't clear to us what the public health agencies and the customers and the people will want. We just want to be ready to have that vaccine in hand should it be needed. I know there's a Verb pack coming up later this month to decide or help decide on what kinds of vaccines we should be asking for in the fall.
So in general, with the side effects, the 72 hours expression, or not expression, but presence of the vaccine, does that prolong the time of side effects that people see, or the appearance of the time of those side effects? You had data on that, but could you just restate that, please?
Yeah, no, it's an excellent question. So the major side effects, we followed very, very closely for the first 7 days, and the vast majority were either mild or moderate. Actually, many people had no side effects whatsoever. And the side effects that did occur really resolved after 1 or 2 days. Those are both the local ones, things like pain and tenderness, as well as the broader ones like fatigue.
Okay, so the side effects that can be seen with many vaccines, are they're from the actual injection versus from the 72 long-term hours of antigen being present?
What it is they're likely to be caused by the immune response against the vaccine, right? So not the act of the vaccine being delivered into your arm, but rather the immune response and inflammation, which comes along with the body reacting to the vaccine and generating the protective immune response.
All right, thank you.
Thank you, Dr. Nelson.
Thank you very much for a very thorough presentation and lineup this morning. I wanted to follow up a little bit on the durability question. I'm intrigued by the possibility that it may last longer. To date, the data that has been shown has been with respect to clinical efficacy as well as the humoral immune response. So what I haven't seen is whether you're generating any cellular immune response data with respect to generation of memory B cells and others that might also provide an explanation. And is there anything unique about your vaccine that is inducing a different cellular response that may impact durability and memory response?
Yeah, let's talk about cellular immune response for a bit. Although I don't have kinetic data on that. So I think I'll disappoint you in being able to look at it over time. But this is, oops, that's not what I wanted. Let's try this one. This is data that we published in the New England Journal by Keach et al. And what it's looking at is the intercellular cytokine profile after vaccination on day 28. On the left-hand side, we stained against Th1 cytokines. In this case, it's IL-2, TNF-alpha, and interferon gamma. And what you can see is we got a really nice bump at day 28. On the right-hand side, you can see the Th2 profiles. We looked at IL-5 and IL-13, which is a lesser bump. Importantly, at least we think importantly, when we looked at those that were polyfunctional, so those that either stained for 2 Th1 cytokines or 3 Th1 cytokines, comparing to those that stained for 2 Th2 cytokines, we saw this polyfunctionality. And we think that's important as far as effector memory cells go, although we don't have the kinetic data to demonstrate that fully.
Is that being required? And certainly this Th1 skewing may impact on the observation of lower immediate systemic effects with respect to anaphylaxis and other immediate type of responses, which is favorable with your platform. Right. My second question is related to the distinction between your Hispanic and Latinx efficacy response. I thought I heard this morning that certainly it's been acknowledged as a difference, no obvious explanation to date. I wondered if you wanted to clarify a little bit more as to what your plans are to tease out whether those differences were indeed due to chance versus something else.
Yeah, so we've obviously been very interested in understanding what this data is trying to tell us. And what's even more interesting is when we looked at the racial profile of the Hispanics, they were all identified as Caucasian and not Black Hispanics. So there's something quirky happening in the data. Well, really, our best chance to understand this data is in our effectiveness studies. Those are planned for the U.S., where we will obviously have the ability to probe that in the Hispanic population to understand if there's a real difference or if there's chance finding. I'm kind of a believer in immune responses, and the immune responses in the Hispanic population give me a lot of comfort that it's going to be a chance finding. I have to say, all of the modern severe cases, there weren't any. So even in that population, all the cases were mild.
Acknowledged. Thank you very much.
Thank you. Dr. McInnes.
Oh, that's exciting.
And thank you, Juby, for your support.
Go ahead, Dr. McGinnis. We can hear you.
Hello.
Yep, we can hear you. Take it away.
Hi. Okay, I have a very simple question. It's for Philip. Hi.
I've
searched these briefing documents, but I can't seem to find out what the placebo was. And it's important to me because I'm measuring a delta between that and the active agent. Can you please tell me what the placebo is?
It's normal saline.
Could you hear me?
Yeah, normal saline.
Normal saline. Okay, thank you.
Dr. Meisner?
Thank you, Dr. Monta. And again, thanks for your persistence, because I appreciate it. Question I have is, there was an earlier study, and I might have missed it this morning during some of the clinical trials, but it was done in South Africa, and it included HIV positive subjects. And I think the vaccine efficacy was reported as 50% or something around that in the HIV positive population, which seems pretty good in view of their degree of immunocompromise, I guess, depending on their reconstitution. But can you provide any further data regarding that group and how this vaccine might work in individuals who are immunocompromised for other reasons?
Yeah, and maybe if I could have the immune responses in people with HIV in study 301, please. Maybe we can start there. Yeah, there we go. So what I'm showing you here is IgG responses, and on the left-hand side, you see those that are seronegative. So meaning seronegative, meaning baseline seronegative against SARS. And you can see the HIV levels are higher than those that were living with HIV, although broadly the confidence intervals overlap. Now in the US, these are people who are well controlled on heart and they were immunologically reconstituted. We had a small number of individuals that I'm showing on the right-hand side who came in previously exposed, and you can see they boosted extremely well with the vaccine. So they achieved titers many-fold higher than was associated with protection in the HIV negative group. Now, our data so far on various levels of immunocompromised individuals is somewhat limited, and we're going to be gathering that data in due course. We've completed enrolling a study in South Africa where we looked at giving 3 doses and giving doses on different schedules to see if we can get an advantage, an immunologic advantage by delivering it in that manner. Now, Now, as far as the results in South Africa, Dr. Mallory, do you want to take a crack at reviewing the South African results? Or what our findings were?
Sorry, who would you like to call on?
Dr. Mallory.
Can you hear me? Can you hear me?
Yep. Yes, we can. Take it away.
Yes, go ahead. Yes, so I just wanted to clarify, Dr. Meisner, that I'm showing the results of the South Africa study here, and we did show an overall efficacy of around 50%. Remember, this study was conducted when the beta antigenic escape mutation was circulating. The efficacy in individuals without HIV was 55%. However, we had a very small number of individuals enrolled in this study who were living with HIV. And in that group, we were not able to demonstrate efficacy, and the study was not powered for it. So I think maybe there's some miscommunication. The 55% is in individuals who are HIV negative in that study.
Oh, thank you.
And all cases, again, there were no severe cases in this study, so the vaccine protected all participants enrolled in severe disease in that study.
Okay, thank you for that clarification.
All right, thank you. Seeing no further hands raised, we're able to move to our next phase of our discussion, and that is the committee looking at the question that we are going to have to vote on shortly, and that is whether we recommend emergency use authorization for the Novavax vaccine. And there is the voting question. I'm going to be officially reading it later on, but just to remind you, this is for the 2-dose series, and it's based on whether the risks outweigh the benefits. So any of you who would like to start the discussion, please raise your hands. We don't have to fill the full 2 hours in if a lot of our questions have already been answered. Dr. Rubin.
Well, I don't want to fill the 2 hours, but just so
you don't have to.
Very simply, then, I think that the data that were presented looks very similar to the data that were presented for the mRNA vaccines that we approved a long time ago. And in fact, that's in part because those trials were done at the same time. But I think that the efficacy is
I had the same
similar.
feeling, Dr. Rubin. It was deja vu.
And you know, if we're going to use the same criteria that we did then, I think that it's not that difficult a decision now. It is disappointing, and we've discussed this already, and we're willing to take this and not willing to take the existing vaccines. I think it's pretty compelling.
Thank you. I'm amazed. I see no hands raised. Anybody who doesn't feel that this is compelling? Dr. Sawyer.
Go ahead, Mark.
Yeah, I'd just like to sort of reiterate the previous comment. It is quite disappointing that we don't have any data in the Omicron era. Clearly, such data could have been presented, but I will follow what I understand is the FDA guidance, which is we're supposed to evaluate this vaccine based on the data presented to date and leave it up to them whether they actually issue the EUA, given the lack of data about Omicron effectiveness, at least as presented on the committee. So I do agree with the previous conclusion that the data that was presented is quite similar to what we've approved in the past with other vaccines.
Thank you, Dr. Reingold.
So I agree with both of those statements, and I certainly will support recommending to FDA that they approve this vaccine. I'm a little skeptical about how many of the vaccine hesitant are just waiting for this vaccine and are going to be convinced that this is better for them than the vaccines that are currently available. So obviously there are individuals who testified to that, but at a population level, I'm hoping to be proven wrong, and that the large numbers of people who sign up for this vaccine who wouldn't take an mRNA vaccine, but count me as skeptical about that. And I do think that it remains to be seen just what the risk of myocarditis is, but we know that certainly has dissuaded some individuals from getting the mRNA vaccines, and it looks like it's likely to be the case that we'll see at least comparable levels following this vaccine.
Thank you. Dr. Gellin.
Thanks. So having been in these discussions before, I know the FDA selects their words very carefully. Could you put it back on the screen? Because I think that the question about the totality of the data available, we've only seen a subset of the totality of the available data. And there's a lot of other data that would help to inform this decision in use currently and going forward that we haven't seen. So maybe they want to talk about totality of evidence. available.
I can tell you, Dr. Dillon, that is the wording that's in the, has been used before, and I think is taken from the regulations, but I'll let the FDA respond. Please.
So we would consider the totality of data available to consist of the data that has been presented and discussed at the meeting today, and primarily the data that have been reviewed and independently verified by FDA as outlined in our briefing document. I think it's important to make sure that the committee members and the public understand that in response to some questions by committee members, Novavax has presented some additional data that FDA has not covered in our briefing document. And the reasons for this are several. First of all, as has been mentioned several times before, we view that there are important manufacturing differences between the product that was studied in the U.S.-Mexico trial and the product that was studied in previous trials, and that in our assessment, due to inherent limitations in product characterization for this platform, we just cannot conclude comparability of those products that would allow us to consider those data. That being said, I think we have laid out a case in our briefing document to support why we think that the available data from the U.S.-Mexico trial could meet the statutory criteria of may be effective that is required to support emergency use authorization. And that rests primarily on the efficacy observed in clinical trial 301 that was conducted in the U.S. and Mexico, and considering those data in the broader context of what we know about other COVID-19 vaccines that were evaluated at the same time and how they have performed in real-world use, including against currently circulating variants. There are additional immunogenicity data that Novavax has presented as well that we did not review or discuss. Some of these relate to binding assays, IgG binding assays that we have not used as the basis for regulatory decision making for any of our EUA decisions, and also come from clinical trials outside of the data that we are really considering in support of this 2-dose series for use in adults 18 years of age and older. And I see that Dr. Marks has also turned on his camera, and he might want to add some additional content.
I think that this issue of why we're seeing a limited amount of the whole picture presented is because we needed to feel comfortable that the process that was used to make the product that was studied was one that we were comfortable with, and that it was one where we felt that going forward, what you would authorize as a committee would be what we would expect to see. Now, granted, it will be in a different era, perhaps, but that what you're seeing is the product that you're getting, and that is the reason for focusing on the manufacturing process that came from the facility that is producing the product currently. We take manufacturing very seriously. I think it's very important for the public to understand that we don't benchmark ourselves against other countries when it comes to manufacturing. We consider that we have a very high standard, and it's why we're often considered a gold standard for our manufacturing. And particularly in the area of vaccines, we owe it to the American public to make sure that we have the highest quality of vaccines. And that means that whether it be in any aspect of this, including whether we will allow release of lots of vaccine to be used, we will need to see the data that supports that before that can actually happen for any vaccine here in the United States. So I think it's important to understand that I fully respect the sovereignty of other countries to release vaccines based on what they see as their benefit risk. But we have certain standards in the United States that we hold to because that is the expectation of the American public. Thanks.
Thank you, Dr. Chatterjee.
Thank you, Dr. Monto. My question actually is for our FDA colleagues. With regard to any kind of cautionary language that would be included were this vaccine to be authorized with regard to the risk for myocarditis, pericarditis, cholecystitis, these quite rare but serious adverse events that seem to be associated with this vaccine.
Yes, thank you for that question. So that is something that is a question that we are discussing as we have been reviewing the proposed EUA fact sheets for this vaccine. And so what I think you might be hinting at is, do we include something along the lines of a warning statement, similar to what we have in the currently authorized and approved mRNA vaccines. And so the regulatory criteria for including a warning statement is to have reasonable evidence of a causal relationship. Now, certainly, I think we can all agree that the extent of evidence for myocarditis being causally related to this vaccine is not at the same level as for mRNA vaccines, where we have many more cases described among much more extensive use of the vaccine. But I would actually like to get the perspective of committee members to weigh in. What do you think, based on the data that you've seen presented for these myocarditis cases, is your impression about the likelihood of a causal relationship, and whether you would see a warning statement being appropriate in this situation?
I actually
Dr. Chatterjee, you're there. Please answer.
Yes, and you're absolutely correct, Dr. Fink. That is what I was alluding to. If we go back and recall the data that were presented initially for authorization, we did not have this concern. It really became evident after the mRNA vaccines began to be used much more extensively. So that often happens, as we know, with vaccines. And so in this instance, we have an indication that there is a potential for these adverse events to occur more as this vaccine gets utilized. So I would be in favor of that type of language being included so that the public is clear, vaccine providers are clear about the risks and can speak to them with their patients.
And since my picture is up there right now, I will say that I agree as well. I think there's a question, and there will be answers, but we must be aware. Dr. Dabrowski.
Yeah, I just thought should hear our perspective on this. It's important to convey an accurate level of risk for the available data. We believe there's insufficient evidence to establish a causal relationship. But we're not really that far from where the FDA is. And as we enter, the final label negotiations, I'm sure we're going to come to a closure on this. Each regulatory agency reviews our clinical and our post-marketing data, and they come up with their own conclusions. And that's what informs their labels. So we completely respect the approach that the FDA is taking.
Any other questions? I see a number of hands raised. I'd like to settle this question, at least in terms of the committee's opinion about the myocarditis issue? Dr. Levy, is that what you're going to be talking about? You had your hands raised before.
Hi. Hello, can you hear me?
We can.
Okay. I had a question for FDA regarding the placement of this vaccine in the broader context of the approval.
Dr. Levy, we wanted to settle the committee's views of the. Okay, did you have anything to say about that? Because then I'll come back to you.
Arnold, can I make a comment?
Yes, please. Go ahead.
Yes. On this risk, I have such a hard time with this problem, and as we all, and there's been such variation in reports of the rates of myocarditis following administration of these vaccines that I think it's very hard to say that it occurs more frequently. It would be, at this stage, difficult to say it occurs more frequently with one vaccine platform than with another. I mean, I think if because, you know, if you look at the Israeli data, it's pretty high. It's higher than the numbers we're seeing here, and they may have better capture of rates of myocarditis for the messenger RNA vaccine. So I don't think we can have enough confidence in the rates because it's such a range. And I think that any statement regarding the risk of myocarditis should be standard between all of the COVID-19 vaccine platforms. I think there is clearly an association, but to try and make a gradation as to whether one platform is more likely to result in myocarditis than another, I don't think we have the numbers to make that statement.
Very interesting
statement.
Thank you,
Dr. Meissner. Yeah, sorry, I agree with you that at this point we don't have enough information to really describe relative risk of this event between different vaccines. But that's not a requirement or a necessity to have a warning statement. A warning statement is justified by, and I'm going to clarify what I said earlier here. A warning statement is justified by reasonable evidence of a causal association.
And you should
and there does not need to be definitive evidence of a causal relationship. It's reasonable evidence of a causal association. And so that really is the question that we're looking for input here. Based on the information that you've heard today, do you consider there to be a reasonable evidence of a causal association? And of course, it would be ideal if we could describe the magnitude of the risk for each vaccine and compared to the others, but we don't have the ability to do that, at least not for this vaccine just yet.
Thank you for that comment, Dr. Fink, and I completely agree with what you said. But I think my point is,
okay, is there anyone on the committee? Is there anyone on the committee that does not agree with that comment? Dr. Gillen, do you have your hand raised? Do you disagree?
Well, my hand is up. My hand was up before you changed the question. So let me just give you the answer before. It's on.
You have to be nimble on this committee.
I got it. On this topic, though, I want to support what Cody raised and what Dr. Fink supported. But I think we also have to put this in context that we talked about earlier about myocarditis that comes from natural infection as well. So people can look at that and weigh those as well. And then most importantly, what Paul Offit raised earlier, that this is a priority question to answer mechanistically. Over.
Okay, I think you've got the message, Dr. Fink, that there is a concern that the topic be further investigated. And it's in your hands in negotiations with the sponsor exactly how that is to be done. But we do agree that there is a concern here.
Okay, thank you.
Can I ask?
Okay.
Dr. Fink, don't you agree that there's a likely association between the messenger RNA vaccine and the Novavax vaccine? And shouldn't it be a standard statement?
Well,
Yes.
What we say in product labeling, including EUA fact sheets, needs to be supported by the available data. And the level of evidence is going to be different for different vaccines. I think we're in a different place and can say more for the mRNA vaccines at this point in time than what we can say for this vaccine.
So you're going to put it on the mRNA
Okay,
vaccines?
Moving on. Dr. Levy, I interrupted you when you were ready to make another point.
Yeah, my question here, can you hear me?
We can.
Yeah, my question here is to FDA. In my view, we've seen great presentations today establishing reasonable safety, demonstrating efficacy, at least against the variants that were circulating at the time this vaccine was evaluated. Now, as we vote to potentially recommend authorization of this vaccine, where does the whole topic of how to place the vaccine in a public health strategy play into this? In other words, it's a very different landscape now than it was half a year or a year ago. There are the well-established mRNA vaccines, some of which are not just authorized, but approved, as this committee knows very well. So is there going to be a pecking order for the mRNA vaccines where there's much more data about level of vaccine efficacy against Omicron? Are those going to be the preferred first-tier vaccines to use? With the Novavax, if and when it's authorized, we've heard a lot of talk about people who might not want to trust or partake in the mRNA platform. They might want to try a different platform. So don't get me wrong. I'm a fan of this vaccine. It has a lot of attractive features, doesn't require freezing. The adjuvant is intriguing. You might get more bang for a buck with that. But where does this get placed in the armamentarium? Because in isolation, this vote would almost imply that it just takes an equal spot on the shelf. But we know that it's more complicated. So, Peter, where does that stand, and does FDA speak to that, or is that just a CDC matter?
So I think we speak to making available another option for those who might not otherwise take a vaccine, because right now, any vaccine, even one that may need to be updated for the variants, right now getting that into someone's arm who has no vaccine is probably going to prevent them from having serious outcomes such as hospitalization and death from COVID-19, even from Omicron, we hope, at least for a period of time. So it's having additional choice. That said, my guess is that CDC will have some discussion here around this as well, about how they might position this. And I can't say how they'll come out on this from ACIP. Of course, Peter. From our perspective, it's making available another option to hopefully get some additional people vaccinated.
Yeah, it makes sense. But in the past, the committee has been asked to take votes on very specifically worded for certain age groups, for certain scenarios. This is a pretty broad statement. You're not crafting a vote question that says for individuals who are reluctant to take mRNA. It's a broader statement. That might be fine, but I'm wondering, did you consider to phrase the question more narrowly, or you want this broad phrasing?
This is the question that we voted on originally back in a year and a half ago.
I think the issue is there were no data for us to suggest that there was a reason to narrow this further at this point in time in terms of adverse safety concerns that might want to make one narrow this. And I'd certainly invite Dr. Fink, if he wants to add anything to that, to add it. But I think that in the absence of data suggesting that a narrowing was necessary, we have asked the broader question here.
Yeah, I will just echo what Dr. Mark said. The more restricted authorization for the Drbeen vaccine was precipitated by a specific safety concern related to thrombosis with thrombocytopenia syndrome. Here, we have a package of data to support broad use in the general population of adults 18 years of age and older. We did not identify a specific safety concern that would cause us to think about a more restricted use of this vaccine, and so that's why the voting question was constructed.
I got prove that all other things being equal, we know more about the efficacy against Omicron of the mRNA than this vaccine. I'm a supporter of this vaccine. I'm just saying, in terms of messaging, it's tricky, isn't it?
We all agree it is tricky. Dr. Perlman, thank you.
Yeah, I just have a question about something we actually didn't talk about much. So this vaccine doesn't, for the most part, induce a CD8 T cell response. It's mostly CD4 and antibody, and that's what was discussed. How does the FDA take that laboratory information? Does it consider that the vaccine clearly works, so maybe it doesn't matter, but I'm just curious how the FDA puts that into its equation in going forward.
I'm not sure there's really a lot to say there. I mean, I think it's something we were aware of, but given the clinical data, that's what we're hanging more of the hat on here. Doran, I'll pass it over to you.
No, I think we really have to look at the clinical data here. It's interesting to see and discuss this data on cellular mediated immunity to the extent that it is available. We don't have a sufficient enough understanding of those data to use it as the primary basis for making regulatory decisions. And so really I would ask the committee to focus on the clinical efficacy data that has been presented.
Right.
Thank you. Seeing no further hands raised, I would like to turn the meeting over to Christina Burt, who will start the voting process.
Yes.
I'd like to remind the committee that after the votes are
My vote is
completed and reread,
maybe label.
we will allow time for those who wish to explain their vote to do so. You don't have to explain your vote if it's clear to you and to the group, but that time will be made available afterwards. So we move to voting.
Thank you, Dr. Monto. Can you hear me okay?
We can.
Okay, great. Only our 10 regular members and 12 temporary voting members, a total of 22, will be voting in today's meeting. With regards to the voting process, Dr. Monto will read the final voting question for the record, and afterwards, all regular voting members and temporary voting members will cast their vote by selecting one of the voting options, which include yes, no, or abstain. You will have 1 minute to cast your vote after the question is read. Please note that once you have cast your vote, you may change your vote within the 1 minute time frame. However, once the poll has closed, all votes will be considered final. Once all of the votes have been placed, we will broadcast the results and read the individual votes out loud for the public record.
I agree with you. I think this is what's going to happen.
So, and also wait till I say start the vote. Does anyone have any questions related to the voting process before I begin? And also, do you feel you need more than 1 minute to cast your vote?
You got a minute to vote.
If you need
Vote
more
before
time,
voting.
or if I need more time to check things, we will continue to keep the vote open for the 2 minutes.
Marco says no.
Okay.
Okay, I'll read the question. I'm sure that you will see everybody voting within the minute, and you'll know.
Yes, Dr. Monto, please read the voting question.
Based on the totality of scientific evidence available, do the benefits of the Novavax COVID-19 vaccine, when administered as a 2-dose series, outweigh its risks for use in individuals 18 years of age and older? So there is the pod.
Please start voting at this time.
Okay, so they're voting. My vote is yes. I may have restricted generally for information that younger men know, but interesting vaccine. Brandon says, how do I, this is just weird talking here. They are voting separately. You got 1 minute. If the votes, I believe no votes are more, then it is not authorized. No, Tracy.
Okay, I'm just checking the votes.
I think you're gonna get it in a few minutes.
Okay, it looks like all the votes are in. We can please end the vote, and then we can broadcast the results.
See, yes, 95%, right? 95%, 0 no, and 1 abstinence. Done. Approved.
We'll close. Okay. Viewing. Okay. All right. The majority, so there's 22 total voting members again today, and we have
1 abstinence.
We have 21 that have voted yes, 0 have voted no, and 1 has abstained. So the majority have voted yes. And I will read the voting responses of each voting member for the record. Hold on a minute. Okay. Dr. Fuller, yes.
1 abstained.
Dr. Berger,
no, 0
yes.
no's.
Dr. Cohn, yes. Dr. Chatterjee, yes.
Abstain
Dr.
is
Monta, yes.
Not positive, not negative.
Dr. Reingold, yes. Dr. Jelen, abstain. Dr. Meisner, yes. Dr. Kim, yes.
Yes, yes.
Dr. Rubin, yes. Dr. Bernstein, yes. Dr. Portnoy, yes. Dr. Lee, yes.
Finally.
Dr. Sawyer, yes. Dr. Morton, yes. Dr. Nelson, yes. Dr. Levy, yes. Dr. McInnes, yes. Dr. Offit, yes. Dr. Perlman, yes. Dr. Pergam, yes. Dr. Morosco, yes. And that is everybody. Yes. Okay. That concludes the voting portion of today's meeting, and I will now hand the meeting over to Dr. Monto for asking the committee for their voting explanation. Thank you.
So anybody who would like to explain their vote, please raise your hand now.
Correct, right.
I do not. Can you hear me? Yes. Am I missing anything? I got kicked out of the. Can you hear me, Arnold?
And committee falls into
I can hear you. I can't see you. I don't know. Something I got kicked out of the meeting, but you can still hear me. So I'm trying to get back in. But you want me to explain mine, or how do you want to proceed? I want to. It's up to you. If you want to explain your vote, please. Oh, I'd love to. So let me just say that this is a conditional yes, and I'll explain that. But conditional yes wasn't an option. I will say that this is a case study of perseverance by the company, and there's nothing about vaccine development that's easy. And the vaccine race that's inspired by COVID, and it was supported by Warp Speed, and I had nothing to do with that. Has brought us vaccines that we didn't think they would have to have an impact that has been impressive. And while global inequity remains, for which additional platforms and more user-friendly presentations will be welcomed, like this vaccine, that's not why we're here. The data that we've heard today and seen today has been impressive and support the original vision for the vaccine from its beginning, that it would provide safety and efficacy and a presentation that didn't require extraordinary logistics. And with attribution to the novel adjuvant, the lower amount of protein appears to make it even less reactogenic. With a focus on safety, as we discussed, myocarditis is a signal that many are paying attention to, and attention to this by the company and the government is critically important. As I said before, highlighting Dr. Offit's intervention, that we need to understand the mechanism here, because this infection and the vaccines against it are going to be with us for the foreseeable future. The question that we're asked is based on the totality of the scientific evidence available. You've already heard me ask about the availability word. And as Dr. Marks reinforced, in looking at the totality of the evidence presented, we can clearly say that it was, in general, safe, including the long-term safety follow-up in the study. And because of its effectiveness, it helped to prevent serious consequences of the viral infection. But we don't know whether that attribute continues to be relevant today. Dr. Levy's important question about the potential of cross-protecting immunity and the limited data that we've seen in response to that are certainly encouraging. But again, we don't really know whether it's likely to be effective going forward and what the duration of that protection might be. In the flu world, we're always challenged by mismatch. Is the vaccine that's being made and distributed likely to be a good match for the flu virus that's likely to circulate? This vaccine, and that's essentially the question here, this vaccine has incredible potential. And a lot has been learned about it that we didn't hear about. That's likely to inform the durability of protection, transmission, the impact of boosting, adjustment to the dosage interval, the impact of mix and match, and it's importantly, impact against circulating variants. So therefore, I want to be clear that I'm not voting against this vaccine, because I did worry that such a vote would be misinterpreted, and hence this conditional vote for it, but as an abstention. But as this is a real product that, if authorized, will be used, and it'll be important to evaluate whatever data is available that can give its insights into its performance, not just voting on the science that tells about its promise. So recognizing we're an advisory committee and we're advising FDA. And we know that FDA, as we heard from Dr. Marks and others, will continue to work with the company on some of the manufacturing issues. And our discussions today are just part of what they will consider going forward in their discussion, in their decisions on authorization. So my conditional vote yes is based on my expectation that the FDA will review the totality of the data that will be available to them,
including the data that we didn't see today, to inform their authorization decision. Thanks.
Shout out to all of us.
Thank you, Dr. Gillen, Dr. Nelson.
Thank you, Dr. Monto. And certainly with the question posed before us, do the benefits outweigh the risk? I'm entirely supportive of a yes there. It does come with a little bit of caveats because included in that question was specific reference to the 2-dose primary vaccine. I think this group was in full recognition that this is probably a 3-dose series and that they'll need to accumulate data supporting the need for booster doses and subsequent doses to probably make it a 3-dose vaccine. But to address the question on the table, certainly the benefits outweigh the risk for a primary series. I also want to make reference to use Dr. Mark's words from this morning that this vaccine does indeed fill some unmet needs. So you didn't ask us specifically how to apply these to the EUA criteria, but I'll offer my humble opinion in that I do feel that it does offer something for fulfilling unmet needs, including those populations who have hesitancy with regards to the messenger RNA vaccines. As an allergist, it offers me an additional tool for individuals who have hypersensitivity responses to initial doses of the messenger RNA vaccine. And there are other advantages that have been referred to today, including storage. Who knows, even with supply chain challenges down the road, it will be nice to have these options going forward. I'll offer one other additional word with respect to myopericarditis. I've done some work in the Department of Defense, and we'll be publishing our work on long-term outcomes of myopericarditis with the smallpox vaccine shortly. This is an important question and should not be ignored. And I will say, Dr. Fink, with utmost confidence that it would be a travesty if we didn't mention it in the EUA documentation for the public to show the concern that we have. Is there evidence that it's a true causal link at a significantly high relative risk? I have my own doubts there, as we've heard from the sponsor as well. But to be silent on the matter, I think, would be a travesty. I also think we should be focusing on the mechanism, as has been discussed, but also to put more effort into identifying what happens with subclinical appearance of myopericarditis. Our signals are those who get admitted to the emergency room in the hospital. I'm quite convinced that there are others who are experiencing cardiac events of a lesser severity that are worthy of being studied, both from a mechanistic and outcome standpoint. So we have a lot of work to do, and I hope this committee and the focus of the FDA and the NIH remain on myopericarditis with all vaccine platforms. And I appreciate the opportunity to express this opinion, Dr. Monto.
Thank you, Dr. Nelson. Dr. Portnoy.
Oh man, now everybody's going to go through their comments.
Thank you. Yeah, I was a little bit torn when I first started this committee this morning. I was a little bit skeptical about the need for an emergency use authorization of this vaccine, since we have 2 other vaccines that are highly effective and relatively safe. And so I was very skeptical about that. We've had those vaccines for a year and a half. If this vaccine had come up for discussion a year and a half ago, there would have been no problem at all getting it approved. I'm pretty sure the committee would have just voted enthusiastically yes. But now we've got these other vaccines. Is there really a need for an additional vaccine? So that's what I was torn about. But I realized that this is a different technology. It's a more traditional protein-based vaccine. I'm very skeptical that vaccine-hesitant people will select to get this vaccine because of that. I'm good friends with a number of vaccine-hesitant people, and their hesitancy is more ideological than technological. So I really doubt that this vaccine is going to crack that nut, but perhaps some individuals would get this when they wouldn't get the other ones. I see this as an opportunity to widely vaccinate people with the protein vaccine and to compare it with mRNA vaccines, which are relatively new technologies, because we know how protein-based vaccines work. We don't know how mRNA vaccines work. This is an opportunity to find out how they compare to each other over the long term when large numbers of people get vaccinated. So I see this as an opportunity. I agree that the benefits definitely outweigh the risks. whether it meets the needs for emergency use. I'm not totally convinced, but I feel that it's at least it was worth voting yes in this case, because the vaccine deserves the opportunity to be given and studied and used by individuals who wish to use this vaccine. Thank you for having such a transparent and open meeting, and I do want to thank the organizers of this meeting for holding it the way that you do. You do an excellent job, so thank you.
Thank you, Dr. Meisner.
Oh man, everybody's going to speak now.
Thank you, Dr. Montel. I just want to note that the messenger RNA vaccines are truly remarkable. I mean, they are a great gift to humanity, and they were the first to cross the finish line. But whether or not they will turn out being the optimal vaccine for these viruses is not clear. And I think I also want to recognize the perseverance from the people at Novavax for developing this vaccine with a novel platform, because I think we still need new vaccines. I don't think we want to rest on just what we have at this point, because there's always an opportunity to improve on a vaccine. And we've talked about several of those issues, such as sterilizing immunity and the duration of the immune response and the breadth of the immune response. And so I certainly think we want to continue to encourage the development of new vaccines, despite the wonderful spot that we find ourselves in today with the two messenger RNA vaccines. And I would also, just in response to Dr. Nelson's comment, again, just want to reiterate, I agree there does appear to be a causal association with the Novavax vaccine, but there's a causal association with the messenger RNA vaccine also. So my point is, I don't want to stigmatize this vaccine inappropriately relative to the messenger RNA vaccine. Thank you.
Thank you, Dr. Meisner. Dr. Marasco.
Yes, so I think to the question posed today, I think that the benefits certainly outweigh the risks. My sort of, I voted yes because I feel that that's really the question that we were posed. I remain somewhat concerned about, the timing of the rollout. I know many of you have to be similar to me. The public knows that there's talk amongst the FDA about reformulating the vaccines in the fall to be more Omicron-centric, if you will. And the real question is, for the people that are vaccine hesitant, are they going to say, great, we finally have a protein-based vaccine like we're familiar with? Or is the question going to be, but should we do it now with an ancestral strain or wait until the fall when the company itself has said they're investigating it? So I think, on balance, we need to get these new vaccine platforms out there. I think there's some certain advantages to the adjuvanted vaccine that I'd like to see more about as we get more data. But it is a concern that I have in my mind about, we're rolling this out. We're having a discussion 2 weeks before we're having another discussion about formulations for the fall. And although no decision has been made, it'll be an active topic of discussion. So, overall, I applaud the company for having the perseverance to getting this platform and the vaccine out. But there are some questions I think remain in my mind.
Thank you, Dr. Marasco. That's the last explanation of vote we have. I would like to turn the meeting over to Prabha, who will ask Dr. Marks to give some closing remarks, And thank you all for a very vigorous and productive meeting. So over to you. I think you're muted.
Yep, probably you double muted.
Yes, I'm sorry. Thank you, Dr. Mancho. Dr. Mark, do you want to address the committee and make some closing remarks, and then we can adjourn the meeting?
Yeah, no, thank you very much. First of all, I want to thank the committee members for a very good discussion today. Also want to thank the sponsor, the open public hearing speakers. Again, they all contribute to what is an important open process, transparent process here. Really appreciate that. And we will do our best to continue to work towards keeping technical glitches down to a minimum. Thank you for your patience with those. I also want to thank Dr. Atreya and the advisory committee staff. They did a wonderful job preparing things for this meeting, and then the entire clinical team and the others that were involved from the various offices in the center preparing for this advisory committee, which took a lot of work. And as you're aware, there are some coming attractions of additional ones, so there's been a lot of work going on. Thank you to everyone for that. Thank you to those who have tuned in today. We very much appreciate that. We will again look forward to working through what's been said today and moving forward. And just appreciate everyone's input today. Prabha, I can turn it back over to you. Thank you again to everyone.
Okay, thank you, Dr. Marks, and I would also like to extend my thanks to Dr. Arnold Mancho for conducting the meeting very smoothly, and then also all the members who have been patiently working together. And so it's such a productive meeting. Thank you so much. And I also thank Mike Kaczynski for facilitating this meeting, and Christina Ward for doing the voting process very effectively. So thank you, and this meeting is adjourned now, and have a good evening.
So just to close it, yes, it is recommended with these votes. It is recommended. And now I think FDA would follow up in a day, 2 days, or 3 days to say EUA granted. I don't think they'll have a reason not to grant it. So with this, thank you very much. Thank you for being here with me today. We've been hanging out since the morning. So I would see if I can prepare something to have a discussion, or this evening, maybe the discussion can be Novavax related questions, open forum. We just discuss like we do in chit chats, and I open up their documentation and we go over whatever data. If that is interesting, tell me. Even when I hang up, you can actually comment and give your opinion. Otherwise, I'll see if I can prepare something in the given time. I have to look after some of my business matters as well. So we may or may not see each other, but if there are questions, we can start our discussion this evening. Thank you very much, and see you later. Bye-bye.